over 100,000 independent plant lines. These are primarily transgenic plants that were either expressing an antisense construct to knock out a gene or overexpressing a protein to see how overexpression affected the plant. And basically, it really became the industry standard for plant phenotype analysis and gene function analysis. The one paper that we published some years ago in Plant Cell that described the overview of this platform is still cited. It's got over 1,000 citations, and it still gets cited several times a week by researchers who use our phenotyping platform or phenotyping strategy in their experiments. So this is just a high-level overview of the gene function factory. This is what was put together in North Carolina. So this was the building, and inside the building we had one very large open lab that was surrounded on all sides by large walk-in growth rooms. So the plants were grown around under different conditions, whatever was happening for that particular assay. And then the plants would be brought out to these stations and analyzed. And you can see some trays of plants here, or carts of plants here. This was all modular. We could move the benches around and construct the space any way that we wanted to facilitate moving plants efficiently through the analysis process. So as far as the process, we had teams that were focused on key aspects of doing the experiments. So we had a cloning team that would crank out up to 100 or 200 agrobacterium clones and get those clones into agrobacterium tumefacients. We had a transformation team that would do all the plant transformation and identify the primary transformants and then validate that they were indeed the plant had the gene construct that we hoped that they had. Then it was transferred to the analysis group, and we had sort of two teams there. We had a team that did plate screens, and by plate screens, I mean looking at young seedlings grown on artificial media in Petri plates. And we had 8 different assays that we ran as plate screens. And then we also had 2 soil screens that we used. And we had a big team that just dedicated itself to growing the plants and maintaining the plants. All of this was controlled by the laboratory information system. So all the plants were tracked. We initially used barcoding systems, but of course now you can use RFIDs to track things. The data linked to samples was collected, put into what we call the data refinery. So that's where we would do all the basic statistics, determining averages, determining statistical parameters, and coming up with data that we could then look at and start to mine. And this was then dumped into our what we called the gene function database. And in this case, this is where we did a lot of data integration. So for example, if we were looking at gene expression and metabolomics data in conjunction with the phenomics data, that would all happen in this database. At the heyday, when we were running at full speed, we had about 150 scientists working in this laboratory. So this is just one example of a screen out of the LIMS system. This represents how we would collect data. We had a lot of pull-down screens that the scientists could use to answer very direct questions. This was very important because it allowed us to have essentially a controlled language, and all the data collected was uniform. So we didn't have people writing down, wow, the flower looked like this or the flower looked like that. They had to choose from specific phenotypes that could then be captured and then quantitated. Wherever possible, we always collected quantitative data. So we measured things, we weighed things, and so on. This LIMS system actually, it didn't take 15 years, but it represented 15 person years of development and customization. As I mentioned, it's very highly integrated into laboratory operations. So at the very beginning, whatever batch of soil was being used was tracked. What lot of plant seeds were tracked. Everything had a trail. So throughout the entire process, we knew where plants came from and what the data linked to those plants was. It was integrated with a number of instruments for image collection, and we used robotics wherever possible. And we could have up to 50 concurrent users inputting data. Again, when we were running at full speed, we were collecting about 1.5 million data points per month out of this platform. So here, I just wanted to give you an example of some of the image analysis that we could do. So we would, we had specialized software where we could just take a light image as shown here. This is under a stress assay, so the plants are accumulating anthocyanins and showing some yellowing. And what we could do then is do a color analysis, and we would just bin what percent of the plant was in these 5 different color buckets, and then we could quantify that. So this gave us a very quantitative assessment for, in this case, for how stressed the plant was. And based on the color combinations, we could also extract some data about what was potentially going on with that plant based on the color change. So for example, certain mutations might really increase the anthocyanin, and so you would get a lot more of a reddish, purplish colors, whereas other times you would just see a lot more yellow. So this was actually, putting it into these color bins was actually very informative. In other screens, we also had the ability to calculate areas. So in a different type of analysis, we could actually calculate the area for the entire seedling. That gave us a measure of growth as well. As I mentioned, we had 8 different plate screens that we ran on these Petri plates, and we had 2 soil assays. So as far as the soil assay, we had a control condition, a non-stress, and this is where we did a lot of the basic biomass and yield measurements. But we also had a drought assay. Of course, drought was a very interesting trait for our partners, Monsanto, Bayer Crop Science, as well as Pioneer. Drought can be a big issue for farmers. And the stress assays, we had, of course, our control, non-stress condition. We had a osmotic stress that we induced using PEG. We had a high salt stress, a cold stress, a heat stress, growth under low phosphorus, growth under low nitrogen, and growth under low light. So these all represented traits that we had validated with the agrochemical companies that these were things that they were very interested in. So here's just some of the phenotypes that we observed in these plate assays. These are the abiotic stress phenotypes. So these are what the plants look like after a couple weeks grown under this control condition, under standard conditions. This is the heat stress phenotype. This is the salinity stress. Under phosphorus, you can see they get these very pronounced, short but fuzzy roots with lots of lateral root formation. Under nitrogen limitation, you get very long roots, again with increased lateral root formation. And then under cold stress, you get these more slower growing plants. So these phenotypes were very reproducible. You can just see on this plate, on these plates, we actually did a fair amount of work developing the methods for preparing the plates and making sure the media and everything was very consistent. So over several years of doing experiments, we could see a very highly reproducible result in these various assays. And that was key, really spending the time to putting in the control processes and the quality control processes to have a highly reproducible system. The other key thing that we did was we really focused on analyzing our phenotypes using a growth stage based system. This was a system that was based on the BBCH scale that was developed by scientists from Bayer, BASF, Ciba-Geigy, and Hoechst. Rather than just collecting data at certain times after the seeds were plated, we actually characterized what developmental stage they were at. So in our plate-based assays, some of the key stages that we captured or monitored was when radical emergence, which was given the number 0.5, the time at which the cotyledons were fully open, when they had 2 rosette leaves, when they had 4 rosette leaves. Under the control condition, so then on different days, we always did standard data collections. And so, for example, at day 3, we always looked at seed status, had it germinated or not, the color, the shape, the size of the seeds. Days 4, 5, and 6, we would look at what growth stage using this chart here. 8 through 14, we continued to monitor the seedling status, look at color, shape, size, again, do the growth stage determination and measure root length. On day 12, we counted the number of lateral roots. And then on day 14, we would look at the root growth vector, root shape, fresh weight, and an image was taken and the fresh weight of the plant material was taken. On the soil-based analysis, we looked at about 50 different attributes. And again, we were using this growth stage-based assessment system. Here are just a few of the additional steps. The early steps, the 1.04, the 110, when they get 4 leaves, 4 true leaves, 10 leaves, 5-10, fruit when the flower is first available, first visible, and so on. So as we did with the plate assays, at different days, we would harvest, do a rosette image, and do tissue harvest. And the harvest would be for gene expression profiling or metabolic profiling. And we would do this throughout the entire growth cycle of the plant. Later on, we collected a lot of information on the flower because of importance for yield. In Arabidopsis, you get these seed pods, that are called siliques. So we always counted the number of seeds in siliques, how many siliques were produced, the size of the silique, and so on. And then at the end, we would essentially do our yield. So we would look at seed mass, look at the quality of the seed through a seed image that would look at shape, size, and color. We'd also do biomass measurements to get vegetative yield as well. So the key thing here is, again, remember what we're putting through this platform are transgenic lines or different mutants that are altered. And in many cases, these mutations would alter the progression of the plant through these growth stages. So a key thing of using this growth stage based approach, when you're trying to integrate phenotype with metabolomics and genotype, is that you want to make sure that you're collecting tissue at the same developmental stage when you're trying to compare 2 different mutants, not the time after sowing. And I'll show you the importance of that shortly. Because you can imagine if a plant is delayed in development and you compare gene expression in leaves from these 2 mutants, the differences that you might see may have nothing to do with the mutation. It would have to do with the fact that the plants were simply, one plant was growing slower than the other, and it was at an earlier developmental stage. So you can see that here. So in this figure, what I'm showing you is a bar graph that marks when a plant is hitting certain developmental stages throughout its life cycle. And down here is the days. So Columbia is the wild type genotype of Arabidopsis that was used in all of our studies. And this is just a series of mutants. Hookless is an ethylene response mutant, for example. So these are all different types of mutations that are well characterized. And as you can see, all of these mutants, their progression through this developmental pathway are all altered from Columbia. In some cases, it's rather subtle. So hookless, this brown growth stage here is a little bit shorter than wild type, but otherwise it's pretty similar. However, this other one, the FAE1-1, this is a fatty acid elongase mutant. It actually starts to flower earlier before it even reaches the 110 stage. So this, what we're calling the brown growth stage, never even happened in this plant. Other plants have certain of the growth stages are very similar to wild type. Others are much shorter and can be much longer. As you can see here, all of these have a unique pattern of progression through the growth stage. So this is very valuable information. And again, getting back to trying to isolate tissues and do gene expression profiling, for example, when you're comparing these different mutants, you'd want to make sure that you're collecting the tissue when they're at the same growth stage, not necessarily the same time after planting. I like to show this example. This is a good example of why it's important to collect all this information and see, what the plant is actually doing over time. And this is an analysis of the ADG1-1 mutant. ADG1 is an ADP glucose pyrophosphorylase. It's involved in starch biosynthesis. So if you just grew up the plants and took them to maturity and isolated the seeds because you were interested in seed yield, what you would see is that there's really no difference between the mutant and the wild type. They have essentially identical seed yields. However, if you start looking at different characteristics of the plant, you see that the mutant has twice as many siliques as compared to wild type, but it has half as many seeds per silique. So the way that this plant produced its yield was very different from wild type. And you would miss some of these parameters if you weren't collecting all these data and monitoring them over time. So this is a very simple example of the types of things that you can pick up by comparing these plants and collecting all the phenotypic data that we collected. Just want to give you a high-level summary. As I said, we analyzed about 17,000 genes, and when we looked, if we look at the gene function summary for the yield traits that we looked at, almost 50% of the genes were of unknown function. So these were genes that no one would have really looked at because they didn't have the DNA sequence didn't really give you a clue about function. And so these were very interesting to start with and to start sorting through. The next group is energy and metabolism. The next group was cell communication, signal transduction, transcription factors, and so on. So although these categories are pretty much what you would expect, it's specific, you would expect energy and metabolism genes to be critical for yield traits. The transcription factors were particularly useful because these are used, you can imagine ways of using these to engineer a plant to make it have a higher yield. It's a little more tricky to start manipulating energy metabolism genes and getting them expressed in a way that's actually going to be beneficial and give you the effect you want. So these transcription factors were a lot of interest for our collaborators. We also did screens to look for essential genes. Essential genes actually represent herbicide targets if you think about it. And we had a big project with Bayer Crop Science to try to identify essential genes. Similarly to the crop trait genes, almost 50% of the genes we tested were of unknown function, so no one would have really tested those or predicted that they were essential genes. In this case, the next group was primary metabolism, which again, you would expect to be essential in many cases. We did find a lot of transcription factors that were also essential. That was very interesting, and so on down these different categories. So now I want to shift gears a little bit and give you some specific data about a phenotypic analysis of G protein function. So heterotrimeric G proteins are very critical single transduction proteins in plants and actually in animals. They're highly conserved across eukaryotes. And these heterotrimeric G proteins, alpha, beta, and gamma, interact in a complex, and they interact with G protein coupled receptors that are responsive to a lot of different stimuli. So these are involved with a lot of hormonal regulation in plants, for example. And the way it works is these are off in the GTP form, but then when they're in the GTP form, this complex breaks up, and they can the alpha subunit can go off and have downstream effects, and the beta gamma subunits can go off and have their own independent effects. So this is the system that we were analyzing, and we did this in collaboration with Alan Jones' lab at the University of North Carolina. So GPA1 encodes the alpha subunit. It's a single copy gene in Arabidopsis, so we didn't have to worry about having compensation from other partners that might confound our analysis. So it's a single copy gene, and it's known to regulate cell proliferation through GA and brassinoid steroid signaling. In this case, we looked at two different mutant alleles, and these were both knockouts. So these were tDNA insertions into the gene, and these were validated not to express any GPA1. The other gene we looked at was the beta subunit, AGB. In this case, again, AGB is a single copy gene in Arabidopsis, and it has a number of functions, but one of the main functions, it's a negative regulator of ABA regulated processes. For this gene, we actually had two alleles. One was a point mutation right at the splice site. So this actually expressed a truncated version of the protein that had an additional 20 amino acids that was not normally found in AGB, as well as a knockout in this exon shown here. So what we did was grow these up, run them through our plate and soil-based assays. And what I'm showing you here is a summary of all the soil data that we collected. So there's about 50 traits on here, as I mentioned before. So what I'm showing you here is we've plotted the phenotype for a particular trait that we measured, for example, seed yield, against the control. And this is the standard error measurement relative to the control. So anything that's between the black bar here, -2 and 2, is not significantly different from the control. So this is the effects from GPA1-1. This is GPA1-2. Remember, these are both knockout mutants. And what you can see is that a number of traits are affected in these mutants. And if they're affected the way that this is plotted, if the effect is similar in both alleles, they should line up close to this red line shown here. And that's what most of these traits do. And since these are both knockout alleles, that's what you'd expect. They should more or less line up on the red line. We could see that one of the major phenotypes, the most dramatic phenotype that we saw, was the effect on pedicel. So pedicel is the small little stalk that the silique is attached to on the inflorescence of Arabidopsis. But we did have some effects on seed yield. We had some pretty strong effects on seed size and shape as well. A number of traits were not affected in either allele. Now, if we look at the AGB mutants, remember AGB1 was a point mutation. It expressed a truncated version, whereas 1-2 is the knockout. So it's not expressing any protein. So what was interesting here was a number of the traits were equally affected in both alleles. So silique length, flower width, some of these flower phenotypes were quite similar in both alleles. We have alleles that were not affected in either, or we have traits that were not affected by either mutation. But then we have some traits here that were not affected in the point mutation, but were affected in the knockout. So seed yield and some of these root traits. We also had some flower timing traits as well as silique traits that were affected in the point mutant, but not in the knockout. So that was very interesting as well. So this is getting to the point I was talking to you about before, whereas you can use this system by collecting a lot of different trait data and start comparing alleles, and you could even do epistasis experiments with different alleles. And I'll show you that here, how you can kind of combine some of these things. So this is plotting, in this case, both of the knockouts, the GPA1 knockout against the AGB knockout. And again, you see there's, interestingly, a number of traits that are equally affected in both mutants. So those phenotypes were quite similar, but you have this pedicel length that I pointed out earlier was very strongly affected in the GPA1 mutant, but it is not affected at all in the AGB1 mutant. So you can imagine now, I think, if you had different mutant alleles or double mutants that you wanted to compare to the single mutants, you could start doing epistasis experiments and figuring out how these different genes might be interacting. So again, now I'm going to switch gears again and talk about transactivation as a tool for dissecting complex phenotypes. This was a fair amount of work, but it turned out to be very important for us, particularly in identifying the essential genes for the herbicide project. So the idea here is, again, and this was taken out of some of the early transcription factor work that was coming out of Boston. The idea here is that you can create these chimeric transcription factors, these artificial transcription factors. In our case, we were taking the GAL4 with the VP16 activation domain, and then linking them to a tissue-specific promoter. So we would be driving the expression of our transcription factor in different tissues or different cell types. And the drivers that we created are shown here. So we had drivers that were flower specific. We had some that were more root specific, and so on. The other partner in this scheme is the target. So this would be our gene of interest, and we would clone the gene either as an antisense construct or as an overexpression construct. And then we would have upstream, we would have an artificial promoter that could not be activated by any endogenous transcription factors, but only activated in the presence of this artificial chimeric transcription factor. So we had what we called our driver line. So this was a homozygous transgenic line that was expressing just this transcription factor. And then we would create our target lines with our gene of interest, and then we could simply cross these, and then in the progeny, we would get activation of the target, and then we could start looking at the phenotypes. So again, I'm going to go back to our G protein, heterotrimeric G protein mutants, and how we would dissect and try to understand whether or not this gene could be useful, even though it had a very complex phenotype. So these AGB mutants had both dramatic shoot as well as root phenotypes. So we had these more compact vegetative growth, but we had these very bushy roots compared to control here. In both of these, Columbia is always the control. And you can see here the leaf phenotypes. They had more rounded, smaller leaves. So you can imagine that, well, this root phenotype might be very nice to have in a crop plant under certain conditions, but this other shoot phenotype was probably not be very good. So that, you know, you would throw this gene away because it had too many effects and might not be useful. But with the driver system, what we could show was that if we used a driver that was expressed primarily in the lateral root, so that's shown here. So this is the driver. This is just a picture of a root from a plant where we crossed the driver line with a construct that had the GUS gene, which is a reporter gene that will give you a blue color when you do a histochemical stain. And you can see that its expression is localized to the lateral root. So when we, again, if we have a 35S, a very strong promoter driving an antisense construct for AGB, we get this plant that doesn't look very healthy, doesn't look very good from a vegetative point of view. But if we take that antisense target, cross it with our driver line, you see that we're able to get the nice root phenotype, enhanced root growth, but we have normal vegetative growth. So we're able to separate and pull out and analyze these complex phenotypes. Of course, this strategy is also useful too, because, you know, it allows us a way to generate root tissue for gene expression or metabolic profiling, where we don't have to worry about the effects being confounded because we have an unhealthy or abnormal vegetative growth going on in that plant. So this driver system is very, very good for this type of experiment. So just to summarize a little bit, well, let me back up. Sorry, I meant to mention, as I said, what we did with this was also to identify essential genes. So if we had knocked out a gene and it was an essential gene, what we would see when we plated those progeny would be that about 50% of the plants wouldn't grow. So that was our indication that we might have an essential gene. So that's summarized here. So these represent potential herbicide targets. It's very difficult to identify these by traditional mutational screens. In many cases, people would say, well, I've tried to identify this particular mutant and I've screened a bazillion plants and I can never find it. That must mean it's an essential gene. So that's not very satisfying. You'd like to have some real proof, tangible proof that a gene was essential. So this transactivation provided a really good tool for that. So as I said, our target line would be an antisense gene construct. We would cross it. In this case, we would use a strong 35S promoter because we wanted to make sure we had good expression. And then we would just test the progeny for growth on plate assays and look for things that gave us about 50% no germination or seedling lethality. I would say most of the essential genes that we identified gave a seedling lethality phenotype. So they would germinate, you'd get a little seedling, but it wouldn't grow after a couple days. Just to give you an idea of how powerful this turned out to be, we identified 45 new herbicide targets in a 3-year period. And that compares to the industry doing more chemical-based screening assays, it took them several decades to identify 13 molecular targets. So this was a very efficient way to develop, or at least identify, potential new targets for herbicides. So now I just want to summarize a bit about the phenotypic platform. It's a very multifaceted platform for gene function identification, and it can easily be adapted to address pretty much any aspect of plant growth and development that you might be interested in. It's applicable to analysis of mutants and transgenic lines, which is what we used it for mostly. However, it's also useful to look at natural variation. We did run about 20 different natural land races of Arabidopsis through the platform, and several of them had very interesting phenotypes. You can also use the platform to look at environmental stress and chemical treatments, and in a minute, I'll talk a little bit more about the chemical treatments. As I mentioned, you can utilize it to compare the effects of mutant alleles and dissect gene networks. A key aspect is it provides growth stage specific links between morphological traits and metabolic profiling data and gene expression profiling data. It really makes it a synergistic, it really synergizes your analysis when you're able to properly integrate these different data types linked to specific growth stage and morphological traits. The tissue-specific transactivation of target genes, as I said, it's very interesting and useful to dissect complex pleiotropic phenotypes that you might see in a particular mutation, and it provides a very facile way of analyzing the function of essential genes or identifying essential genes. And again, for us, it really provided a platform for the integration of diverse data sets and production of what we called coherent data sets for gene function determination. So now I want to talk about chemical genetics. This was a project that we came up with, and I think still has a lot of potential. I really would like to further develop this project. And again, this is based on trying to leverage all the tools that we developed in our phenotyping platform. And really the goal of this project is to apply systematic screening approaches to identify small molecules and natural products that have specific trait enhancing effects in plants. So it's akin to, what the big ag companies had been doing for years for screening large libraries for compounds. But we were going to take advantage of some of our high throughput phenotypic screens to look for essentially chemicals that affected traits. So we started out with our two really robust screens. One was a nitrogen utilization screen, and one was cold tolerance, where the cold treatment was an 8°C treatment. And then down the road, I think it would be really interesting to develop target gene-based approaches where once we know the particular function of a gene, we can make reporter gene fusions for transcriptional activation. And then when we look, then we can then do chemical screens and look for the chemical's ability to activate a particular reporter. So if we know cold tolerance is associated with the activation of a specific gene, we can use the promoter from that gene, make the reporter, and then simply look for activation of the reporter instead of having to go through and do a complete assay on the plant throughout the whole assay screen. So this was the format. We did it in 96 well plates. We put 5 Arabidopsis seeds into each well, and we did duplicate plates for each assay. And the readout for this was very straightforward. We would take a fluorescence image of the plate. As you know, chlorophyll will fluoresce under UV and give you a nice red color. We could take an image and then use image analysis to quantify how much fluorescence was observed in each well. Generally speaking, the amount of chlorophyll you would detect would be an indication of the health of the plant or the biomass of the plant. So it gave us a very quick quantitative way of assessing whether the chemical that we put in these wells had any effect. So we could put one chemical, a different chemical in each well. So we could screen up to, well, we screened about 90 compounds per plate because we always had controls and we always wanted to have a good control baseline. Once we did the primary screen and identified potential hits, we did a secondary validation. And for that, we rescreened the hits using the standard assays, so the longer term phenotypic assays that were part of our core platform to validate whether or not the low nitrogen, the cold phenotype. We also would do cold shock. The cold stress screen was the plants were grown under constant cold temperature. In a cold shock assay, they're grown under normal temperatures until you have a nice seedling, and then they're shifted to the cold temperature to see how they respond to that. And then, of course, our standard growth condition was always there for a control. We did a little bit of a modified version of our physiology platform. We looked for growth stage progression, color, root length, fresh weights, and then we did image analysis. And I'll show you, of course, our primary screen was done with the Arabidopsis, but we also did some secondary screens with other crop plants, canola, which is a relative of Arabidopsis, tomato, and maize. So this is just gives you an example of one screen. So this is the sort of the effect of the fluorescence reading that we're getting. And you can see that, you know, there's some noise here, but you do see some plants that have very high fluorescence, in this case under cold temperature. So these would be the ones that we would be most interested in, because they're showing more growth under cold stress. This is just an example of the nitrogen assay. Again, you've got a lot of plants down here, but you do have some plants that are showing much higher growth under these low nitrogen conditions. So these would be some of the initial hits that we would then look at further. We've done some screens in the cold assay. We screened 4,880 compounds, and we got 160 primary hits, which was about a 3% hit rate. But then when we took those into the validation assay, only 54 of those validated. So that was a validation rate of 34%. So our overall sort of hit rate was about 1%. So this was definitely a hit rate that was capable of identifying a sufficient number of initial compounds that would ultimately gives us the numbers that we think would be required to end up actually getting something through later levels of validation and ultimately developed as a product. A similar analysis of the low nitrogen assay. We did about 5,200 chemicals for this screen. We had a 240 initial hits, so that's a 4.6% hit rate, a little bit higher than the cold screen. Of these 240, 85 validated. So the validation rate was about the same in both of these very diverse screens. The hit rate was 1.6%, just a little bit higher than the cold screen. The other thing that you can do, which I think is interesting and is another validation step, was to look for specificity of the response. So what we're plotting here is the T values relative to the control for these two different stresses, where the blue is the nitrogen assay T value and the cold assay is in the magenta. So anything that's between 2 and -2 is not significantly different. As you can see, some of the cold assay hits actually have a phenotype in the nitrogen assay. But on most of these, the nitrogen effect is specific to nitrogen. There isn't any effect in the cold assay. And similarly, things that are showing an effect in the cold assay really don't have a significant effect in the nitrogen assay. So identifying compounds that are specific and aren't having some sort of pleiotropic effect is another way to do a secondary validation. Just to give you a little more data here on the cold assay, when we started looking at in more detail at the phenotypes caused by these compounds, so this is the control condition on cold stress. You get germination at day 6, but in this case, with these two compounds, we had faster germination and root growth. If you look at another one of these compounds, para 3, you can see in the cold plate assay, so in the validation assay, at day 28, the plants have grown significantly more than the control, both roots and the shoots. If you look at quantitative data, so this is the average fresh weight of a number of these compounds. You can see that we do have some pretty dramatic effects where some of these compounds give us as much as 2 times as much growth as the control. And you know, we identified a number of these compounds. As I said, we also tested some of these to see if they would have any protective effect in a cold shock assay. So this is where you grow the seedlings up to a good seedling size and then hit them with the cold treatment. And you can see that this compound picked up in the cold assay also shows increased growth in a cold shock assay. So that was encouraging to see that. Again, similar quantitative data if the cold shock hits. In many of the compounds that provided increased growth under continuous cold shock also provided, or continuous cold also provided protection in this cold shock assay. And then this is an example of a further validation where we use canola. Canola was nice because we could also use a plate assay here. And you can see that one of our compounds, a para 43, did also provide protection to canola in this case. This is the cold stress where it's under continuous cold. We also went and validated a few of these in some secondary screens based on soil. So this is a cold germination assay. So again, this was all done at 8 degrees centigrade. Plants were sown in soil. As you can see, the control does germinate, but you get a very small, very purple stress plant. But treating these plants with these various compounds, and the way these were applied was simply dripping the compounds on the plant as they started to grow. And they were able to protect the plant, give increased growth, even under these soil conditions. So just to summarize the chemical genetics program, we think this is a pretty novel approach that really leveraged the capabilities that we developed through our high throughput plant phenotype analysis. We did a couple proof of concept experiments and validated the approach, both for the 96 well nitrogen utilization and the cold assays. And we got some quality hits for some follow-up studies. We developed both plate and soil assays that validated how robust this screen could be. And what was interesting was that the assays generated, you know, a number of hits, but we had very broad chemical diversity. These all weren't derivatives of one chemical class. It represented a variety of different chemical types. So we were able to identify a wealth of new information about how certain chemicals might be used improve plant growth. So I think this is really a significant opportunity, and this is an area that I think is ripe for further development. So last but not least, I want to finish up. I want to tell you a little bit about what we're doing with Sativagen. Two of the co-founders of Paradigm Genetics, where we did some of the initial work with the Gene Function Factory, have decided that this is an opportune time to build a revised Gene Function Factory and apply it to cannabis. So the question is, why cannabis? It's rapidly an expanding market worldwide. There's a lot of uses. I mean, people, think about cannabis as ganja, but I mean, hemp is a very large and growing market in the United States. There's a lot of use, and there's actually some of the first FDA approved drugs using cannabinoids isolated from cannabis has now been approved. So this plant is garnering a lot more interest and is going to be developed substantially, I think, over the next decade. One key factor is that modern agricultural research, biotech research, has not been applied to this crop. I mean, it's been illegal and continues to be illegal in a lot of jurisdictions. So all the tools and all the work that's been done for other crops have not been applied to cannabis. And we believe that as the products coming out of cannabis are more regulated, then existing practices in germplasm will need to be modernized. So we think this is a sort of a crop plant that's wide open for further development, and we think that our tools will be very useful. So really, right now, a comprehensive industrial R&D effort is needed to bring cannabis up to speed, and is going to be important for companies that want to remain to be competitive in this industry as the regulators get more involved in who can grow this crop and how it has to be grown. So this just exemplifies, the problem that's facing the cannabis industry. It completely missed the ag revolution. So if you just look over time, the advances in agriculture that have occurred, this period here is where cannabis was highly regulated or prohibited, particularly in the United States. You couldn't do anything with cannabis. And this was the same period of time where agriculture and monoculture, all the inbred lines, all the hybrid lines were being developed. Then the GMO and transgenic plant lines being developed, and the advent of precision agriculture. Cannabis was stuck way back here where people were sort of doing things in their backyard and not really tracking and having proper breeding programs. So what we plan on doing and are trying to raise money right now to do is create Gene Function Factory SG 2020. Sorry, I had a typo here, 2020. And really, we're just going to use our comprehensive industrialized Gene Function Factory and use it to analyze gene variants in cannabis, as well as the large number of varieties that are already out there, and understand by integrating the phenotypes with the metabolomic data, the genetic data, and by genetic data here, I mean genomic sequence, as well as our phenomics data, and use our data integration approaches to determine gene function. And this will create the intellectual property that will then drive our collaborations with other cannabis companies and so on. So really, we're very excited about the opportunity to create a new, better gene function factory. And this time we're really going to focus heavily on metabolomics, because a lot of the products that are important coming out of cannabis are secondary metabolites. So with that, I'll end and just give the acknowledgments. So a lot of the original gene function factory work was done. Jorn Gorlach and Sandy Stewart, those are the two collaborators who I'm currently working with in the cannabis company. The rest of these are the group leaders for the different groups that we're running the gene function factory. As I mentioned, that G protein data, that was done in collaboration with Alan Jones and his student and postdoc. So with that, I'll stop and take any questions. I'm not sure how you're doing questions, but happy to address anything that you would like to talk about.
Yeah, thank you, sir. Thank you for the great presentation. And we have some queries in the chat box. Before reading that, I would like to give a general announcement to the participants. Dear participants, you can ask questions either by putting, that is as a typed message in the chat box, or you can raise your hand in the raise your hand and then unmute and ask. So these are the options for asking your queries. Anyways, I am going to start with the first question in the chat box. So this is from Dr. Deepthi Madai. Could we tackle diseases by understanding the gene function and thus improve crop yield?
I'm sorry,
Dr.
you broke
Keith?
up. Let me see if I can pull up the chat here.
I'm ready for you, Dr. Keith.
Yes. You broke up. I couldn't hear the question.
Shabana ma'am. Shabana ma'am.
Hello.
Shabana ma'am, can you please repeat the question?
Yeah, sure. Dr. Keith, am I audible?
Yes.
Okay, this is from Dr. Deepthi Madai. Could we tackle plant diseases by understanding the gene function and thus improve crop yield?
Yeah, absolutely. I mean, a lot of my academic work was in plant disease resistance, and it would be very straightforward to develop screens and implement those into such a high throughput mechanism, both using transgenic plants or mutants of whatever type. And I think you could also use chemical. It would also be good for chemical screens. But yeah, you definitely could implement assays to test specific plant pathogen interactions.
Thank you, sir. Next question is from Shraddha. What role, am I audible? Hello?
Yes.
Thank you, sir. What role can CRISPR
You're breaking up. Was this the?
Is this the? She's explaining his question that Shimen, can we control drought stress response? Hello.
Yeah. Is this the CRISPR
Hello?
question?
Yes, sir. CRISPR question from Shraddha.
Yeah. Okay.
What role can CRISPR-Cas gene editing
I
mitigate the plant disease?
Yeah, I've got it. Yeah, I mean, actually, that's what we're doing with the new improved platform is getting away from the older antisense overexpression approaches and using CRISPR to make targeted mutations either in promoters or in the coding regions of the gene. And as far as drought, yeah, I mean, we had drought assays and we identified genes that provided drought tolerance. So.
Okay, sir. Next question.
Yeah,
Sir, you got the question, sir?
no, I didn't hear anything. Is this about
Yeah, I
Arabidopsis?
will repeat, sir. I may repeat. Can we use any other plants, or still we have to stick on to Arabidopsis for this experiment?
Yeah, I mean, that was actually when Dr. Isha asked me to present. The reason I thought I would give this talk is I think that the concept or the approach of using this platform can be adapted for any plant. And actually, when when we were acquired by Monsanto, we had pretty much finished everything that we wanted to do with the Arabidopsis, and it was converted. They modified the platform and started using corn. And so I think, you won't be able to do, as many things perhaps. I mean, it's going to be hard to do plate-based screens with most any other plant. But I think the key ideas of developing very robust and quantitative phenotypic assays is very powerful and can be done with any plant. And now with the improved next generation RNA-seq, it's getting simpler to do RNA expression, gene expression studies. And as I said, we have particularly great expertise in metabolomics. So we have the best metabolomics, I think, that will be out there. And that, again, we're applying it to cannabis. So I think if you can apply it to cannabis, you can apply it to probably any other plant you want. I mean, maybe not trees, certain very large plants, it might be hard, but I think you can use these concepts and even use it in large tree species. Because the point is really developing assays for key traits and measuring them quantitatively and linking those to either specific mutations or alterations in gene function.
Thank you, sir. And the next question is from Dr. Sahaya Shibu. He first acknowledges you for your nice presentation. And he has asked 2 questions. I will go with the first one. Shall we use Arabidopsis as a model system for monocots? Especially, he just wanted to know about orchids.
Well, that's from the very beginning when people were really pushing to use Arabidopsis as a model system. There was a lot of discussion and controversy about whether anything we found in Arabidopsis would be very useful. What's been shown over the years is that a number of things are conserved across monocots and dicots, but not everything. So it really depends on what it is you're trying to study and the level of conservation across, monocots and dicots. But I will say the other aspect, I didn't really get into this much, but one thing that we've been pushing, and we're gonna do a lot of this for the cannabis work, is using Arabidopsis essentially as a heterologous system. So since it's much easier to test gene function in Arabidopsis, we're going to move genes from, say, cannabis and mutate those genes and put them into Arabidopsis and see what happens there. So there's certain things that you can do. So if you're studying something that is highly specific to monocots or orchids, chances are it probably won't work in Arabidopsis. But if it's a more conserved trait, then it might.
Okay, sir. And his next question is that, do you have your own database?
Do we have our own database? Well, actually, right now we don't have a database. We're starting pretty much from scratch. Monsanto had, and now since Bayer bought Monsanto, they have our database, but we don't have access to it anymore. but I do remember a lot of things, and we're going to use all of the knowledge that we developed before and apply it to cannabis.
Thank you, sir. I think the questions, and there is one more question from Nagma Aftab. Shall I ask? She is asking, can Arabidopsis used as a model for curing human diseases?
Well, I would say off the top of my head, I can't think of a good example of how that might work, to tell you the truth. I mean, well, I guess, I mean, one way that it can, and this kind of gets back to some work that I was doing not too long ago, using plants as an expression system to make biologic drugs. So, I mean, if you're, where it is being used, well, mostly it's being done in tobacco, but you could do this in Arabidopsis, is making various constructs for expression in plants, and then producing those proteins in plants, because plants are an easy system to make things compared to animal cell cultures, and then sort through your different constructs to figure out which one gives you an active protein, and then you could develop that further. I think there are examples now where people are actually producing the protein drugs in tobacco, in plants, in plant cells. There's one approved drug where they are producing a enzyme for a metabolic deficiency syndrome that's being produced in carrot cell cultures, and a number of vaccines and monoclonal antibodies are being produced. For example, was it 5 or 6 years ago, there was an Ebola outbreak. The monoclonal antibody cocktail that was used to treat some of the doctors who got infected during that outbreak was made in tobacco, actually. So I think maybe as a research tool to do something, but as far as trying to figure out whether a gene, a disease gene in humans, what the function of that gene is in Arabidopsis, I think that would be very difficult.
Oh, well, it's a very nice information, sir. Thank you so much. Thank you, dear participants, for asking questions. And I think no more questions are there, and no hands have raised in the chat box also. So let us move on to the concluding session. I would like to request Dr. Asha B, Assistant Professor, Department of Microbiology, SIAS, also the convener of Bio-Inventiyon 2020, to give away the formal vote of thanks. Ma'am, please.
Thank you, Shabana. Hope I'm audible.
Yes, ma'am, you are.
Okay. So, very good morning to everyone. I'm honored to deliver the vote of thanks today. First of all, I would like to thank our guest, Dr. Keith R. Davis, for his excellent talk on plant gene function discovery using the high throughput gene function factory. The topic was really informative and the presentation was very interesting. Thank you very much, sir, for patiently clarifying all the queries and for being with us in these late hours. Next, I would like to thank Professor Dr. Sabu Thomas, VC, MG University, Professor E.P. Imbichikoya, Principal, Safi Institute of Advanced Study, Professor Dr. K. Jayachandran, Director, School of Biosciences, and Dr. Jisha M.S., Professor, School of Biosciences, for all support and encouragements. I thank Dr. Shabana, Research Director, and Dr. Shaini N.K., IQAC Coordinator, Safi College, other faculty members and research scholars of both the institutions who are behind this program for organizing it with such coordination and perfection. Next, I thank Mr. Muhammad Safir, Division Manager, Toastmasters, KSA, for all the technical support in this event. Last but not the least, I thank all the delegates present here from different institutions all around the world for their valuable presence. I once again thank everyone here and wish you a good day. Thank you. Shabana ma'am.
Thank you so much, Asha ma'am. Thank you so much, Dr. Keith, once again for the excellent presentation and clearing doubts. And I would like to ask our participants to take the 10-minute session break now, and we will continue with the oral presentations after 10 minutes, exactly 10 minutes. And I request for the first category of oral category, to get ready with your presentations and be on a queue for the information provided from the desk. So once again, I thank everyone and stay safe. Have a nice day. Thank you.
Okay. 11:45 അല്ലേ? Okay. സാർ പോയോ?
I'm here. Yes, I'm here.
Okay, sir. കീർത്തി ടീച്ചർ ജോയിൻ ചെയ്തോ? ചേട്ടൻ ഒരു 11:40 ആകുമ്പോൾ ഇൻട്രൊഡ്യൂസ് ചെയ്യാൻ പറയും.
ടീച്ചറെ ഞാനിവിടെ ഉണ്ടേ.
ഓക്കെ കീർത്തി, ഇപ്പൊ കേൾക്കത്തില്ലേ? കേൾക്കുന്നുണ്ടോ?
സെഷൻ കഴിഞ്ഞു. ഇങ്ങനെ ഓക്കെ.
Hello everyone, welcome back. Hope everyone is ready and got refreshed. Once again, thank you for your participation in Bio-Inventiyon 2020. And dear friends, now it's the time for oral presentation, to start the oral presentation. And I would like to hand over the mic to the young scholars, the young research scholars, and the active co-organizers of Bio-Inventiyon 2020, the research scholars, Ms. Sherin and Ms. Edna, to take care of the session. Sharon and Edna, all the best, and participants, all the best. And please be keen always for the instructions from the desk.
Hello. Hello.
Yes, dears, you are audible. Please start.
Hello. Okay. Thank you, Shabana ma'am. Good morning, all. I welcome all the participants to the presentation sessions of Bio-Inventiyon 2020 web conference.
You can see here the thick vegetation that is present. This is far away from
The first session,
the karst plateau on Google.
am I audible?
Pictures.
Hello?
Yes,
Am I
yes,
audible?
ma'am.
Okay. The first
Yes,
session,
audible, ma'am.
okay. The first session is from 11:45 a.m. to 1:45 p.m.
Bombay, not a people
and
will
that
come.
will be
Millions
best
of people
paper
visit this
faculty
temple
category.
every day. And now there is restriction for taking the
The
vehicles on the
session
top
will
of
be
this
chaired
plateau.
by
But they can park and far
Dr.
away from that
Jayachandran
place
K,
and then enter, go by walk.
Professor, School of
But for old
Biosciences.
people or have physically challenged people like me, may be difficult to walk the distance. But I visited this place.
Hello? Am I audible now?
Yes, dear. Yes.
Okay. So, first chair would be Dr. Jayachandran K, Professor, School of Bioscience, Mahatma Gandhi University, Kottayam, Kerala. Second, Dr. Kirti TR, Professor, School of Biosciences, Mahatma Gandhi University, Kottayam, Kerala. I cordially welcome them to the session. Participants, kindly pay attention to the general instructions. Each participant is allotted 10 minutes for their presentation. There will be a reminder after 9 minutes. The presentation after 10 minutes will not be valued. So kindly stick on to the time duration. The question answer session for each participant will be 5 minutes. If you have any difficulty in sharing your screen, you can send your slides to our mail bioinvention2020@gmail.com in advance. Kirti ma'am, are you ready?
Yes, I am ready.
Jain sir, are you ready?
Yes, yes.
So shall we start, sir?
Sure.
Okay, so I invite the first participant, code number OPFC1, Dr. Nanno Shafakatullah.
Yes, I'm here.
Sir, you can present now.
Okay, thank you. Okay, can you see the screen?
Yes, sir.
Yes.
Please
Okay.
make it full screen.
One second. Yeah. Thank you. Good morning, everyone. Myself, Dr. Nanusha Fagatullah from Mangalore University. So here I am to present a small topic from my own research, a small topic on evaluation of probiotic potentials of bacteria isolated from oyster. So generally we use these marine oysters have been used for isolation and identification of good bacteria. So here it's about the probiotic bacteria which are responsible to give good benefit to the individual. So when you take the definition about these probiotics, these are live organism which when we intake them, so they're going to give a health effect to the individual. And these probiotics, we are having a large classes of the probiotics. So generally we are calling them as the lactic acid bacteria. So we are having lactobacillus, lactococcus, bifidobacteria, streptococcus, leuconostoc, and other bacteria we are having. So to call them as the probiotic bacteria, they should have some special character, like since we are intaking them along with our diet or along with the food, so they should have the capacity to survive in our GI tract, that is in the gastrointestinal tract, or especially maybe in the stomach or intestine. So they should be able to survive. They should be able to proliferate or colonize. They should not have, because this organism what we are intaking, they should not be pathogenic, because we are intaking orally. They are entering into the intestinal tube or intestinal tract, and they have to survive there for certain periods so that they can give some good effect on the individual. And they should not have any mutation effect, or they should not have any carcinogenic effect on the individual, and they should be stable. And since these organisms are resistant to certain antibiotics, there should not be any plasmid transfer to the organism which are already present in the intestine. Maybe in the intestine we're having E. coli. So the resistant plasmid present in this bacteria should not be transferred to those E. coli organism. And they should have the defense mechanism, because in intestine where we'll be having the pathogenic organism. So here they should have the defense mechanism, and they should be stabilization to the local flora. Immunomodulation should be there. They should tolerate to the lactose intolerance should be there. Prevention of viral bacterial infection should be there. So overall, when you take, so these organisms are going
Everyone,
to
please
give.
be muted, please. Please. Sir, please continue, sir. Sorry.
Please unmute your mic. It's not audible.
Not audible, not audible.
Yes. And in this, our research, we have isolated three different organisms. One is the Enterococcus faecium, Pediococcus acidilactiaceae, and Pediococcus pentoschisis. So we'll see the normal isolation process. So for this isolation, we have used the fresh marine oysters have been procured. So with the help of fishermen, we got this oysters brought to the laboratory, and we have separated the meat along with the fluid which is present within these oysters, and that has been crushed and used for normal isolation process. And for the isolation, we have used the MRS general media for the specific media for the probiotic isolation. So in that, the normal identification process, we have used morphological identification has been done for the general identification. In that, then we have done biochemical characteristics. So for the initial identification, then carbohydrate fermentation test. So this carbohydrate fermentation test can give you a little bit clue about the specific organism, the belonging of the specific organism. Then we have done physiological characteristics, then the molecular identification to prove which organism it is. Finally, we have done the 16S rRNA sequencing to find out what which organism it is. Then we have studied about their probiotic properties, because we isolated the organism. You want to know whether they are having this probiotic properties or not. So for that, according to the ICMR as well as FAO and WHO guidelines, we are having certain specific characteristics. So the probiotic should have some specific properties. So for those specific property, we have done all these 11 to 12 tests. So now we'll see what outcome we got by doing all these tests. So in the morphological test or morphological, physiological, and biochemical investigation, we got this 4 organisms, and these organisms were gram positive, because when it is gram negative, that becomes the pathogenic organism, and these were catalyst negative organisms. So these are the general description about the morphological, physiological characteristics of the organism. Then we have done the carbohydrate fermentation, this one test to differentiate between the different organisms based on the Burgess manual. We have used around 23 carbohydrates, and we have got the positive and negative results. Some are going to ferment this specific carbohydrate, and some were unable to ferment the specific carbohydrates. Then we have done the next one, that is about their survival in the higher salt concentration, because since we are using this probiotic should have that capacity to survive when you're storing them, because when you're making any product and we are putting this organism, and when you're storing this organism, they should have that capacity to survive. So we have done their NaCl tolerance, and this organism, that is Enterococcus faecium, Pediococcus acidilactase, and pentose, they have shown good tolerance to the higher salt concentration. Then we have done their temperature as well as pH tolerance. So this is the result of their temperature as well as pH tolerance, and they can tolerate a pH of around 2 and 3 also, because they are having that acidic nature tolerance capacity. Then the important one here is we have done the probiotic characterization in that acid tolerance, bile acid. All these are the properties given by this one specified by the ICMR. So when organism they are following these properties, or when they are showing positive result to these properties, we can consider them as a probiotic organism. So the first one is acid tolerance we have done. So we have done this acid tolerance because when you're intaking these probiotics, they're entering into the stomach, and when the organism is inside the stomach, the pH of the stomach, it is in acidic nature, so the organism should be able to survive in that acidic nature. So we have done 2 characters, that is, one is at the pH 2 and pH 3, we have seen the survival, and both at pH 2 the organism have shown around, that is the first organism, that is the Enterococcus, has shown around 12% of survival, and other 2 organisms they have shown in between 8 to 10% of the survival. So we can see that they got certain property where they can tolerate the acidic nature of the stomach. Then in the stomach, when you see, we are also having, there is a bile concentration. So bile can also act as one inhibitory compound for the organism, but whatever probiotics we are intaking, they should not have, they should not be sensitive to the bile present in the stomach or in the duodenum. So we have done their sensitivity to the bile, and the organism could tolerate the bile for about 180 minutes till 180 minutes. We have done their survival to study whether they can survive. So the organism has seen around 80% of survival in the bile concentration. Then we have done about this one, that is the bile salt hydrolase activity. So this bile salt hydrolase activity is of the organism is helping to eliminate the cholesterol which is present in the system. So here the organism have shown good bile salt hydrolase activity, because that can separate our bile acid, that is cholesterol plus glycine can be separated easily, and the cholesterol which is getting separated, that can be eliminated from the system. So for that got the result where the Enterococcus faecium, Pediococcus as well as pentose, they have shown a good bile salt hydrolase activity, means they can help to eliminate the excess amount of the cholesterol which is present in the body. Then we have done the gastric juice tolerance. So because we have to see whether the gastric juice which is present, maybe for
Hello,
example,
sir.
yes,
Hello, sir.
yes,
You have 1 more minute to go.
okay, it's all 55, it is 45 I started. Okay, yeah, no problem. Then we have seen about the gastric juice concentration. This is the findings of gastric juice concentration. Then the intestinal tolerance we have seen. The organism has shown good tolerance to the intestinal juice also. Then auto-aggregation, because they have to survive in the intestine, so they should have that auto-aggregation as well as co-aggregation properties, and they should have the adhesion capacity because they should be remaining inside the intestinal tube. They should have the capacity to remain attached to the intestinal tube, so they have shown good adhesion property also. Then we have seen about the hydrophobicity. This also helping for the adhesion so that organism can colonize in the system, and there is good correlation between the adhesion and co-aggregation and adhesion as well as hydrophobicity. Then we have seen about the antibiotic susceptibility because they have to show the resistance to the antibiotics when you are taking along with certain drugs. So the organisms have shown good resistance to the antibiotics. Then we have seen about the antimicrobial properties because they should not be inhibited by the pathogens which are present in the intestine. So that also we have done. Then we've seen about their bacteria antimicrobial product which is produced by them so that they can inhibit the growth of other pathogenic organism. You have seen about the bacteriocin, lactic acid as well as hydrogen peroxide activity. And everywhere we got almost a good result about this organism where they produce the lactic acid as well as hydrogen peroxide. So in conclusion, we can say that this all three organisms which we isolated in this study. So the first organism, Enterococcus has shown good property compared to the remaining two Pediococcus organisms. So and this time I thank this ICMR for giving a grant to do this specific research, and this research was based on the non-fermentable food because especially for isolation of probiotics, fermentable foods will be used, but our intention was to find out the organism from non-fermented food, and we got a good result from the non-fermented foods. And these are my few references, and thank you.
Okay, sir. Okay, sir. Good presentation. Good work you have done, and thank you for your active participation in this. I think Professor Kirti teacher will now say a few remarks on this work.
Yeah.
Okay, thank you, sir. Okay. Good presentation and good work. Actually, I am also working in this area. Okay, I have one question. Your potent isolate is Enterococcus faecium, isn't it?
Yes, ma'am.
Yes, okay. How can you select this as a probiotic? It is an opportunistic pathogen.
Yeah, it is opportunistic organism, or it is like a contamination only. So the probiotic properties we are calling them as probiotics because they have to follow certain guidelines which are given or certain properties.
Okay. When we consider a probiotic, it should be a GRAS status, or there is some test to select probiotic. That is, it will be non-pathogenic. Okay, non-toxic. You have to prove it. What is the test you have follow for this Enterococcus faecium is non-pathogenic?
And the non-toxicity has not been studied in this. So
yes, it is because in the yes.
Yeah,
because Enterococcus faecium is not under GRAS status.
Yes, ma'am. Yeah.
So you should go for this. There is 2-3 toxicity testing.
Yes, ma'am. Yeah, that is true.
Otherwise, you can't select Enterococcus faecium as probiotic.
Yes, that's true, ma'am.
Then another question: you have conducted bile tolerance. Okay.
Yes, ma'am.
Yes. What methodology you have followed? You take turbidity, OD value.
Yes, ma'am. Turbidity only.
Okay, because it's a wrong method. Because bile, it's a tendency to form micelle formation. Okay.
Yes, ma'am.
Okay, you have to then check with all the CFU, colony count.
Colony forming mutant. Yes, ma'am.
Okay. Okay.
And for bile, we use colony forming only. This percentage colony forming in this one. CFU only.
Yes.
Percentage of survival. You can see in the graph. Turbidity is for another test, ma'am. CFU only used percentage of survival.
Okay. Okay. Good.
Yeah.
Okay, Kirti ma'am, are you done?
Yes. Okay, over.
Okay, thank you, Dr. Nannu Shafakatullah, sir.
Thank you.
So the next participant, OPFC2, Dr. Kamlesh Kumar Arsha. You can join, sir.
Hello.
Hello,
May I audible, ma'am?
I'm audible. Hello? Hello?
Yeah, may I
Am I
audible, ma'am?
audible?
Yeah, ma'am, please.
Yeah, okay.
You have to
Sir,
share
you
the
can
screen.
start. Please share your screen, sir.
Yeah, I can. I do that.
Yeah,
Have you
this
visualized my?
screen is visible.
Okay. So shall we start, ma'am?
Please switch on the slideshow, sir.
Okay. It is audible. It is a visualize,
Yeah,
ma'am.
yeah, we can see your slides and it's audible also.
Okay. So shall we start, ma'am?
Uh, okay, sir, you can start.
Okay. So myself, Dr. Kamlesh Shah, head of the department in biotechnology, Pramukh Swami Science and H.D. Patel Arts College, SV Campus, Gujarat. My topic is media optimization for lipase production by actinomycetes isolate from cotton seed stock. Normally, there are two major things in my presentation. One that is a cotton soft stock, and second thing that is actinomycetes. Why I am choosing these things? Because cotton soft stock, that is the one of the waste material which is extracted after the oil, because in nearby our place there are a lot of cotton industry and oil refinery industry, and they produce a lot of cotton soft stock, because in Gujarat and all the almost in all the state, cotton oil is now edible oil and it can be consumed as a edible oil. So after the refinery products, the cotton substocks are formed, and these cotton substocks are dark amorphous structures, and it having a lot of cellulosic, pectinosic, phenolic compounds, which is create a pollution in this environment. If it is a throw in water, then it can be a, water pollution, and if it is a throw in a solid, then it is a directly created the solid waste pollutions. So how to solve that problem? So we are thinking on that local problems, and we find that if we are taking indigenous microorganism from the substock and check the enzyme activities, and if we are apply that organisms on the that substock, then it can be a useful, because substock having a oil that can be a converted into a glycerin, or it can be a converted into biodiesel also. So we are think on that why actinomycetes, because actinomycete is a link between fungi and bacteria, and it is a very good potential organisms, because a lot of scientists work on the bacteria, fungi and protozoa, and also work on the actinomycetes. Actinomycetes have a very good ability to tolerant, as well as it have a ability to produce a lot of enzyme, as well as a antibiotic and various other substance which can be useful for the industrial applications. So here the actinomycetes which we are isolated, that is a 9A, and it having the sequence, it can be identified by biochemical as well as a 16S RNA. So after the 16S RNA, we confirm that it is a Streptomyces castalis species, and this species is almost a novel species, because it is not a completely match the available database species, which is we can go for the phylogeny tree. So it is some novel species. Then we have also checked the cellulase enzyme, lipase enzyme, amylase and protease enzyme. Why? Because cellulase, protease, lipase and amylase, these all are the industrial applicable enzymes, even though we are working on the soft stock, but soft stock containing the oil, cellulose material, so we have to do that. And also we are also going for the amylase and protease activity for the various organisms, so that we can confirm whether this organism apply in the enzyme production in solid state, or we can use as a bioremediation agent. So for the optimization process, we have isolate the actinomycetes, identified by 16S RNA and media optimization. Here the cotton seed stock is a dark gelatinous 7% oil-rich chemical constituents which is generated from the oil refinery. Actinomycetes are gram-positive free-living saprophytic bacteria widely distributed in a soil, water, even it is present in marine environment also, it is present in extreme condition also, and very diverse morphology. They are potential source for the bioactive compound and also important for the industrial enzymes. A screening of novel microorganisms for the lipolytic enzymes which is extracted from the indigenous, isolated from the indigenous substance. So here we are targeted on lipase, because a lipase that is a very good enzymes which can be useful for the cutting of oil, as well as it is also useful for the biodiesel productions. So how a sample collection from the cotton stock cotton seed oil industry, refinery industry situated near the Kadi, Gujarat. So a sample is collected from the indigenous refinery where the cotton peeling and cotton oil extraction is carried out. All substock sample is collected and site washer discharge and pipe in the sterile container. Now we are go for the enrichment. So Bushnell and Hayes medium using 100 gram of cotton seed oil substock were enriched in 100 ml Bushnell Hayes medium for the growth of indigenous microorganism, and we are putting in static condition for 37 degrees Celsius for 5 days. And after the enriched 1 ml of sample were incubated to 100 ml tributylene broth medium. TBA broth was inoculated at 37 degrees Celsius in static condition for 48 hours, and then after enrichment performed over the 7 days incubations. Now the methods that is applied for the isolation of lipolytic actinomycetes, a serial dilution from 10 power minus 1 to 10 power minus 10 were prepared and from the enriched sample, and this dilution were used in sprayed on the tributylene agar plate 100 microliter and incubate for 37 degrees Celsius for 7 days. Normally after the 2 to 3 days the lipase production is going on, and actinomycetes are the slow grower, that's why we are putting to 7 days. Various fungal as well as actinomycetes also isolated from the this culture. So fungal is separately isolated from the potato because it is a mix, so fungal organism can also be grow. Now lipase production by actinomycetes, that is a qualitative plate assay on the nutrient agar plate 1% tributylene or tributylene agar plate. Then culture plug was inoculated at 37 degrees Celsius for 5 days in the medium. Zone of clearance which observed around the colonies. The colony was observed due to the hydrolysis of tributylene by the lipase production. So this is a normal plate assay. The composition of production medium for lipase that are the containing the peptone is textured NaCl and cotton seed oil, and pH is a 7. Here the quantitative analysis of lipase enzyme under the submerged fermentation condition. 5 days old actinomycetes active culture grow on the TBA used for the inoculum. 2 plugs, we are using the solid plate is used, that's why our plugging system plugging method is used. Here the 2 plug actinomycetes culture were inoculated in 100 ml and culture incubated for 37 degrees Celsius. Culture filtrate removed from the flask, centrifuge 10,000 rpm for 10 minutes, 4 degrees suspension, and the enzyme activity was performed by using the substrate para-nitrophenol palmitate, and this is the substrate which is useful for the lipase activity, and the product which is released by the after lipase activity, that is a para-nitrophenol, which can be a measure at 410 nanometer because it is a yellow color. So this is the cellularizing another enzyme activity, that is we perform cellulolytic activity. 1% methyl cellulose is incorporated into the medium, and then a culture plug was inoculated after the 37 degrees 30 to 37 degrees Celsius for 5 days. We put one after incubation sprayed 0.1% Negro Congo red stain on the plate, so it can be visualize a colorless observation surrounding the colony. Then proteolytic activity same, milk substrate or you can say casein is added 1%. Then plugging method is again a zone of clearance we can observe. This is the identification methods which is we follow: molecular identification and morphological identification. Morphological identification by the staining as well as a biochemical method, and molecular identification by 16S rRNA partial gene sequencing, and phylogenetic tree was constructed by the help of BLAST methods. This is the optimization process. We optimize the culture for the media. So 5 days old culture on TBA was used. Mycelia, that is a plug with the help of actinomyce, with the help of plugging methods, and inoculate into the specific medium where the cottonseed oil is present. The culture filtrate removed. Yes, ma'am.
You have 1 more minute to go.
Okay, ma'am. So here the optimization process is carried out. Then after optimization, we have checked the different pH and temperature in optimization process. Also, we have checked the substrate, different substrate: groundnut oil, coconut oil, sunflower oil, tributylene oil, and olive oil. So this is the our result. You can see a clear zone is found. Then here the cellulitic activities, that is used by congruent methods. Then proteolytic activity by the using of casein media. This is the our graph shows that lipase activity, that is a quantitative analyze where the we can found 72 hours maximum lipase activity, and the p value is less than 0.001. And this is the identification method where we are applying the microscopic as well as a colonic identifications. Here the colony is a earlier white colony, then after aging it formed the gray color. So this is the molecular identification. You can see the molecular identified by the 16S rRNA, and here the Streptomyces caselix, that is a totally new organisms which is not completely matched with the these ancient organisms. This is our lipase activity which is found on the inoculum size. So here after 1 day, 2 days, the lipase activity is a maximum 25, nearby 25 units, and p value is a less than 0.01. pH, normally the pH is a you can see the 7 6 different pH we have taken. So here the pH 7, that gives the maximum lipase activity. In the case of temperature, normally nearby the 45 and 40 40 degree, 35 degree, and 40 degree. This is a optimization temperature. So here the 45-degree temperature and gives the optimum activity. Substrate, normally groundnut oil gives the very good activity in the culture. So here the conclusion is the cotton seed stock sample was the collected from the refinery, and we can isolate a novel organism actinomycetes, and it shows the very good enzyme profile, as well as it having a very good capacity to break down the oil, and it is a potent organism for the future development of biodiesel or some other product from the cotton stock. Thank you very much.
I believe Jayachandran sir is also.
Dr. Kamlesh.
Yes, sir.
Yeah, good work. This is having some
Thank you, sir.
application also. Very good.
Thank you, sir.
Shall I ask you one thing? Shall I ask you one thing? You have done the optimization by one optimization, the conversion technique you have used. Am I right?
Yes, sir.
Why not you use statistical modeling like Plackett and Berman and other things are there?
Sir, we have applied Packett-Berman theorem and used that, but it is already presented in a somewhat other conference, and it is a published paper. That's why I am not taking this part into this conference.
Okay, very good. And what about the results from the statistical
we have also started the application-based study, sir. In that we can successfully produce a biodiesel and another 4 actinomycetes, which is isolated from the substock, which is a very novel, and it having a very good capacity to degrade the phenolic compound also.
Okay. about the cotton soap stock, what about its composition, whether it varies from time to time?
Yeah, it is regularly because in nearby the Kadi there are a lot of oil refinery industry, almost 200 oil refinery industries surrounding the Kadi, and Kadi is a hub for the cotton. Near North Gujarat is a hub for the cotton productions. So here cotton production is also high, and normally the after cotton peeling the oil is extracted, and whatever the debris because it is apply a lot of things for the extraction of oil, like sodium hydroxides, then different chemicals is also applied, some solvent is applied. So the mixture of that waste material, that is a dark amorphous structure, and it is having almost 7% oil, because 100% oil cannot be extracted by the chemical method. So this soft stock normally use, if it is a higher content of oil, that is a useful the soap productions. That's why it is called the soft stock. It is a useful for the soap, but not 100% soft stock are used by the soap, because it is a if low content of oil, then it is a waste, and it is a dump into the various sites like water and soil. So it create the problem surrounding the ecosystems, and that's why we are taking the normal problems in the our work.
Okay, fine, good. Kirti teacher, please.
Thank you, sir.
The substrate. Okay. Kamlesh sir, what is the substrate you
Yes,
have
sir,
used
please.
for solid state fermentation? What are the substrates you have
Cotton
used
soft
for
stock,
solid state fermentation?
cotton soft stock itself, ma'am. It is a solid state fermentation which is we are carried out on the 10% soft stock, cotton soft stock itself.
Then I think you please see your slide 25.
Yes. This is a
This is why I'm asking.
enzyme activity which is we are carried out on different substrate oil, lipase
Oh yes,
activity.
yes, 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 yes. Okay.
It is a lipase activity, ma'am. So we are
Okay.
also focused. It is a cotton soft stock, but lipase is work on different substrate also. So we are study how lipase is a work on the other oil, groundnut oil, cotton oil, olive oil, coconut oil. So we are using different substrate for optimizing the lipase activity, and which substrate lipase gives the maximum activity. So normally groundnut oil gives a very good activity, lipase activity, ma'am.
Then what is the significance regarding this result?
Uh, can you pardon, please, ma'am?
You got coconut oil. The lipase is more active towards groundnut oil, isn't it?
Yeah.
Okay. Then what about
Yeah.
the significance related to this? What is the significance of your work?
Ma'am, we are study on the lipase activity. Apart from the organisms, we use a different substrate. So, which oil is the best? But in cotton soft stock, apart from cotton soft stock, there are other oil industry like groundnut oil industry, then soya bean oil industry. They also generate the soft stock, and this soft stock containing the oil. So we are thinking that in our area only the cotton oil industry, but in Saurashtra region, in south region, there are the different oil is extracted by the use of this same method, chemical method, and they also generate the soft stock. So if the cotton seed is apply cotton is used here, but somewhere the organisms must be work on the same soft stock, like a soybean soft stock, like a groundnut soft stock. That's why we are taking a different substrate for the lipase activity. It is a universal work, that is a different oil we are taking, because all oil industry work as the same for the oil extraction, chemical method is applied, and they generate the soft stock.
Okay, then can you correlate the structure this coconut oil and your substrate? Groundnut oil and your substrate. Is it identical chemically? How this
Groundnut,
enzyme will,
ma'am, normally in a
I'm asking how this enzyme will act on groundnut oil and your substrate. What is the action of enzyme?
okay. Ma'am, normally what we are doing, we are taking the organisms and extract the lipase enzymes and then put into the groundnut oil, that is a substrate, And then finally, we are checking the free fatty acids, which is a present.
It's okay. Yes, okay. Answer. Continue.
Okay, so apply the lipase enzymes and then we taste the free fatty acid. Then we are taking the oil for the solid state fermentation, and then we cultivate the organism and again find the enzyme production and the product. So enzyme is once it's generated, then we are starting the PNPA assay. So first we are screening and first we are checking the lipase activity, free fatty acids methods, that is a titrimetric method, and then after confirmation by the PNPA methods after
I'm
solid state
not
fermentation.
asking about the procedure. What is the principle behind, or this? Why this other oil not working with the more working with this lipase than this groundnut oil? What is why? What is the reason? That's why I'm asking.
Ma'am,
What is the oil
this
nature of
is
this?
a depend.
Yes.
Yes, enzymes. Lipase enzyme belongs to esterase activity. Different kind of esterase is
Yes.
there.
Yes. Yes.
It is a very
Yes.
big class. So whatever the organism is generated, whatever the organism lipase is generated, that we are just to use the different substrate, whether it is a work or not. It is a very good work in the groundnut, but not in a work in sunflower oil. So this is a our
That's the
study. We are
reason.
not
That's the
working on
reason.
one organism,
I am asking the reason.
and this is a study, and that study shows that if we are combine the organisms, we have a 35 isolates bacteria. So if we are making the consortium, that it will be a good product for the degradation of various substrate. So this is one of the part of that study, man.
That is this lipase. Your
We
lipase
are making the
is
consortium.
your lipase function is oil specific, or
No, 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 no. We are not working on oil specific. We are working on the degradation of oil present in the substrate, and it can be converted into biodiesel or biochar, or it can be useful for the glycerol production. So it can useful for the cosmetic industry.
okay, okay.
Okay, Kirti ma'am. Thank you, Dr.
Thank you.
Kamlesh Kumar.
Thank you. Can I leave, ma'am? I have some work
Yes.
also. So can I leave?
Yes, sir, you can.
Please. Thank you, ma'am.
Okay, the next participant, OPFC3, Dr. Sona A.
Yes, hello.
Yes, ma'am, you are audible. Can you please share your screen?
Yes, I'm sharing.
Yes. Can you put it on the slideshow?
Yes.
Okay, ma'am. It's
Shall I
clear.
start? Okay.
Yes.
Respected
Yes, ma'am, you can start.
Respected chairpersons, members of the organizing committee, and dear participant friends, I am here to present the topic phytochemical analysis and antibacterial activity using N-hexane extract of the leaves of Asparagus racemosus. The work was done by Megha for a MSc project. Asparagus racemosus, commonly known as shadavari, is a perennial shrub belonging to the family Asparagaceae. Its active constituent is steroidal saponins called shadavarins 1 to 4, isoflavones, asparagamine, racemosol, polysaccharide, mucilage, vitamins A, B, C, E, folic acid, minerals like magnesium, phosphorus, calcium, iron are also found in the roots. It is found to have essential oils, asparagine, arginine, tyrosine, flavonoids like camphorol, quercetin, and rutin, resin, and tannin, reported by Chawla et al. Presence of alkaloids, flavonoids, steroids, and terpenoids in the percent 30, 47, 65, and 43 respectively by Verma and Parashar. The leaves and roots of the plants was used for phytochemical screening, and all plant parts were found to contain alkaloids, terpenoids, and saponins and flavonoids by Pascaline. It is used in the Ayurvedic medicine. It is having anti-stress properties, anti-diarrheal, adaptogenic, anti-ulcerogenic action, antioxidant, and cardio. Able to cure the reproductive and hormonal issues in women, phytoestrogenic properties to cure disease, and also to cure disease like osteoporosis has been reported by Asha Jyoti et al. It is a potent antioxidant, immunostimulant, anti-dyspepsia, and anti-tussive effect, and it is having galactagogue effect, anti-hepatotoxic effect, and immunomodulatory activities. The aim of the study is to study the phytochemical constituents and antibacterial activity of Asparagus racemosus, and N-hexane extract was used for this. Leaves of the Asparagus racemosus were collected from the local areas of Tannitod Grama Panchayat in Koni of Pathanamthitta district. The leaves were dried, air dried, and powdered, and the powdered form was used for the extraction. 5 grams of the leaf powder was extracted with 25 ml of N-hexane, and the following tests were performed. Tests for steroids, glycosides, cardiac, anthraquinones, saponin glycosides, alkaloids, tannins, phenols, flavonoids, terpenoids, and saponin foams. For determination of the antibacterial activity, 2 positive strains, gram-positive strains, Staphylococcus aureus and Bacillus, and gram-negative strains, E. coli and Pseudomonas was taken, and McConkey agar was taken as a media, and susceptibility test discs were prepared with the asparagus extracts, and antibacterial assay was performed. So these are the results of various phytochemicals present in N-hexane extract. For steroids, a dark green layer was obtained. For glycosides, dark green color. For cardiacs, reddish brown ring. For saponin glycosides, there was absence of a persistent foam, so the test was negative. For alkaloids, a brown precipitate. For tannins, light green color. For phenol, dark green color. And flavonoids, there was absence of red color, so the test is negative. Terpenoids with light green color. And saponin foams, there was absence of honeycomb-like froth, so the test gave negative result. And for anthraquinone, reddish at the top layer and blue bottom layer. Antibacterial activity was done against the 4 strains, E. coli, Bacillus, Staphylococcus, and Pseudomonas, and with the gentamicin as positive control and the extract as the negative control. The asparagus gave a zone of 5 mm against the E. coli and Pseudomonas, whereas with Bacillus, 10 mm zone was measured. However, not though as effective as gentamicin. So these are the results. Phytochemical analysis revealed the presence of steroids, glycosides, cardiac, alkaloids, tannins, phenolics, terpenoids, and anthraquinones. However, saponins, flavonoids, and saponin foams was found to be absent. Some papers have been reported to contain all these components also, maybe because of the geographical location. These samples, these components, phytochemicals were not being detected here. Then the maximum zone of inhibition was against Bacillus and Staphylococcus with 10 mm, while only 5 mm zone was against Bacillus and Pseudomonas. So from the study, it is evident that the leaf of the plant possesses major phytochemical constituents and is effective against the bacterial strains, Bacillus and Staphylococcus. So on comparison, it was more effective against the gram-positive strains. This study supports the medicinal importance of the plant. Thank you.
Okay. Okay, Sona teacher, good,
yes,
very good presentation. Teacher, shall I ask one thing? Whether can you just suggest which of these constituents are responsible for this antibacterial activity? Have you gone to that extent? Have you separated these constituents?
No, sir, not isolated, sir. It is just a qualitative analysis only.
Qualitative. You have tested the crude extract as such.
Yes, sir.
Okay, good, good, very good. Kirti teacher, please. Kirti teacher.
Okay, so okay. Sonia teacher, okay. A small good piece of work. Okay.
Yes,
It may be your MSc student. Yeah. Okay. It is your
yes,
MSc
yes,
student.
ma'am.
Okay, good. I have one question, that is this, asparagus, which is already traditionally used Ayurvedic medicine. Okay.
Yes, yes, ma'am.
In what type of
Mainly
diseases we
roots.
use? Yeah. Okay, root.
Yes, ma'am.
For
In Ayurveda, for gastric problems, even for female reproductive disorders, as hormonal imbalances, for all these it is being used.
Oh, okay, good. Then you have used pathogenic bacterial strains. Okay.
Yes, ma'am.
What is the source of this pathogenic strains? From where you collect it?
It is being maintained in our college laboratory.
Okay. You may purchase from somewhere, isn't it?
Not but that is, it is being subcultured and maintained.
It's already available in your department.
Yes, ma'am. Yes, ma'am.
What are there are a lot of bioactive compounds present in this leaf extract, isn't it?
Yes, ma'am.
Okay. You have performed antibacterial activity.
Yes, ma'am.
What is the bioactive compound responsible for this this antibacterial activity?
That I'm not sure, ma'am.
Okay.
Maybe because of all these phytochemical constituents, but I am not sure what is actually
A lot of
responsible.
phytochemical compound you have already
Yes, ma'am.
checked. Okay.
Yes, ma'am. We didn't get
Which
flavonoids and saponins only. All others were present.
Okay. Okay,
Okay,
my
ma'am.
question is over.
Okay, ma'am. Any more questions? You can take one more question.
Chat box question.
I think there is a question in the chat box. Can you read it, ma'am? Sona, ma'am?
Why selected N-hexane? Yes, we also selected three other solvents. For this, I am presenting only with N-hexane. We had done extraction with petroleum ether, acetone, water, and N-hexane. So for this, I have given only the N-hexane part.
Okay, are you done, ma'am? Any more questions in the chat box?
No, Edna.
No.
Okay. Okay, then. Thank you, ma'am.
Okay,
thank you, Dr. Sona A. Next participant, OPFC4, Dr. Harish Chandra Anandaram. Sir, please join. Dr. Harish Chandra Anandaram. Sir, are you there? Okay, then we can go to the next participant. No, sir, you're not audible. Dr. Harish Chandra, you're not audible now. Yeah, now you're audible.
Okay, fine then. Okay, I'll be sharing my screen.
Okay, sir. Please share your screen. Sir?
Yes, I'm sharing my screen.
So it's not visible.
Yeah, wait a minute. I'm just once again trying it. Is it okay now? Screen sharing?
So it's loading.
Yes. This now visualize.
Yes.
Okay. Okay, sir. You can put it in the slideshow.
Yes.
Sir, please stick on to the time limit.
Okay, fine.
So are you ready to start?
Yes. I'm ready to start. Okay, fine.
Okay, sir, you can start now.
Actually,
Excuse me, one clarification. Excuse me, one clarification. You are research scholar or you are faculty?
now I'm a faculty. Recently submitted my thesis.
Okay.
Fine then. Actually, in my abstract, the title is given one approach, but since I've completed all 4 approaches and got my degree, I'm presenting all approaches here. And the overview is I'm trying to understand the regulation of microRNAs with respect to the association of pharmacogenomics and psoriasis. And guided by Dr. Daniel Alexanand, and DC members were Dr. Muniraj and Uma Maheshwari, and the Indian examiners from IIT Madras, Dr. Karthik Raman, and the nominee of the foreign examiner is Dr. Jayaganthan, and the foreign examiner is Dr. James. Fine. And with respect to the basics of psoriasis, as everyone knows, it's an autoimmune disorder, and both genetic and environmental factor play a role in it. And system biology is required to understand the intermediate players in the pathogenesis of psoriasis. And the process of gene regulation is not only an occurrence in pathology, it's also an occurrence in physiology. And all macromolecules constitute for the gene regulation. It's not the DNA alone. And with respect to the prevalence of psoriasis in global level, it's twice more common in males compared to females. Until now, there is no reliable biomarker to diagnose or treat psoriasis, and the whole world is working towards it. And with respect to India, it's only between 0.4 to 0.7, and in world population, it's 2%. And there is prevalence that it may increase to 2.8 in future because they are getting more cases nowadays. And then with respect to the recent developments, it is not only a single disease, it can also give rise to obesity, metabolic syndrome, and other diseases based on the genetic factors. And that's how the psoriatic March map is constructed for analysis. And with respect to the recent developments, the HapMap project and GWAS studies gave us a lot of insights to work in these areas. And then the SNPs were the major trigger biomarker for pharmacogenomic outcomes. So SNPs were more focused. And with respect to various approaches, we have looked into the text mining of the literature associated genes, as well as the genes in the differential gene expression studies, as well as the small RNAs in the differential gene expression, and then the transcription factors in the associated pathways. So I have formed all these 4 approaches based on the network construction and analysis to identify its significance. And this is a direct approach where the variants of the SNPs were taken and performed further analysis. And in indirect approach, the pathway associated transcription factors were taken. And obviously, the pathways were reanalyzed with respect to the genes in David. And as you can see, though there is a link between this gene and mRNA, only very few are thermodynamically feasible and shows complementarity. So those specific hits were only taken. And you might wonder, like he has taken only from database and tools and expression studies. Is there any valid argument for the specificity of the transcription factor? Yes, human genome browser, we have verified the presence of transcription factors in those genes and identify the specific factors and got certain specific network motifs with respect to the factors. And STAT2 were already reported and we got in our analysis also. And similarly, it's not only mRNAs were involved in post-transcriptional repression, it's also involved in translational repression and regulation. So this specific mRNA, which has not yet reported previously for the disease, and we are reporting in our study. And then, as you can see from our reports, when we try to correlate with the experimental studies in previous report, the regulation of ACTLA transcription factors is to be explored in psoriasis, so that we are considering as a novel target to treat psoriasis. And then with respect to the protein-protein interaction, because you might wonder, all transcription factors of proteins, but not the vice versa. So we have analyzed the associated proteins also for finding the functional enrichment between the ontology terms. And as you can see in some approaches, we are getting the similar pathway, cytokine chemokine interaction for our study. And then with respect to our results, we have proposed a dynamic of a model to understand how the regulation changes with respect to time, and simulated the model. And then we have got the n-dimensional network, and by using some statistical parameters and experimental criteria, we have converted into a two-dimensional network where we have got some specific hit, like WIF1 is also a gene which is actively explored in psoriasis, and some specific genes like STAT3 and all were already explored, and the associated factors were further subjected to experimental analysis. And similarly, for this gene, as you can see, there are most of the mRNAs form a triplex, so it's vital in psoriasis. And with respect to the scope and significance, this particular transcription factor performs the activation function in this gene, so the mathematical model can be formed between these three factors. And the specific regulatory motifs, now we have considered various biomarkers and how the regulation happens in respect to pathogenesis to reconstruct the pathway. These are the specific network motifs which we got from this approach. And then with respect to many proteins, there are only few interactions to form the specific ontology term to connect with the disease. And similarly, a mathematical model is reconstructed with respect to the co-expression also. And then with respect to SNPs, there are very few SNPs of pharmacogenomic specificity for the psoriasis associated genes, and we got those hits. And as you can see, some comorbidities were also present, and once again we checked for the indexing of comorbidities, whether the comorbidities are highly relevant or not with respect to clinical significance. Then only we have taken those comorbidities like rheumatoid arthritis and type 2 diabetes and systemic lupus erythematosus are the comorbidities. And as you can see, with respect to various comorbidities, IL-12B has more association with both psoriasis and arthritis. So that was taken to explain the concept of epidermal hyperplasia, main pathogenetic pathway to be analyzed. And then, as you can see, cellular communication is the highly associated ontology term with respect to various association. And with respect to the pharmacogenomic hit, the specific identity which we got is the MTHFR gene, which is yet to be explored in psoriasis. Only one particular variant is studied, and further studies are done all over the world with respect to this gene on psoriasis. And then it has 3 sites for binding compared to others and all where they have 1 or 2 sites, so that only it has got more specificity in the study. And 125A3P was explored in cancer, but it is yet to be explored in psoriasis with respect to pathogenesis, and we are exploring now from our study. And this particular transcription factor act as a hub in connection. So while modeling, we are giving a higher weightage for this factor. And similarly, the associated motif in forming triplets. And finally, initially we have done in the gene level. Now we are constructing in the system level how the epidermal hyperplasia can be modeled with respect to this. So we have formed a mathematical model to explain the thing, and more experimental works are required to
Sir,
establish a model. Yes.
you have 1 more minute to go.
Okay. Okay. Fine. I'll finish it off. And then the whole pathway reconstruction is done from modeling the network. And the subnetwork is constructed with respect to the modules using M-code. And then whether all the regulatory pathway were directly connected or not with respect to psoriasis experimental studies, we have gone the cascade of results. And finally, the forms this consolidation and specific hubs and the associated process in this network. And these are the key results from my finding, which was not reported. We'll be reporting in. And the result, the study has aided a basic understanding in analyzing the complexities involved in pathogenesis, and we have got a lot of factors which can be further studied in the network to explain the entire pathogenesis. And obviously, the optimization require more biological experiment to standardize. Okay. And further work is like simulation of this model with respect to more parameters and further analysis to establish mRNA as a specific marker in pathology. And these are some of the works we have published, presented it. And these are the key results in various publications. And with respect to the queries of Indian and foreign examiners, like purpose of modeling and those things are analyzed. And I'm done with my session and hope you can go for discussion.
A humble request to all the participants, kindly remain muted.
Harishankar,
Yes.
the PhD is being awarded, isn't it? Or you have PhD is being awarded. You are now Dr. Harishankar, isn't it?
Yes, sir.
Very good, very good, very good. Excellent work now. Very good work. Congratulations for that.
Thanks, sir.
Specifically, the mathematical modeling which you have done, comparing regulation with respect to time, is very interesting. Very good. We appreciate that work.
Thanks, sir. Because most people work on expressions, sir, but regulation is the initial cascade to get the outcome of expressions. Sir. In that area, I'm more interested.
Very rarely we see mathematical modeling being applied, and this is a very good idea. That's what I understand from that. Normally, your approach is exclusively, the way in which you have applied modeling to gene expression. Wonderful. Anyway, what about the stat1, stat2, you have identified, you have traced 92 genes. That's what I understand. 92.
Actually, sir, initially I have identified 92 genes, sir, and STAT2 were well explored actually. Only ACTL8.
Already done. Right,
yes, sir, it has been proved. Only ACTL8 and MTHFR were unexplored, sir. And the mRNA.
What about WIF1? What about that?
Yeah, WIF1 is also explored in cancer, sir, but not in psoriasis.
Oh, right. But you are the one to start with WIF1?
Yes, sir. Actually, it's a part of collaborative projects between us and Georgetown University, USA, and IIT Madras. Sir.
Okay. So CTCF is a transcription factor of WIF1. That's what you said.
Yes, sir. It's a transcription factor of WIF1 actually.
Yes. So CTCF is having a regulatory role in other genes, or is it highly specific to?
Actually, sir, no, it has role in most of the genes, sir, but the number of binding sites with respect to WIF1 is more, sir. It has 3 binding sites actually, if you look into the human genome browser compared to other genes.
So very good. Congratulations. Good presentation. Kirti teacher, please.
Yes, excellent work, Dr. Harishankar. Okay.
Thanks, ma'am.
I have asking some question related to this. Actually, we know this psoriasis. It is a especially genetic disorder. You have mentioned. You already mentioned that in nowadays in India the ratio is increasing, isn't it?
Yes, ma'am. From 0.4, it has gone to 2.8, ma'am, in the last year.
Okay. Okay. What may be the reason? Any environmental factor also involved in this?
Actually, ma'am, environmental factors are involved, but in most cases, when we consult with doctors, ma'am, lack of vitamin D, ma'am.
Oh yes, okay.
Because lifestyle and other things and all, most doctors gave report, and instead of using analogs, ma'am, some people recommend direct contact of vitamin D itself. Because some doctors, what they say is, patients know in between 11 to 12, if they have a sunlight exposure, the lesions are reduced actually.
Then another question, why males are more susceptible than female? Any?
Yes, ma'am. With respect to genetic organization, ma'am, it is more prone to Y chromosome, ma'am, actually, compared to the X chromosome. Not only psoriasis, most of the autoimmune disorders.
Okay. Thank you.
Dr. Harish Chandra, you can
Yes,
just check the chat box. There are a few questions in there. You can take one or two.
okay, fine. Well,
Ekta,
one
you
person
can read the
asked
question.
about, yeah, vitamin D. Is it vitamin D3 means, like, not specifically vitamin D3, more all subclasses. And then with respect to the specificity of gene suppression, it is, we didn't use BLAST actually. We used only the human genome browsers tab browsing option.
Okay, sir, that's it.
And then
So,
okay. And then, like,
thank
we have not performed cell culture. We have performed only the RNA-seq and the whole genome based annotation for our RNA-seq experiments. Okay, fine. I think I am done with the chat box questions.
okay, sir. Thank you. Thank you very much, sir.
Okay, fine.
Chairpersons, please note, contestant number OPFC 8 is absent. Chairpersons, please note, participant OPFC 8 is absent. Now we'll move on to the next participant, OPFC 5, Dr. Ranjita K.
Can I start now?
Yeah, you are audible, ma'am. Can you please share your screen?
Sure. Is it okay, ma'am?
Ma'am,
Hello,
it's not visible.
It's not visible, right?
Hello, Dr. Ranjita. Please upload
Yes.
your slide.
I'm doing it. Is it visible now?
No, it's not visible. Till now, it's not visible. Yes, it's loading.
Yes. Okay.
Okay, ma'am. Ma'am, you can start now. After putting it to the slideshow, please start.
Yes, ma'am. So, good morning, everyone. I'm Ranjita. I am from Division of Post Harvest Technology and Agriculture Engineering of ICR Indian Institute of Horticultural Research. So, I would like to share.
Ma'am, can you please put it in the slideshow? Okay.
Yeah, it is.
Okay,
Ma'am.
ma'am. Okay, okay, ma'am. Thank you, thank you.
So today I will share a small piece of work done as an institute project program going on here. That is the microbiological changes in fresh cut papaya and enzymatic properties of the fruit endophytes. So we have a project on standardization of protocol for different fruits. So this particular one, that minimally processed papaya, also have the similar challenges of spoilage, because once if a mechanical damage or the tissue injury happens during the cutting peeling operations, definitely it leads to the further biochemical changes of the fruit, and then it goes to the further physiological aging and microbial growth. Fresh cut, why I have done this experiment was that nowadays the demand for this fresh cut papaya and other fruits are increasing owing to the change in the lifestyle. So the first issue what we addressed was to improve the shelf life of this fresh cut papaya, and we were successful in that by using a protocol in which there was an antimicrobial agent and then going for a passive modified atmosphere packaging. In passive modified atmosphere packaging, we help the packages to have, or the food to have, a optimal carbon dioxide and oxygen levels inside the packages, so that the respiration is reduced, and thus the overall quality can be maintained for a longer time. And ICAR has developed a special variety called Arka Prabhat variety. This has got the distinct features for minimal processing use, and with this particular variety and the optimized protocol, we could get a shelf life of more than 10 days actually at commercial open chiller temperature, which is the temperature usually maintained in supermarkets. So this is the difference we can see. 10 days of shelf life we could get. The controlled or the untreated ones, there were a lot of fungus as well as softening of the tissue. But in the optimized one, the last one, these are different stages of optimization which is displayed here. The last stage, definitely the quality is much better. And the major objective of the project, today's presentation, is the microbial aspects of the study. The background of the study was that the sustained release of antimicrobials throughout the storage period was essentially required for the enhancement of shelf life. So, as a food technologist or as a food microbiologist, always our aim is to reduce the antimicrobial chemical dose or the treatment. So one of the possible aspect is that improving the hygiene during the processing should help in prolonging the shelf life and improving the microbiological quality. So for that, whether that will help or not, we have done by cutting the papaya in different conditions. One was in totally sterile condition, the other one was in non-sterile conditions, and both of these lots were packed in antimicrobial containing system as well as a system without antimicrobial. But when we checked the microbiological quality during storage, what we have seen was, irrespective of the process hygiene followed, the aerobic plate count or yeast and mold count or lactic acid bacterial count has increased without the use of antimicrobials. That means there is endophytic proliferation in the tissue in the fresh cut foods, even though process hygiene is followed. Therefore, it's essential to provide a continuous source of antimicrobial during storage. Then we studied what is the role of these endophytes. What is the role of these endophytes in the fruit? What could be the probable role? So for that, we studied the enzymatic characteristics such as amylase, cellulase, esterase, pectinase, and ACC deaminase. Among these first four enzymes are hydrolytic enzymes, which are normally present in general in most of the organisms, I should say. And among these four hydrolytic enzymes, maximum presence was of esterases. That itself indicates that these endophytic organisms or those organisms which are colonized within the naturally within the tissue of the food has got high potential of aroma development when the food ripens. So it leads to the theory that probably it's not only the food that itself is responsible for the flavor development during the ripening. It's the microbial part of it, like the gut microbiome. The food microbiome is also helpful in flavor development. And another thing is another enzyme, that is ACC deaminase, that was invariably present in most of the isolates which we have. In most of the isolates which we have obtained. So this is another aspect which we wanted to study further. Actually, ACC deaminase enzyme is an ethylene curbing enzyme. As I hope most of you are aware that ethylene is the senescence or the ripening hormone in plants. ACC or the aminocyclopropane 1-carboxylic acid, that is the precursor of ethylene. Once if ACC is formed from S-adenosyl methionine, and if ACC aminase is present, ethylene formation is stopped because ACC is converted to ammonia and alpha ketobutyrate by this enzyme. So it is very likely that these organisms are helping in prolonging the senescence of the fruit, or they want to have a better life for themselves. In turn, it helps the fruit for improving the life of the fruit itself. So then the next aspect of the study was like, is it possible to modulate or delay the ripening through the activity of ACC deaminase producing microorganisms? Even we have tested it using a model climacteric fruit, or the fruit where there is an ethylene surge during the ripening process. And usually for post-harvest management, the model fruit or the Drosophila of post-harvest management scientists are tomato. Even though it is a papaya isolate, we have studied that in tomato, and then we found that when the isolates were applied to harvest maturity or the harvest or the tomato which was freshly harvested in the breaker stage, it delayed the ripening. So this is as far as I know, this is the first time such a report comes out. So we got the proof of concept in a model climacteric fruit, that is the bioagents can help in delay of fruit ripening. Definitely it is it shows the potential of using the endophytes for modulating the fruit ripening and post-harvest management. So far, a certain chemical ethylene inhibitors are used. None of them have got any permission or registration in countries like India. So bioagents, if they are safe, or the GRAS status, what we have studied was also lactic acid bacteria obtained from the fruit itself. So there is a possibility that we can increase the shelf life or delay ripening of the fruit using bioagents. And another thing is, we should have a relook into the microbiological criteria in fresh cut foods and vegetables by the food regulatory agencies. It is just showing the Eastern molds or the bacterial count or other, mainly Eastern mold and bacterial count only. Actually, with the ripening of the fruit, even if it is very safe to eat also, usually the endophytic organisms proliferate. The cutoff or the criteria given is a very, very low level. So that also has to be relooked into. The third aspect is that we can study the... Yes, ma'am?
You have 1 more minute to go.
Okay, done, ma'am. Another thing is that the food microbiome in relation with the gut microbiome is to be studied, because these raw fruits or the in general fresh produce are the major sources of probiotics or any kind of organisms to the human body. Thank you. So I'm done with the presentation, ma'am.
Okay, madam. Thank you for your active participation in the program. It was really good. Yeah, increasing the shelf life with antimicrobial agents, and then again checking for endophytes. So, how many endophytes you put? Have you isolated any endophyte?
Yeah, we have isolated set 150. That enzymatic study what we have done was 150 numbers of isolates of each group. One was general bacteria which was grown in plate counter. The second one was yeast and molds that was grown in PDA, and the third one was lactic acid bacteria which was grown MRS. And whatever study further we have done was all using lactic acid.
Oh, you are going to proceed with the lactic acid bacteria. Oh, that's okay. I think
Yes,
the probiotic effect can also be evaluated. No, the probiotic effect can also be evaluated.
That can also be tested. So that can also be tested. My major intention was why these organisms are naturally present inside the fruit, whether they have got any role in the development of the fruit itself.
Yes, very interesting. And we can apply the same thing to other cases also. Your approach can be applied to other cases. Yeah, very good. I think Kirti teacher will be asking more about probiotics and its significance in the work. Kirti teacher, please.
Yes, okay. Good piece of work, madam. Interesting, very interesting. I have heard a new term, that is fruit microbiome. I am very much interested about this. Okay. You got a lot of organism from this papaya, isn't it?
Yeah, yes, ma'am, it comes. That's why even after, whether it is properly cut, actually that sterile environment during that processing was done within the laminar flow. Even then, the number of microorganisms that proliferated during the storage of the produce was equal to almost similar level to that of the one which was cut just outside it, I mean in the regular room. That means all these organisms are present within the fruit itself.
Then what is your conclusion? That is, all this organism may function either singly or in combination during this development of this papaya fruit?
Yes, yes.
In all stages?
Yeah, definitely it will be there, man. There are not much work done actually. Even this.
Okay, you have to study. You have to study one by one, isn't it?
Yes, yes. Even
Okay.
IAA production was also observed by this organism. Definitely, fruit development is an interplay between ethylene and IAA.
Okay, you have studied the organism which are producing ACC deaminase.
Yeah, mainly that one, because invariably 99% of the organisms possess with this particular enzyme activity. And normally it is reported only in the PGPR, rhizosphere organisms. They tell it's one of the characteristics contributing to the PGPR plant growth promotion activity. And when we check, it is present even in the food microbiome endophytes also.
Maybe it is applicable to all other food. It is present in all other fruits.
It will be there, and their levels also will be definitely different from different stages of development. And if you see that earlier slide, in that particular one, I have seen that the pectinase enzyme was totally absent. Again, the pectinase is the one which helps in
ripening, for ripening. Okay.
Softening.
Ripening and food softening.
So these organisms do not have that organ because pectinase.
So that means it will help for increase the life, shelf life.
Pardon me?
It may help for increase the shelf life.
Yeah, that's a possibility. Yes, yes. Delay ripening, because several times
yes.
management is to be practiced when during transport of this product, foods and all. There is a need for delaying ripening.
Then how can this study can use in what is the application level? What do you think about in next days?
Post harvest management, ma'am. Like we are a horticulture institute, so we know how difficult it is. For example, mango. From green stage to the ripening stage, it has to remain in the green stage for a longer time, so as to facilitate the transport of this mango from a cultivated area to the place of consumption. And we use a special ethylene curbing agent called 1-MCP for our experimental studies, even though it's 1-methyl cyclopropene. it actually binds to the ethylene sites.
So yes, I can. You can develop a consortium as a bioactive agent consortium, isn't it? Am I correct?
Yes, ma'am. Yes, we can do that. So now it's done only in the typical model fruit, that is tomato. Definitely, with more studies, we should be able to extend it. Actually, I have not seen any such reports. Even it's a
Very
small
good.
study,
Okay, yes. I'm also
probably
first time
not
heard
more
about this
reports.
fruit microbiome study. Okay, good, madam. Okay, thank you.
Thank you, ma'am.
Any more questions? I think there are no questions in the chat box also. So thank you, Dr. Ranjita K.
Thank you, ma'am.
So the next participant, OPFC6, Dr. Bindu B. Dr. Bindu?
Yes, can I
Yes, you are
start
audible.
my presentation?
Yes, you are audible. You can put in the
Good
slideshow
afternoon,
and start.
Good afternoon, most respected dignitaries. Myself, Dr. Bindu B, Assistant Professor, working in Kerala Agriculture University, in Farming System Research Station, Siddhantapuram. Topic I am going to present is the integrated nutrient management of papaya, use of biofertilizers for enhancing its yield. Papaya is one of the most emerging fruit crop of Kerala, and it is one of the fruit crop that has become very popular because of its nutraceutical properties. It's a highly nutrient responsive crop, and balanced nutrition play a very important role on plant growth, its yield, and fruit quality. Under high-intensity cropping system, the interactive advantages of combining organic and inorganic sources of nutrients in integrated nutrient management have proved to be superior to the use of each component singly. And also, there is a pressing need to enhance the productivity and improve the quality of the papaya because the crop is now becoming very popular nowadays. So the studies have shown that adoption of integrated nutrient management practices in papaya has increased its fruit yield and quality. In addition, that we are using organic manures also. In that aspects also it increases the soil or organic matter content and also microbial content, and finally results in the enhanced yield. So the experiment is conducted to study the effect of integrated nutrient management on growth, yield, and quality of papaya, and to assess the changes in soil nutrient status due to the integrated nutrient management. And the trial is conducted in a randomized block design with 11 treatments, and it is replicated thrice. And the number of plants, observation plants per plot was 4, and spacing adopted is 2 meter by 2 meter. And the variety selected for this trial is IHR papaya variety Suya. The treatment includes T1 is the 240 is to 240 to is to 480 gram NPK. It is a recommended package of practice of KU. And T2 instead of the N 25% is replaced as neem cake enriched vermicompost. Along with that, 75% of the N and full dose of phosphorus and potassium is given as inorganic fertilizers. In treatment 3, 25% of AN is given as poultry manure enriched vermicompost, and along with that, 75% of the recommended dose of nitrogen and full dose of phosphorus and K is given as inorganic fertilizers. Treatment 4 consists of the 25% of the azospirillum enriched vermicompost. Azospirillum is nitrogen fixing bacteria, and along with that, nitrogen of 75% and full dose of potassium and phosphorus is given as inorganic sources. And treatment 5, it composed of 25% of the NPK requirement of the plant is met through PGPR mix 1. PGPR mix 1 is a biofertilizer. It is an NPK biofertilizer, and it consists of the N fixes, nitrogen fixes like azospirillum, azotobacter, and along with that it contains phosphorus solubilizer like Bacillus megatherium and K solubilizer like Bacillus species. So along with this PGPR mix enriched vermicompost, we are giving 75% of the recommended dose of NPK as inorganic sources. And treatment 6 consists of nitrogen requirement 50% is met through neem cake enriched vermicompost, and along with that 50% of the nitrogen and full dose of phosphorus and potassium is given as inorganic sources. And treatment 9 consists of 50% of the nitrogen requirement is met through poultry manure enriched vermicompost. Along with that, 50% of the nitrogen and full P and K is met through inorganic fertilizers. And treatment 8 compost 50% of nitrogen as azospirillum enriched vermicompost. Along with that, 50% of nitrogen and full dose of P and K through inorganic fertilizers. And treatment 9 compost 50% requirement of NPK is met through PGPR enriched vermicompost. Along with that, 50% of nitrogen, phosphorus, and potassium is met through the inorganic fertilizers. Treatment 10 consists of line application only, and treatment 11 is absolute control. And format manure 10 kg per plant as per the recommendation will be given. It has been given uniformly all plants except the treatment 10 and 11, and the land requirement is calculated based on the soil test. And the inorganic sources of fertilizers are urea, crash course, and muriate for NPK respectively. And this manures and manures are given as based on the nitrogen equivalent basis. And these manures are applied to plants at the interval of 2 months. The 6 splits is given, and first fertilizer application started 1 month after transplanting of the seedlings to the main field. In addition to that, biofungicide Pseudomonas fluorescens as foliar spray 2% and Trichoderma verde 5 gram per is also biofungicide 5 gram per plant as basal application was given all plants except Q10 and Q11. And the main observations are noted during the study are biometric characters, yield characters, quality characters, soil analysis before and after the experiment, plant analysis, pest and disease incidents, economic analysis, and statistical analysis. These are the different stages of the crop growth. As if you view, then results shown that at all stages of the growth, the plant height is highestly reported by the treatment T5, that is, the 25% of the nitrogen requirement is met through PGPR enriched vermicompost, along with the 75% of the recommended dose of P and K through inorganic sources, and the least plant height was noted in the absolute control. And same treatment gave us best performance in the with regard to the girth of the plant studied during the all stages of the crop growth from 2 months up to 1 year. And regarding this number of leaves formed, it's also the same treatment reported highest and lowest, highest number of leaves and lowest leaves are reported from the absolute control. And regarding the height at first flowering, lowest height at first flowering and also lowest duration and least number of days for the first flowering was noticed in the treatment, that is 25% nitrogen requirement is met through PGPR enriched vermicompost, along with the 75% NPK through chemical fertilizers. The same is also the treatment give highest fruit set percentage and least number of days for harvest. Regarding the yield parameters studied, highest fruit weight, fruit length, girth, and also this is the best treatment, that is PGPR enriched vermicompost, along with 75% of recommended dose of NPK. And also highest fruit volume, pulp percentage, and the number of fruits per plant was also reported highest with the treatment PGPR enriched vermicompost 25%, and also 75% NPK as chemical fertilizers. And this treatment also gives the highest total number of yields, and it reduced the days for maturity. And regarding this fruit quality parameter study, it's shown that highest TSS, least acidity, highest carotenoid content, highest ascorbic acid content, and also highest total sugar and reducing sugar content of the fruit was reported from the treatment, that is PGPR enriched vermicompost, along with the 75% of NPK through chemical fertilizers. This is the best treatment, that PGPR enriched vermicompost application treatment. These are the absolute control, fruits from the absolute control. And regarding this
fruit color of the peel, fruit peel, it ranges from yellow to deep yellow, and pulp color ranges from orange red to deep red, and firmness of fruit it ranges from fairly firm to firm. And regarding this shelf life, integrated nutrient management doesn't have any significant role in this in controlling the shelf life of the fruits. And we have also conducted the organoleptic evaluation of the papaya fruits, and it seems that overall acceptability of the fruits regarding the appearance, color, flavor, taste, texture, and less papain odor, these are all seen in the treatment. That is 25% N requirement that is met through the PGPR enriched vermicompost, and rest 75% NPK through chemical fertilizers.
Ma'am,
And regarding
you
this,
have 1 more minute to go.
okay, soil pH is improved content, and regarding the soil NPK content, concerned micronutrients, copper, iron, manganese, and zinc content also increased by the application of this PGPR enriched treatment. And we have taken the soil microbial count also. It seems that bacterial count, fungal, actinomycet content was increased in this T5 treatment. And regarding the plant analysis, plant NPK content and plant microbial content, copper and manganese content are also found to be highest in this T5 treatment. Major pest and disease observed is only foot rot, but its incidence was less than 1%. And regarding the highest BC ratio was also reported from this treatment T5. And to conclude that application of the NPK as 25% as PGPR mix and enriched vermicompost and rest 75% of NPK as of the recommended dose of fertilizers as inorganic source, it increased the plant height, girth, yield characters, and quality characters, and it has highest BC ratio, and it also increased in soil microbial content, soil NPK content, and plant NPK content. So the final conclusion of this work is that application of the NPK 25% as PGPR mix 1 enriched vermicompost and rest 75% NPK as inorganic fertilizers was found to increase the growth, yield, and quality of papaya, and it is also economically viable under Kerala conditions. Thank you.
Yes, Dr. Bindu, teacher, thank you for sharing your details of your research work in this conference. It's very interesting. Integrated nutrient management, very interesting. You can exploit waste also, isn't it? Waste can be considered as a wealth.
Okay,
so,
sir.
And one concern, teacher, when you supplement waste with PGPR, will it be economic? So plant growth regulating substances, when you add to a waste, will that be economic?
Okay, sir. Certainly, I will try.
We would like to get your comments on that. Like when you add PG. Okay. Anyway, very good. Kirti teacher, please.
Okay, teacher, good presentation. Application level work. Okay. What about any pest? Any threat of pest or anything you have studied during this year work?
One thing, the major disease incidents that have noted in our fruit is fruit rot incidents. Especially this fruit rot incidents was more prevalent during the rainy season, and its incidence was only less than 1%. We can able to manage it effectively.
Okay, in that condition also this formula is more
yes ma'am. Yes ma'am.
Okay, then another thing, when you use this formula, what about the soil fertility after this harvesting? Any change in soil texture?
Yes, madam. PGP plant growth promoting rhizobacteria mix 1 is a NPK biofertilizer. Usually we have biofertilizer having fixing single nutrients, either they fix nitrogen or phosphorus or potassium. It is only one biofertilizer through which we can fix both nitrogen, phosphorus, and potassium. So, and it consists of Acetobacter, Azospirillum, P solubilizers, and K solubilizers. And it is certainly, and in our study, it enhances the soil NPK content and this NPK content of the respective elements in the plant tissues also.
pH of the soil?
Yes.
What about the pH of the soil? Any change?
pH of the soil enhanced, and it comes most nearly to the up to it enhanced up at the time of the starting of work. It's nearly 5.67, and it enhanced to about nearly to 6.2 like that. Soil pH also enhanced, and the soil microbial content increased, and soil organic matter content also enhanced. The soil organic carbon content also enhanced.
Okay. Another question I have already asked Jayachandran sir. That is, what is the this economics of this formula, this fertilizer when we applied?
Yes, yeah, it has a
economically feasible.
Yes, it's feasible, and we have calculated the benefit ratio, and it is comes to 1.9%, madam.
Okay, okay, teacher, good. Okay.
Ma'am, I have a doubt.
Yes.
Ma'am, did you have any treatment with PGPR mix alone?
No. We have. I am used as in integrated management. We are trying to use maximum organic sources. So I applied organic manures along with these biofertilizers. That's in maximum way we are increasing the soil organic content and nitrogen content. So we have I have used the base as the enriched compost as the base. That is vermicompost. So I'm enriching vermicompost with different sources. One among that is taken as neem cake. Other is a poultry manure. One is azospirillum, and that as PGPR mix 1.
Okay, ma'am. Any more questions? If there are no questions, then we'll move on to the next participant. Thank you, Dr. Bindu B.
Thank you.
Okay, the next participant and the final one, OPFCA 7, Dr. Deepti Madai. Dr. Deepti?
Yes, can you hear me?
Yes, you are audible.
Okay.
Are you sharing the screen, ma'am?
Yes, I'm sharing the screen. I've shared the screen. Can you visualize it?
It's loading, and yes, it has come.
Okay.
Yes, it has come. Ma'am, you can start now.
All right. Respected chairpersons, delegates, and my dear participants, this is a part of my PhD work, and currently I'm working as a guest faculty at the Department of Biotechnology, University of Calicut. The topic is microscopic and fluorescent spectroscopic analysis of a lectin purified from Leukemia leukocephala lambda-wheat. So what are lectins? So the outline of the presentation would be as to what are lectins, planned lectin classification, the workflow, the objectives of the study, lectin studies on this particular plant Leukemia leukocephala, major findings of the study, conclusion, and future perspectives. So what are lectins? Lectins were first discovered in 1888 by Stillmark when he observed that the castor bean seeds were found to agglutinate red blood cells. Subsequently, many plants were found to contain these agglutinates. So these were named lectins by later on due to the property of the ability or due to the ability to distinguish between the sugars present on the human ABO blood groups. And they have been found ubiquitously in plants, animals, bacteria, fungi, algae, viruses, protozoa, insects, and humans. Lectins possess at least one non-catalytic domain which can bind reversibly with free sugars or sugar residues or polysaccharides, glycoproteins, or glycolipids. So Vandam et al. Classified lectins based on two important categories, based on the carbohydrate binding domain and based on the sequence similarity. So based on the carbohydrate binding domain, lectins have been classified into merolectins, hololectins, chimerolectins, and superlectins. And based on the sequence similarity of lectins, they have been classified into legume lectins, chitin binding lectins, type 2 RIP, and related lectins, monocot mannose binding lectins, jacqueline-related lectins, amaranthine-lectin family, and cucurbitaceae phloem lectins. So the objectives of my study were to screen the plants for the lectins, isolate and purify the lectin from the selected plant, and to perform the biophysical and biochemical characterization of the lectin. So the plant used for the study was Leucaena leucocephala. It belongs to the family Fabaceae. So it's commonly called as subabul or ipil in Malayalam. It is cultivated mainly for fodder and green manure, and due to its high protein content, it has been used for phytoremediation. The seed extracts have been found to exhibit anti-helminthic, anti-diabetic, and possess antibacterial activity. So the workflow employed for the study was the seeds were used from which the crude extract was prepared and the ammonium sulfate precipitation was mainly carried out. The various fractions were then purified using the ion exchange chromatography, and the eluted fractions were assayed for the lectin activity, and the active fractions were pooled, concentrated, and then subjected to a second round of purification using size exclusion chromatography, after which the purified lectin was then analyzed for biochemical, biophysical, and biological applications. So the buffer used for the preparation of the crude extract was 50 millimolar tris HCl containing 0.15 molar NaCl pH 7.4. So this is the result obtained after the ammonium sulfate fractionation. And here you can see that the 60 to 80% ammonium sulfate precipitate fraction possessed the maximum specific activity, and therefore it was used for the first round of purification using the ion exchange chromatography by employing DEAE cellulose packed into a column. And the buffer used was a tris buffer, pH 8.4, and increasing and a continuous gradient of sodium chloride ranging from 0.1 to 1 molar NaCl was mainly used. Flow rate was maintained at 0.69 ml per minute. And two peaks were mainly observed, peak 1 and peak 2, of which peak 1 possessed maximum activity, these fractions were then pooled and concentrated and then subjected to SIAS page and specific activity determination by hemagglutination assay. So from the STS page, a number of bands were seen. So it was so we needed a second round of purification to purify the lectin further. So the active fractions were then concentrated and then purified using a size exclusion chromatography using 50 millimolar tris buffer pH 8.4, and of which 3 major peaks were obtained. So the of which peak 3, as we can see here, peak 1, peak 2, and peak 3. Peak 3 possessed the lectin activity. So the fractions were then eluted, and then it was confirmed for the lectin activity by the hemagglutination assay. This is the protein purification profile of the lectin from the Leucaena leucocephala. So here we can see that the concentration of the lectin obtained was around 0.84 milligram per ml with a high specific activity and a high protein purification fold. So then the STS page analysis was then performed. Reducing, non-reducing STS page and native page was mainly performed. 3 prominent bands were seen corresponding to 37 kilodalton, 27 kilodalton, and 20 kilodalton respectively. So under a native condition, a diffused band was mainly seen. So these one of the properties of the lectins is to undergo this glycoprotein staining. So PAS staining was mainly used to visualize the lectins. So under the PAS staining, you can see that the lane 2 shows the presence of 3 pinkish bands, whereas lane 3 and lane 4 are used as a positive controls, which are ovalbumin and IgM respectively. Lane 5 is the BSA, which is used as a negative control. Lane 1 corresponds to the crude extract. So the active fraction or the purified fraction obtained after size exclusion chromatography was then subjected to HPLC analysis using C18 column using 50:50 volume by volume acetonitrile water for 10 minutes, and the proteins were detected by monitoring the absorbance at 280 nanometer. So 3 peaks were seen in the chromatogram with a retention time of 2.8, 3.3, and 3.8 respectively. So this indicates the possibility of this the lectins or the 3 lectins to be isolectins. So then we were interested to visualize the agglutinating ability of the lectin against the erythrocytes. So for that purpose, we prepared three different concentrations of the lectin, 10 microgram per ml, 100 microgram per ml, and 0.5 milligram per ml. And we visualized it, mixed it with the 2% rabbit erythrocyte suspension and observed it under a glass slide. Under a phase contrast microscope, and we observed that a prominent agglutination was seen in the case of 0.5 milligram per ml as indicated in the figure, and relatively low agglutination was seen in the case of 10 microgram per ml. Then to determine the sugar as to which is specific to this particular lectin, we used a different variety of monosaccharides, of which glucose and mannose was found to be the most specific sugar, and of which glucose was selected for further studies. And both the sugars exhibited a minimum inhibitory concentration of 0.024 millimolar. Then, to study the association studies between the sugar and the lectin, the fluorescence spectroscopic analysis was mainly done. First, the lectin was scanned in the wavelength range of 250 to 700 nanometer, and an absorbance maximum was obtained at 280 nanometer. Hence, the excitation wavelength of 280 nanometer was selected for the fluorescence studies. So this particular lectin was scanned for fluorescence emission in the excitation wavelength of 200 to 700 nanometer. So an excitation wavelength of 280 nanometer was selected, and the lectin was titrated with D-glucose as the final sugar concentration ranging from 0.4 millimolar to 4.8 millimolar. And each time after adding the sugar, the solution was mixed and the fluorescence was recorded using a fluorescence spectrophotometer with a 10 millimeter path length. And the spectra was constructed by plotting the wavelength versus the intensity. So here we can see that the fluorescence emission of the lectin was found to be quenched regularly. That is, a decrease in the intensity of the fluorescence was mainly seen as the concentration of the glucose increased up to a concentration of 4.8 millimolar. Then we were interested to calculate the association constant. So we determined the association constant by plotting the scattered plot, for which the association constant or the Ka value was found. The binding constant was found to be 0.159 into 10 raised to 3 molar inverse. That to determine the bimolecular rate constant and the binding stoichiometry, we determined the Stern-Wellmer plot for the steady state fluorescence emissions intensity experiments. And the bimolecular rate constant was found to be 3.55 into 10 raised to 11. Yes.
Ma'am, you have 1 more minute to go.
Okay, thank you. So finally, the 3D fluorescence study further validated our studies, and it proved that at 280 nanometer, the maximum fluorescence excitation was mainly C. So to summarize my work, we obtained a rabbit blood group specific lectin with a high purification fold, and the electrophoretic analysis shows 3 bands corresponding to 37, 27, and 20 kilodalton, and only D-glucose and D-mannose exhibited a sugar inhibition at a concentration of 0.024 nanometer. So this main application of this lectin could be to, we could use this particular lectin to study the to investigate the cell surface glycosylation changes associated with certain diseases, especially cancer. And hence this could play a very important role in studying various diseases, as well as it could be used in cancer cell glycosylation studies. Finally, I would like to thank the University Research Fellowship and Department of Biotechnology and other sister departments of the university for providing the help and the research facilities for this work to be conducted. Thank you for your attention.
Thank you, Dr. Deepthi. Dr. Elias sir was here quite a number of years back. Actually, he joined here initially, and from here he went to CUSAT, and from CUSAT he reached Calicut University. So convey our regards to Elias sir. Anyway, good presentation, good things. Lectin is very important topic. So it can also be from various plants also, isn't it? You can get lectins from, okay. So how can you compare these two, getting lectins from plants, characterizing them, and from in this case, which is challenging, more challenging?
Actually, screening for lectins is quite simple. We have a simple assay, hemagglutination assay, but purification of lectins is very difficult depending upon the plant part. And also the seasonal changes also affect. So sometimes when you screen for lectins from the leaves during particular months of the year, the activity may be high, but at other times it may be very difficult to purify, as well as there may be certain other polyphenols compounds which may interfere with the lectin purification process.
Ma'am, any more questions? Kirti ma'am?
Yes, yes. Okay, Dr. Deepti, good presentation and good piece of work. Okay, what is the fault of purification of your the enzyme after purification?
Okay, I'll get back to my table. The protein purification fold was 5.6 into 10 raised to 4.
Okay. Then this lectin after your purification and SCH page analysis, you found that it is an isomer, isn't it?
Yeah, it was isolectins actually, because we were not able to separate it further. Even under HPLC analysis, we found the 3 peaks corresponding to these 3 lectins. So we concluded that they may be isolectins. A number of isolectins have been studied from the Fabaceae family, and so we have concluded that these might be isolectins.
Okay, will you try for gel elution method to purify this?
You mean, ma'am, cutting out the bands from the
Yes.
No. I haven't tried that, but I have tried a Western blot by the erythrocyte overlay assay, wherein we have transferred this particular gel to a nitrocellulose membrane, and then we have overlaid it with the blood erythrocyte, and we found prominent agglutination in this particular region corresponding to these bands. I have not included that particular data in this particular presentation, ma'am.
Okay, Dr. Deepti. Okay.
Okay, I think there are no more questions in the chat box. So thank you, Dr. Deepti Madai. I think now we can wind up our first session. Can take a 10-minute lunch break. Chairpersons can send their marks to our conference email ID Bio-Inventiyon2020@gmail.com. Okay, thank you. We'll meet exactly at 1:45. Okay, we'll meet exactly at 1:50.
Hello. Hello. Shabana ma'am, are you there?
Yes, ma'am. Yes, ma'am. Yes, ma'am. Tell me.
Shall we start the second session, this second technical session?
Yeah, sure, sure. And before that, make sure that everyone is there and the judges are ready, chairpersons are ready.
Judges are ready,
Jaykumar
Shabana.
sir is there.
Okay, sir. Okay, then we can start. Please call the first participant.
But what about Linu ma'am?
Linu ma'am ഉണ്ട് ഷെറിൻ join ചെയ്തിട്ടുണ്ടോ?
Also.
Okay, fine.
You can start, Sherin. I am here.
Okay, ma'am. Thank you.
Okay, a very good afternoon, everyone. I hope all the participants belonging to the researcher category have joined this session. I would like to welcome you all on behalf of the organizers of Bio-Inventiyon 2020. Now, we will be commencing with the second technical session of presentations, which will be presented by our delegates belonging to the researcher category. This session will be chaired in parts by our eminent chairpersons. Session 2A will be chaired by Dr. Jayakumar Nair, Head of the Department of Biotechnology, University of Kerala, and Dr. Linu Matthew, Associate Professor, School of Biosciences, MG University. The session 2B will be chaired by Dr. Annie Y, Assistant Professor, School of Biosciences, MG University, and Dr. Annie Mole from St. Xavier's College, Alava. As we are all aware that this is an online platform where we might face certain technical issues like network problems. There are even a few problems from the delegates that they can't share their screen. So I kindly request all the participants presenting in this session to be highly alert and be ready with your presentations so that we don't lag behind. During the contest for oral presentations, 10 minutes will be allotted to the participants to present their work. A warning reminder will be given to the participants from the host side at the 9th minute. Those who are crossing the time limit will be disqualified from the contest. The chairpersons are requested to complete the question and answer session within 5 minutes due to the greater number of participants. However, the chair has a right to ask questions to evaluate the candidate to the precise level. The participants are provided with a presentation code as an identification code. I kindly request the chairpersons to please note down these presentation code as the identity of the participant. Before we start the session, the chairpersons, we have a slight change in the time slot given. In session 2A, OPRC 14 will be presenting first. Chairpersons, please note the change. OPRC 14 will be presenting first in session 2A. And
Yes,
the rest will be followed by OPRC 1 to OPRC 6 in session 2A.
I have a doubt. Actually, I have given a list with from OPRC 1 to OPRC 9. So instead of 9, 14 will be presenting. Am I right?
Yes, ma'am. Yes, ma'am.
No, yes, ma'am.
Instead of OPRC 9, 14 will be presenting, and the presentation will start with OPRC 14.
Yes, ma'am.
Yes, ma'am. For your session, the session 2A will be starting with OPRC 14.
Okay, thank you.
Okay, and the rest, OPRC 1 to 6 will be continued after OPRC 14. With that, our session 2A will be completed. Then session 2B will be started with the rest of the slot.
See, one doubt for me. I have a
Yes,
list
sir.
of 7, OPRC 1 to 7. And it's
Yes,
given
sir.
session A, second session A. Is it the
Yes,
correct
sir.
one?
Yeah. So, sir, in that list, we'll be taking OPRC 7 to the second session.
Okay.
That is 2B. And in that, in the place of OPRC 7, we are carrying on OPRC 14 code presentation. Sir, is that fine?
Yeah. So total.
Sir, is it clear, sir?
There are total 7 presentations, isn't it?
Yes, sir. 7
Yes, sir.
presentation
Yes, sir.
in one
Yes.
section.
Okay, we start.
Sir, okay, sir. So I kindly request you, sir, to please take over the session.
Okay. So thank you for giving me this chance of chairing this session. So we'll start with the first one. All the regulations and other things has been described earlier. So there are 7 presentations for this contest, and let us start with the 14th one, isn't it? For the first
Yes,
one.
sir.
Yeah.
Yes.
So I request that person to present her work.
Yeah. Okay. Am I, sir? Good afternoon, sir. Sir, am I audible?
Yeah.
Yeah. Thank you, sir.
Yes, sir, you're audible. Sir, can you please share the screen?
Yeah, ma'am.
Yeah, just change it to the slideshow mode.
Ma'am, is it visible now?
Yes,
Yeah,
it's visible. Please change it to a slideshow mode.
Yeah, ma'am. I have changed it.
Okay, okay,
It
fine,
is in
fine.
slideshow mode.
Okay.
Ma'am, shall I start?
Yeah.
Yeah, you can
Yeah.
start.
Yeah, a very good afternoon to all of you. This is Jahangir Mohammed, a research scholar from Madhyanchal Professional University, and my topic is impact of pollutants on physico-chemical characteristics of the Manasbal Lake under temperate conditions of the Kashmir Valley. Introduction. A lake is a reflection of its watershed. As watershed landscape, the topography, soil, geology, and vegetation, and determine the kind of materials that are entering into the lake, and in turn reflects its water quality. Nutrient enrichment of lakes is considered to be one of the major environmental problems in many countries. That is the aging of the lakes, or what we call it, in other words, the eutrophication. Though the eutrophication stimulates the growth of higher plants, nutrient enrichment ultimately leads to deterioration of the water quality and hence entire ecosystem. In recent decades, population growth, agricultural practices, and sewage runoff from urban areas have increased nutrient inputs many folds to the level of their natural occurrence, resulting in accelerated eutrophication. Study area. Manasbal Lake, with this latitude and the longitude, is considered to be the deepest lake of the Kashmir Valley, is located in the district Ganderbal, about 25 kilometers northwest of Srinagar city in the Union Territory of the Jammu and Kashmir. The lake catchment covers an area of about 22 square kilometers, and it receives water from the catchment and the springs that are occurring in the basin. And it's believed that there are near about 1,200 natural springs within the basin. The lake is surrounded The lake is surrounded by Baladur Mountains on the east by an elevated plateau known as Kareva, comprising lacustrine, fluviatile, and losic deposits on the north and bounded by Artuk Hills in the south, which are used for limestone extraction. This semi-drainage water body serves as an important source of water, but also offers facilities for navigation and transportation, fisheries, harvesting of economically useful plants, sightseeing, tourism, and recreation. This is the general map of the Manasbal Lake, showing the roads, the villages, the catchment area. The bluish is the Manasbal Lake, and this is the entire catchment boundary of the Manasbal Lake. Now, the objectives of the study. The very first objective is the evaluation of the water quality status of the Manasbal Lake. And second is to identify the factors responsible for variation in water quality parameters of the lake. And next is to evaluate overall pollution stats of the lake. And next, to identify the factors that are responsible for variation in water quality among the different sites. The last one is to suggest some conservation measures or the recommendations for the restoration of the degraded lake. So beside this is the results and the discussion. Beside this, there's the air temperature, water temperature. I haven't shown them here, and we will start from the electrical conductivity. If you see the general, I have taken 7 sites for the study. If we see from the graph that the overall high electrical conductivity and the pH at the site 7 and the site 6 is very much high. This is because of the a large sewage disposal, the solid waste dumping, the municipal solid waste, the sewage that are directly coming from the residential complexes, the residential areas, from the shopping complexes, that all the drains are directly going into the Manasbal Lake. There is not a single sewage treatment plant in any inlet of the lake, because there is a lot of encouragement, illegal encouragement happen, and these two sites are very much polluted. If we see in terms of the sewage disposal, a lot of activities are happening, and that is why the site 6 and the site 7 are very much polluted. When we see the electrical conductivity or the pH of the this Manasbal Lake of the Kashmir Valley. If you see the total alkalinity, if this total alkalinity, the higher values of the total alkalinity may be attributed to the increase in organic decomposition, during which carbon dioxide is liberated. The excessive increase in the concentration of total alkalinity at site 7 may be attributed to sewage and wastewaters from the residential areas. So if you see the transparency graph here at the site 7, the transparency is so much low. So the decrease in the transparency or increase in the algal bloom, that is what is called the eutrophication, is very much high at the site 7. So from this and this very the low transparency is a result for diminishing of the light penetration to the lake. If you see now the total hardness and the calcium hardness, the values of the hardness is crossing 5 milligrams per liter at site 7. And besides this, if you see then that there are the anthropogenic sources, but if you see the entire mountains, the mountains located near the site 7, there's a lot of limestone extraction is happening at site 7, because there are a lot of limestone mountains, and that may be the another reason for the calcium hardness in the Manasbal Lake. Now the chloride is considered to be the one of the factor, a major factor in the eutrophication process. Also, we can say that is the pollution caused by sewage and other waste outlets. And besides that, the upper catchment area, there is a surface runoff that is coming directly from the upper catchment area, and there is no bunding, there is no such treatment which can stop the siltation that is directly happening into the Manasbal Lake, and there is a lot of siltation going on in the Manasbal Lake. Now the dissolved oxygen concentration. If you see the dissolved oxygen concentration at site 7 has fallen below 4 milligrams per liter. So as per the World Health Organization and the Bureau of Indian Standards, that any water body, any lake, any aquatic ecosystem where the dissolved oxygen concentration will fall below 4 milligrams per liter, and it is very difficult for the aquatic life to sustain. So from this parameter, we can say that this lake is in a degrading condition. This lake is in a extinction this situation, because the dissolved oxygen concentration at the site 7, at the site 6 is continuously decreasing. So why it is decreasing? Because there are a lot of factors. The anthropogenic pressures, the siltation is there, the sewage disposal is there, the solvent dumping is there. So this entire area has not been changed into a sitting in marsh, because the dissolved oxygen concentration has crossed the limit what has been given the standard by the BIS, World Health Organization, and it is below than that. So from this parameter, we can say that it is this Manasbal Lake is in a diminishing condition. The other sites are not so much polluted, but these two sites are very much polluted, and if you see that, and slowly it will erode the entire this beautiful lake of the Gandharbal city of the Kashmir Valley. So is the sodium. The sodium concentration was high near Konabal site. The high content of the sodium in the fresh water owing to source to the domestic sewage and effluents from the drainage system. And the sodium, this potassium and the phosphorus. And if you see the phosphorus and the nitrates are considered to be one of the major ingredients as a nutrient elements for the acceleration, what is called in the scientific terminology as a cultural eutrophication. Phosphorus is regarded as a key element in eutrophication process. In case of management, the maximum phosphorus show a gradual increase from site first to site 7, where the concentration ranged from 520 to 543 micrograms. Ammonical nitrogen and the sulfate, and same the graph is having the same trend. The site 7, site 6, same the polluted because of no policy, no planning, no treatment, no sewage treatment plant, nothing is there. So this is one of the ammonical nitrogen higher concentration, sulfate is again the higher concentration. The higher values are due to effluent is containing sulfate that increase the concentration in the lake. Now these are the different peaks. That these the top peaks is the general area of the Manasba Lake. Now you can see below the drain is from residential areas.
Yeah,
Yes, ma'am.
Jahangir.
Yeah, ma'am.
Hello. You have
Ma'am.
1 minute left.
Entering into the water body, these are the residential drains. So this is the lake that is disposed that is choked by different macrophytes, solid waste dumping, encouragement in the form of human habitation, azolla species. So after conducting extensive, we can say that large scale illegal encouragement on the periphery has happened. The sewage, there is not a single sewage treatment plant. So now I can give the recommendation, the preventive measures if we will follow, such as the periodic removal and disposal of weeds from the lake, desilting of the lake, construction of silt traps, sewage treatment plants, constructive wetlands. They can help the catchment area treatment, prevention of pollution from point source, and the public awareness and public participation is important, because recently an NGO is working who have cleaned near the more than 550 natural springs. They were totally extincted. So public participation and the public awareness is important. So conclusion from the present studies.
Jahangir, your time is over.
Yeah, ma'am. Okay, the ecological, the anthropogenic pressure, siltation, the garbage are the factors that are responsible for the deterioration of the water quality. So if you are not taking the immediate steps for the restoration of this Manasba Lake, so very soon this entire ecosystem will finish. Thank you, ma'am. Thank you.
Jaykumaran sir, you can ask questions.
This is open for discussion. Those who want to ask, they can ask questions. So I want to ask one question. You have studied only the chemical composition of the water.
No,
Could
no,
you
ma'am.
study about?
Yeah. No, sir. No, sir. Actually, I just here presented only the physicochemical characteristics. I have my topic is the ecological study of the Manasba Lake, where the study of phytoplankton and study of zooplankton and study of the macrophytes, the different macrophytes that have engulfed the entire Manasba Lake. So that is also my study. But here I have shown the small patch of that my study.
That means the microbial study also is your part, isn't it?
Yes, sir. Yes, sir. Phytoplankton and zooplanktons.
Okay, is there any other question?
Linu ma'am.
Shall I ask?
Yes, ma'am.
Okay. Okay, Jahangir,
Yes,
thank you
ma'am.
for the nice presentation.
Thank you.
And I have a few questions to ask.
Yeah, sure, ma'am.
How did you selected your sites?
Yeah,
How did you select sites for sampling?
Yes, ma'am. Yes, ma'am. And there are different factors. If you see the site, the site that I've selected, taking all the considerations such as the pollution load, the impact of the human activities. Where for example, the site 7 and the site 6, where there is a lot of the.
I'm asking what was the criteria you have used for selecting your sites for sampling? Was it random, or have you gone for specific sites?
No, ma'am. The sites that I've selected are not random. They are particular sites where after every 3 months I am going and taking my samples and taking them to the laboratory. They have already selected sites.
How did you select these sites? Have you used any criteria for selecting the sites? Actually, we will not be going for we will not give a preferential treatment to any site. We will be randomly selecting the sites. That's why I'm asking.
No, no, ma'am. No, 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 ma'am. Why I have selected these sites is because some sites are very much polluted. Some sites have a very much impact from the human activity. Some have not. So that is why I have selected some sites where there is a less human interference, and some sites that are selected where there is a more human interference. Taking those considerations.
Okay, Jehangir.
Yes.
Second question is: was there any seasonal difference in this status of the chemical pollutants?
Yeah, ma'am. Definitely, seasonal differences there definitely, because if you see there in the Kashmir Valley, that during the tourism, when there is a maximum tourism inflow in the Kashmir Valley, during that very period, there is not much wastage released into the Manasbar Lake, and there is not much variation in the lake, but there is a slight seasonal variation definitely there.
And also, what are the feeders of this? What are the feeders of this lake? Is it rain-fed or snow-fed?
No, ma'am. It is neither a rain-fed nor a snow-fed. I said that there are near about 1,200 different natural springs that are located within the basin.
So, okay,
there is only a one
spring-fed.
channel. Yeah. There is only a one canal that is coming outside, but that is mostly fed by the sewage, this water from the nearby residential areas. That is why I said that near about 50 springs in recent days has been cleaned and restored by NGO, which were extinct. And it is believed there are 1,200 different these natural springs. There is no artificial source of the water or natural this fed. It is naturally these springs that are occurred within the basin of the lake.
Okay.
Now I think, is there any question? So thank you for your nice presentation.
Thank you,
about
sir.
the pollution in Kashmir Valley.
Yeah, sir. Thank you so much, sir.
Okay, thank you. Now I think we can go for the second presentation.
Okay, sir. Thank you.
Okay, thank you, Jahangir. We are moving on to the second presentation. Our presentation code is OPRC1. Isolation, characterization, and potential applications of lipase from soil bacteria for removal of lipase stains on cotton fabrics.
Good afternoon, ma'am.
Good afternoon.
Am I audible, ma'am?
Yeah.
Yes, you are audible. Can you please share your screen?
Okay, ma'am. Is it audible, ma'am?
It's audible.
Okay, ma'am. Yeah, very good afternoon to one and all. My work entitled as Isolation, Characterization and Potential Application of Lipase from Soil Bacterium for Removal of Lipid Stain on Cotton Fabric under the guidance of Dr. Lolly Grother, ma'am, Hindustan College of Arts and Science, Coimbatore. And these enzymes are most important products obtained from microbial sources for human need, and it is very important in biotechnological industry. Nowadays, this enzyme grow from 6.32 billion by 2020 due to increases of this enzyme in various industries such as paper, leather, textile, and detergent industries. And other than the enzyme produced from these plants and plants and animal, the microorganism producing enzymes are high yield and ease of genetically manipulated, and due to absence of seasonal fluctuation, it available in large amount. And there are various types of enzymes, and my preferred enzyme is lipases, and these lipases are glycerol ester hydrolases, which catalyze the hydrolysis of triglycerides to free fatty acid and glycerol. And this enzymes produced by microorganism may be by intracellularly or extracellularly, and also it produced by both type of fermentation such as submerged and soil state fermentation. And first the fermentation originally developed by submerged fermentation for production of antibiotics and enzyme, and this bacterial lipase produced by various nutrients and various physicochemical factors such as temperature, pH, carbon, nitrogen, and inorganic salts. And after production of this enzyme, go for various purifications and applied in industries. And the objectives of the study, first isolated the bacterial strain from a soil sample around oil mill and identified this organism by various biochemical tests and confirmed by 16S rRNA sequencing and go for production by submerged fermentation and then optimization by both physical and chemical parameters and purification, characterization of lipase, and then molecular determination by SDS phase and applied in detergent. And materials, first the soil sample were collected from various oil factories such as Pilamade, Salem, Vellore and Chennai, and this isolated sample were isolated by nutrient agar and then identified by various biochemical tests to identify the particular organism, and then screening of this lipase by tributylene agar plate assay method, and after identified whether this organism able to produce this lipase, go for production of this enzyme by using the method of Mubarak Kwamsamy et al. 2011 as submerged fermentation. And after production of this enzyme, assay carried out by spectrophotometric method using para-nitrophenyl palmitate as a substrate, and determined this lipase activity using unit per ml calculations, and the same activity will be carried out for protein estimation by Lowry et al. Method. And after production, the effectiveness of this lipase will be carried out by various physical and chemical parameters. And for this, first the physical parameter, the temperature 20 to 60. By varying this temperature, determining the optimum condition of this lipase, and incubation period from 12 to 16 hours, and pH 4 to 8. This is for physical parameters. And chemical parameters, carbon source, nitrogen source, metal ion, detergent, and oils will be carried out for this lipase optimum activity determination. And after production of this optimization, that is identify the particular parameter and production, we go for purification by ammonium sulfate precipitation by various concentration 30 to 80 and determine the constant value of this concentration, and then dialysis with 10 mm tris HCl buffer at pH 7.5, overnight dialysis precipitation, and after precipitated, the pellet will be carried to DEAE cellulose column chromatography, and then determine the molecular weight of this lipase by using SDS page. And then characterization of this enzyme with a different pH 4 to 8 and temperature 20 to 60 and substrate concentration by maximum reaction velocity, and then the molecular characterization by 16S rRNA staining will be done, and this sequence were deposited in gene bank. And then application, the lipase produced were applied in cotton fabric. For this, first we determine the stability of this lipase in detergent. So we have selected different detergent and adding this detergent of 1 mg with 1 ml of this lipase and incubated for 1 hour at 30 degree. And within this 1 hour, before, that is after 15 minutes of interval, we carry out this, taken this sample and assayed for enzyme activity to determine the stability of this detergent. And after determine the stability, the cotton fabric of 5 into 10 centimeter were soiled with the 0.5 ml of oil with a micro pipette, and the quantity of this oil on this cotton fabric is 100 mg. And after soiled that cotton fabric, go for washing solution For this, there are 4 types of washing solution. That is B determined buffer, and the next one buffer and lipase, and next one is buffer and detergent solution, and next one is buffer, detergent and lipase solution. So these 4 different solution will be prepared for 100 ml volume. And this cotton fabric that is sorted with oil, this will be washed to this particular washing solution. And that is washing solution for, we have to wash this for 20 minutes, and after 20 minutes with 100 rpm in shaker, we have washed this with 100 ml of water for 3 times, and then air dried. And we have determined this oil removal in percentage by using this formula. Determine the before washing and after washing, determine this particular oil removal. And same, we are determining this temperature, varying temperature. That particular temperature is used to remove the oil content by particular time. So by varying the temperature 30 to 60 and with the time period 20 to 50 minutes, we have determined the effective removal of oil at particular time and temperature. And results, under this soil sample, there are 40 different organisms were identified from various area, that is Salem, Coimbatore, Velur and Chennai. These are the various organisms were identified, and out of 48 organisms, only 16 organisms able to produce lipase, that is clear zone identification on tributylene agar plate, which is determined, and this go for further studies. And this result determine the biochemical test results for this 16 organisms. And from the 16 organisms, the 5 isolated organisms such as Bacillus species, Serratia, Enterobacter, and another Serratia species and Stenotrophomonas species were able to produce 11 unit per ml of activity. So the out of 16 organisms, these 5 isolates were selected for further study. And this table
Ma'am,
interpreted
you
the
have 1
lipase.
minute left.
Okay, ma'am. And
You have 1 minute left.
Yes, ma'am. And this 16 organism, the table denoted the result for this 16 organisms. And next, after the production by using various optimization, that is here the glucose shows the maximum activity in carbon source, and peptone identify the maximum activity in nitrogen, and calcium chloride will produce its maximum activity. So all this activity will be carried out and go for response surface methodological chart, and the interaction between this parameter will be interpreted. And also the statistical determination of this 5 different organism also updated. And after identification, go for a purification step with ammonium sulfate. 40% will produce maximum activity, and purification process finally 20% of recovery will updated from this. And SDS, it produces 49 kilodalton, and
Ma'am, your
oil
time is up.
removal. Okay, ma'am. Thank you, ma'am. And from this, aerial and non-detergent such as Tween 80 will produce this maximum activity of oil removal from this cotton fabric. And from this conclusion, this bacterial strain are used in laundry detergent to remove this oil, which is difficult to remove the normal washing. Thank you, ma'am.
Okay,
Sir, do you have
now
any questions?
This also for open for discussion. Those who want to ask questions can ask. Lino, Matthew, want to ask some questions?
Thank you, sir. Joy Ruth, I have some simple questions to ask you. Okay, what is the principle of DA cellulose chromatography?
Okay. Ma'am, this chromatographical method is used to separate the protein from that particular crude.
Okay, what is the principle behind this DEA cellulose chromatography? We have
Chromatographic
applied this
method
method.
mainly use.
No, you
Yeah,
have to
ma'am.
say
Yeah,
the
ma'am.
principle. You have to say the principle. That's all. Okay, what was the grade of
This
DEA cellulose?
chromatograph.
Okay, please.
What, ma'am? Not audible.
Okay, please.
Ma'am?
Please, what is the principle of DEA cellulose chromatography? Are you going to answer that particular question, or shall I move on to next question?
DEA, ma'am.
Okay, DEA cellulose chromatography. What is the principle? You have used that method, no? Right? Am I right?
Yes, ma'am. Yes, ma'am. Yes, ma'am. Ma'am, this
Can I ask?
DEA is used to separate the charge particle, that is diethyl amino ether group. Here the resin is coated with positive charge ions, ma'am.
Okay, what was another question is very simple question only. What is meant by 16S rRNA? What is 16S rRNA?
Yes, ma'am. We have to sequencing that particular
Okay, that is
organism.
okay. That is okay. What is meant by 16S rRNA?
16 is rRNA. Ma'am, you have to sequencing, that is a ribosomal rRNA used to
Are you sequencing the
separated
ribosomal rRNA? Are you sequencing the ribosomal rRNA?
What, ma'am?
Are you sequencing the ribosomal rRNA?
Yeah, ma'am. 16 is ribosomal rRNA, ma'am.
What
Then
is that
one more
RRNA?
question. What is the specific activity of an enzyme? What is the specific activity of an enzyme? How can you define the specific activity?
Specific activity by using the formula, ma'am.
Okay, Joyru, thank you.
Yes, ma'am. Thank you.
I just want to ask one question.
Sir?
Your work, your
Yes, sir.
isolated lipase, whether it has got any other activity other than lipase? Did you check that?
No, sir.
No.
Because I'm concentrated in lipase only, sir.
Yeah. No, usually lipase will have the esterase activity also. Another thing, how this work is different from previous works? What is the novelty in this work?
It is organism, sir, based on the organism. And also the application work, sir.
Applications are already there. It's not the first one. There are a lot of this type of papers published with the same type of things. But how your protein, your enzyme, differ from the previously reported proteins? Is there any novelty in that by molecular weight or by speed of activity, that is the velocity of the reaction, specific activity?
It is a, sir, you're asking the difference from that previous one, isn't it, sir?
Yeah, means the novelty of your work, whether it is higher grade, means it has got some positive aspects than that of the previously reported lipases.
Okay, sir. Sir, here what happened, since I will compared with 5 different.
Okay, now the time is over now. Anyways, okay, you can insert these are some questions that you have to answer when you are even going for PhD Viva. Okay. Okay, thank you for your presentation. Now I think we can move on to the third one.
Yes, sir. So our third presentation will be done by OPRC2, actinomycete application, degradation of cottonseed soap stalks.
Okay, ma'am.
Okay, ma'am.
Hello.
Are you ready?
I am ready for
Excuse
presentation.
can you share your screen?
Miss Joyrut,
Yes, ma'am.
please stop
I
sharing.
can share the screen.
Joyruth, can you please stop sharing the screen?
I unable to share the screen, ma'am. So, okay.
Okay,
Now I
thank
can
you.
share the screen.
Gayatri, you can share your screen.
Oh, visualize my screen.
Yes,
And
it's visible. It's visible now.
okay. Good afternoon, all respected sir and ma'am and dear colleagues. My oral presentation topic is actinomycetes application in degradation of cotton seed soft stocks, and my oral presentation code is OPRC2 and my name is Gayatri Patel. Now, this is the content of my oral presentation. First of all, abstract. Novelty of my research is the first report on the utilization of soft stocks by actinomycetes. Soft stocks were degraded to a non-toxic value-added commercial product, and study was extended microbial strain used for biodegradation of oil spill polluted environment. Now, introduction. Oil refineries generate oily sludge, and the microbial degradation is the foremost mechanism for removal of oil spill from the environment. Now, the substrate, that is a soft stock, is the dark gelatinous 7% oil richer chemical constituent that can be generated from the oil refineries. Now, the screening of novel microorganism and its lipolytic enzymes will open the simple route for a synthesis process to solve the environmental problem. The metabolic activity and lipolytic enzymes will open the simple route for synthesis process to solve the environmental problem. Now, methods. The sample collection. The sample is collected from a cotton seed oil refinery industries at washer discharge end of the pipe in a sterile container. Now, enrichment process of actinomycetes adapted to degrade the soft stocks. So, 1A cultures inoculated into 100 ml synthetic medium containing 1% soft stocks and enriched for 28 days incubation in a 4 subsequent transfer. Enriched sample inoculated onto a synthetic medium containing 1% in a solid as well as in a liquid medium and observed the degradation. In a solid medium, by a plate assay, the culture was inoculated onto a 1% synthetic medium plate at a 37 Celsius incubate for 5 to 8 days and observed the whole formation on a synthetic medium. In liquid medium, the culture was inoculated onto 100 ml of 1% cotton seed soft stock containing liquid synthetic medium and incubate at 37 Celsius for 20 days, and regular interval aliquot was collected and analyzed for lipid extraction, TLC and GCMS analysis. Now, for a lipid extraction, derivatization is performed as a part of GCMS analysis in a lipid residues. So, 250 milligram of soft stock samples can be dispersed, collected and determine the lipid as a fatty acid methyl esters by a transesterification process using acids, 5% HCl. This is the procedure for a lipid extraction. The final residues in a hexane was used in GCMS analysis and TLC analysis. For a TLC analysis, I can use the hexane diethyl ether and formic acid as a solvent system and cupric acid acetate orthophosphoric acid spraying reagent for observing the spots. Now, further conformation test was used for hydrogen utilization. In this screening procedure, redox indicator, that is a 2,6
dichlorophenol indophenol DCPIP can used. In this procedure, 0.2 ml of active 1A culture was inoculated into a test tubes containing 10 ml of synthetic medium broth containing 1% cotton seed soft stocks and 0.16 milligram per ml of DEPCIP redox indicator and incubate at 60 RPM at 37 Celsius till 20 days, and data was collected at regular time interval. During observation, the microbial oxidation of hydrocarbon electrons are transferred to a electron acceptor DCPIP to culture media, and the ability of microorganisms to utilize the hydrocarbon was observed. The color changed from blue oxidized to a colorless reduced state. Okay, the biodegradation activity of microbes observed the changes of DCPIP blue color to a colorless. Now, meanwhile, the measurement of growth was done during the biodegradation process. The growth monitoring in the form of dry biomass. Okay, dry biomass, emulsification activity, surface hydrophobicity, and FESEM, the microscopic observation can be done. For the measurement of a growth in the form of dry weight, we drew the samples and centrifuge the pellet and was in a phosphate buffer. And the cell biomass was dry at a 60 Celsius. The content was a constant weight. Now, in a emulsification activity of cell-free supernatant was done. In this procedure, 1 ml of cell-free supernatant plus 200 microliter of soft stocks were incubated for 2 hours and observed the measurement at 600 nanometer. 1 unit of emulsification activity resulting in a absorbance values of 1 at 600 under the assay condition. Now, cell surface hydrophobicity determination, the culture growth centrifuge and the pellet was washed twice with salt buffer. Absorbance at 600 nanometer, all suspension was absorbed at adjusted to 0.75. 1 ml of aliquots of this suspension and 200 microliters of soft stocks was added. Vertex and phase separation can be achieved after a 1 hour's incubation at 37 Celsius. The aqueous phase was carefully separated and a 600 nanometer reading was recorded, and the synthetic media without the soft stocks was grown on the grown cells used as a negative control, and hydrophobicity was expressed by this formula. This is the formula to find out the hydrophobicity. Now, the microscopic observation, 1A culture was grown on a soft stocks and observed on a FESEM, the active grown 1A strain in a synthetic medium and without samples, without soft stock, that is a control, and with soft stock, that is a test, and the sample was mixed in a glutaraldehyde and dehydrated in a ethanol, and platinum coating was used for this study. This is the result of a plate assay. On the plate assay, 2.5 centimeter halos formation on a 1% soft stock containing synthetic medium. Now, this is the result of lipid extraction. There is a 2 different separate layers was observed in a test and only 1 in a control test tubes. Now, this is the result of TLC, where the samples were separated in a LAN 1 container control soft stock sample supernatant. There is a 1 layer. Then LAN 2 and 3. There is a soft stock
you have 1
samples
minute left.
with. Okay. this is the result of TLC. Now this is the result of this is the result of microorganisms upper layers. GCMS result. This is the compare with a control. In lower there is a control and upper is a enzymes. This is the result of hydrocarbon utilization conformation test. On the 11th day, you can observe the degradation, the color change from blue to colorless. This is the result of actinomycetes. Dry biomass maximum 1.2 can be obtained on the 9th day. This is the result of emulsification activity. On the 7th day, 137 unit per ml emulsification activity can be observed. This is the result of SEM. The cell morphology can be changed when utilized soft stock as a substrate. Now this is the conclusion. It can be useful for to control the oil spill pollution. Thank you, sir.
Okay.
Hello.
Okay, now it's open for discussion.
Okay.
So Linu can ask.
Thank you, sir. So, Gayatri, I have a few questions. Why did you concentrate on actinomycetes?
I can study lipase production from a bacteria and fungi also, but now the highest activity can be formed in the actinomycetes. It is a highly potent strain which I can select it. Other organism I can also select it, but highly potent is actinomycetes 1A.
Okay, another question is, how did you isolate the actinomycetes? What was the procedure used for actinomycete isolation?
There is a, I can select, the procedure is, I can take the cotton seed soft stock samples and it can be enriched in a Bursner husk medium. Then I can isolate from that soft stock material and I can enrich for their degradation. There is I can already done, this is the application portion, so I can include this portion on.
Okay. What is meant by derivatization?
Derivatization means in a GCMS analysis, the lipid containing materials है, तो उनका जो fraction compound selection में selection क्या peak में problem होती है, तो I can without derivation I can perform the GCMS and then I can perform the derivation for a lipid fraction, so I can get the good result in derivation.
I'm asking you why what is the need for derivatization in GCMS?
GCMS can separate the compound and can get the good result. When you
what is the principle of GCMS? What is the principle of GCMS, Gayatri?
Gas to more volatile compounds, samples can be converted into. I know the procedure, but
okay, Gayatri, thank you.
Okay. Any other question?
Okay, sir. So shall we move on to the next presentation? Sir, are you there?
No, you can
You
move
are
on to
muted.
Yeah. Sorry.
So, sir, shall we move on to the next presentation?
The next one.
Okay, our next presentation will be done by OPRC3, micro remediation and utilization of natural technology towards remediation of plastic toxicity.
A very warm good. Am I audible to everyone?
Yes, you're audible.
Yeah, a very warm good afternoon to all the delegates present here today. I am Devashri, a research scholar. The problem to my hypothesis is the plastic pollution and its impact on soil and living organism. So I have done the very basic or preliminary work. The future plans also I have discussed here. So in this slides, I will be discussing about the outlines, introductions, materials, methods, results, conclusions, till now, whatever we have got. Plastics, as we all know, is defined as a polymers which on heating become mobile and cast into molds. So this is very commonly used in our day-to-day life because we are using many products which have been manufactured with the help of plastics, like electrical goods, plastics, furnitures, defense materials, agricultural pipes, packages, sanitary wares, plastics, plastic bags, PVC shoes, etc., etc. So the accumulation of this huge amount of plastic became a global issue as the degradation rate of plastic is very slow and very less. So the commonly plastics which we used in day-to-day life includes packagings, disposal, diaper baking, agricultural films, and fishing nets. So these the plastic materials which are used for making manufacturing these products are basically low density polyethylene, high density polyethylene, polypropylene, polystyrene, PBT, polybutylene tetraphenol. The aims and objective of this study is isolation of microorganisms from plastics dumped in soil, identification of plastic degrading bacteria isolated from plastics, and to study the biodegradative ability of these organisms. Just a minute, Mom. So if we move on to the materials and methods, the first material is a sample collection from already plastic dumped soil, isolation of bacteria from the plastic dumped plastics, and identifications of microorganisms also from the roots of some medicinal plants, morphological characteristics of the isolates, biochemical characteristics, and in vitro microbial degradations of plastics. These are the biochemical tests which we have done on the isolates, that is oxidase test, catalase test, gas production test, and carbohydrate utilization test. So in vitro microbial degradation of plastics, first of all, pre-weighted disc of plastic 1 centimeter diameter prepared from polythene bags were aseptically transferred to the conical flask containing 50 ml of culture broth medium, inoculated with different bacterial species which we have isolated from the plastic dumped soil. Control was maintained with plastic disc in the micro-free medium. Different flasks were maintained for each treatment and left in a shaker. After 1 month of shaking, the plastic materials were recollected from the media, washed thoroughly using distilled water, and then subjected for their final weight. From the data collected, we got the weight loss of the plastics. That is the first result, identifications of bacterial strength, which we obtained from the dumped plastics. These are large round white colonies, small round yellow, small round white, large irregular white. We have done dilution up to 10 to the power minus 4, and we got different varieties of microorganisms in each dilutions. And these are the codes used according to their morphological characteristics. Bacterial count, the total bacterial count we obtain, the CFU, number of CFU, the total bacterial count is basically number of CFU divided by volume plated into dilution factor. So by this formula, we have calculated and we obtained the microorganism as 0.0343 into 10 to the power 4. And these are the gram staining results. The gram staining showed the gram positive as well as gram negative bacteria with rod-shaped and circular-shaped coccus. So the table, we got both gram positive, gram negative, and they were bacillus and coccus rod-shaped as well as circular-shaped. These are the result of the biochemical test which we have done on the isolates, that is catalase test, all are positive. Oxidase test is all showed oxidase test positive, and gas production, fermentation of glucose. gas production under Durham's tube, we have seen only 3 isolates which showed positive, rest are negative. So biochemical tests for catalase oxidase of all strains were found to be positive. Result of carbohydrate test. So we have also done the carbohydrate and motility test, catalase test, mannitol test, positive, and this is a oxidase test, this is a carbohydrate test along with the biochemical test chart. The result of in vitro degradation, initial weight of the plastics which we have taken from the dump soil was 0.60, 0.60, 0.60. We have taken a standard weight of the plastic. Large, round, white, small. These are the isolates which we obtained from our isolation serial dilution method. The final weight, it shows reduction in the final weight. So these are the weight loss weight loss along with the percentage. So the conclusion from this study is that this study basically deals with the isolation, identification, and degradative ability of plastic degrading microorganisms from soil. The isolated microbes were native to the site of polyethylene disposal and showed some degradability in natural conditions. The study mainly come to a conclusion that there are certain microorganisms which can undergo degradation in both natural as well as in vitro conditions. So the microorganism shows the degradative ability both in the natural as well as in vitro conditions. So our future plan of the study is 16S rRNA sequencing of the isolates, enzyme assay of microorganism, and treatment of fresh plastics with isolated microbes and enzymes extracted from the microbes. So thank you so much for this. I would like to express my thanks and gratitude to the organizer and convener of this webinar for giving us an amazing opportunities for presenting our work. And now I am open to questions.
Okay, thank you. Now it's open for discussion.
Yeah.
Your name is Purnima, I think.
Yes, sir.
Your name Purnima.
Devasri.
Yes, Devasri.
Devasri. See, you know, what are these plastics chemically?
So we have taken the plastics which has been already dumped in the soil. The plastics which has been dumped long back. It's polyethylene.
By using the term plastics, what are the chemical? What are the classes of chemicals that is coming? Is it just a polyethylene, or is there any other type of?
Sir, can you please repeat? Your voice is a bit breaking. Can you please repeat, sir?
What I'm asking is, by using the term plastics, so what chemicals we can represent? Whether it is a polyethylene alone or other type of polymers also.
Plastics basically use polymers which can be molded into any kind of shapes. The plastics.
That I know.
Yes.
That I know. But what I'm asking
basically
is,
we are using basically we are focusing on the common plastics. Basically, we are focusing on the basically we are focusing on the common plastics which have been used in our day-to-day life, and obviously like polyethylene, high density, low density.
Suppose, just listen. If you are using a particular microorganism for degrading polyethylene, that may not work for other type of plastics or other type of polymers.
That's what this is a confirmation study that plastic is being degraded by certain bacteria. So specifically, this is in our future plan. Specifically, we will be dealing with a plastic, as I have discussed a future plans here. So treatment of fresh plastic with isolated microbes. Once we are done with the 16S rRNA sequencing, then we will decide the bacterial strain as well as the plastics which we will be taking.
I'm concerned only about the chemical nature of the plastics that you have been using for your studies.
Chemical nature has been aromatic. Aromatic natures plastics are also there in the environment.
Others can ask questions. I'm not taking.
May, sir?
Yeah.
May? Okay. What is the principle behind 16S rRNA sequencing?
16S rRNA. Yes, ma'am. 16S rRNA is a part of 30S prokaryotic ribosome, and it is a very conserved sequence which undergo very slow degradation, which undergo very slow evolution. So it is used for phylogenetic study as it is a very conserved sequence of 30S ribosomal prokaryotic ribosome. So
What is okay?
Yes, ma'am.
When you are saying 16S rRNA sequencing, are you sequencing rRNA or some other genes?
We are mainly focusing on
what is the source of you are taking rRNA or some other source is being taken for sequencing?
Basically we will be, because we have not done this yet, we are planning for doing it. So basically we'll be focusing on rRNA and the nucleic acid.
Isolating RNA and this. Okay, I'm asking you.
Yes, ma'am. Yes. Am I audible?
Yeah, you're audible.
Yes, ma'am. Yes, you were saying something.
It's a question. Not, she's asking what about the.
I think Linumis has lost the connection.
I lost connection. Sorry. I'm asking, will you be isolating rRNA and will you be using this rRNA for sequencing or some other source will be used for sequencing? Is it rRNA directly?
Yes, we will be using basically rRNA only for sequencing. And along with that, we will be focusing on some nucleic acid, and the study of enzyme. This only we will be using nucleic acid, rRNA.
So what are the possible end products of microbial digestion of plastics?
Carbon dioxide, as per the previous review of literature, we have seen that there are certain microorganisms which degrades the plastics and converted it into carbon dioxide and water. So we have not done this yet. So we have only studied the weight loss.
Okay, will this degradation have any effect on global warming?
Degradation is obviously, ma'am, degradation will be having an effect on global warming since it is producing carbon dioxide, but that will be utilized, but some extent of carbon dioxide will be utilized by plant also for the photosynthesis. So I think it will be less harmful compared to the raw plastics or the dumped plastics in the soil and in the environment, which leads to soil pollution.
Thank you, Debasri.
Thank you, ma'am. Thank you,
So
everyone.
any other questions? Okay, if there is no other questions, we can move on to the next one.
Okay, thank you.
Number 4.
Okay, sir. So our next presentation will be my OPRC 4 on-farm production protocol for AMF. Arbuscular mycorrhizal fungi.
Hello.
Yeah, sorry.
Hello.
You can share the.
Yes, ma'am.
Am I audible? My screen is.
Yes, ma'am. Yes, ma'am. Your screen is still loading.
My screen is visible, sir?
Screen, now it's visible.
Yeah, now it's visible.
Now, okay.
Ma'am, can you show it in the slideshow mode? Yeah, okay, fine. Now it's fine.
Now it's in slideshow mode. Okay, now it's okay.
Yeah, now it's fine. You can start. You can start.
Okay, thank you. A very good afternoon. Myself, Dr. Purnima Yadav from Kerala Agriculture University. Basically, I am an agronomist, but presently working as a scientist in the extension of the system, that is Krishi Vigyan Kendras of Kerala Agriculture University. Today, I'm going to present a small work, but I think it is high use among Kollam district. My partner scientists are Dr. Manu, Dr. Lekha, and Dr. Meena Kumari. Ma'am, she's a microbiologist, head of Bellini Microbiology Department, is my project fellow. Before going to the details, I would like to give some introduction about this arbuscular mycorrhizal fungi. My topic is on-farm production protocol for arbuscular mycorrhizal fungi or endomycorrhiza, which refers to the obligate symbiotic association between certain fungi and higher plants, in which They are mutually beneficial to both organisms. These fungi cannot be grown in pure culture but can be grown in association with plant roots only. This is a main constraint in the mass production of AMF because of its obligate nature. This AMF it is very important as far as LISA is concerned, sustainable agriculture, because in here we call this as a water fungi because in agriculture that it mobilizes phosphorus from the soil. As far as our Kollam district phosphorus status is about 45 to 400 kg per hectare. That is very high, very high. So use of this AMF, it can mobilize this phosphorus to plants. And apart from this P, it can also mobilize potassium, secondary nutrients like magnesium, sulfur, and micro. Realizing its importance, we conducted some study in our on-farm testing programs, and we found out that this use of this fungi, it helps the plant to tolerate some biotic stresses, especially disease tolerance. That is in our field situations. So we started its mass production in KVK Kollam. I proudly say that our KVK Kollam, our research station, our station is the first government. It's a multiplication as well as that means it's a production and distribution among the farming community. Meanwhile, this mycorrhizae, the quantity of these crops, it is a very huge quantity we record because it contains substrates. So this process is a very production process. That is, we are using some substrates like perlite, vermiculite, soil, cow dung, etc. We are using. These materials are less available, especially this perlite and vermiculite, and it is also costly also. We started on-farm production of reduce the processing as well as its transportation cost. Here, this is a picture that showing how these mycorrhizae infects the plants. Graphs that is taken from our lab using the mobile phone. So with this background, let me coming to my objective to develop the production protocol of AMF at farm level. The methodology, design it is a support culture experiment, consists of 6 treatments and 3 replications. Mycorrhizal, the culture used, mixed inoculum consisting of Globus fasciculatum and Globus etunicatum from Department of Agriculture Microbiology, College of Agriculture, Vilani. The treatments includes roasted substrates as well as sterilized substrates. Roasted because on-farm production always the general method of on-farm production is use of soil, roasted soil. We are using roasted soil for growing these maize plants, and we are growing these plants. We are applying the mycorrhizal culture. That is a general on-farm. Think about some alternate mediums, that is Kyrpith compost 50%, Kyr 15%, cow dung 10%. Then T2, it is a potting mixture. It is a universal potting mixture that is 1:1:1. Soil, Kyrpith, cow dung in equal proportion, that is volume by volume. And the T3, it is soil. And then the next set of treatments, that is, this is roasted, and here conducted at KVK Kollam, that is from June 2017 to May 2019. That is 3 seasons we conducted this trial, and but June to June Kharif, then Rabi, but the summer 2017-18 it is failed, so we repeated in 19. That is 3 seasons. This is the production technology, just how we mycorrhizal sorghum plants. And here we never gave any fertilizer. The fertilizer we given through foliar, that is 1919, as well as Sampurna KU by KU, then calcium nitrate and borax, because it showed some micronutrient deficiencies. These are the various observations we will discuss in later our experiment. Now coming to the results, All these graphs shows the biometric observations. Here, Karif, Rabi, and T2, T4, and T5, respective the season, these maximum growth in as well as the number of leaves. That is at 90 days even. So all these are statistically significant. All these T1, these are comparable. Comparable results. At the same time, the soil alone, that is in the poor growth, so the height as well as the number of leaves were low. Now coming to the shoot weight. Root weight is one of the very important as far as mycorrhizal production is concerned. You can see that in all the cases, that is in the Karif, shoot weight and root weight, this T3 as well as T6 that recorded the lower value, that is fresh shoot weight as well as fresh root weight. T1, T2, T4, and T5, that is our coating mixture, and the various substrate combination, including our Carpet compost, Kyle fiber, soil as well as counting. It recorded a comparable value, higher as well as a high comparable value. Now coming to the dry root shoot weight, dry root weight, root length, that also was followed. So I think there is no explanation for this slide. The same trend, what I said earlier, that is the root colonization. Here the data given this in value, and this is the transformed value, and here you can see this T1, T2, T4, and T5. That is about the higher value, that is ranges from 85 to 96 percentage, and all these are statistically significant. And this T3 as well as soil alone, roasted as well as sterilized soil, that recorded very low values. And this spore count there in these T1, T2, T4, and T5 treatments. The other one, it was 15 to 25 numbers only here. The 100 numbers we can see, but this data is not statistically analyzed. Conducted this experiment for 3 seasons. We pulled the data and pulled also the same trend was followed.
We have 1 more minute left.
And with this root length, and these are the arbuscules and vesicles photographs, and a study clearly indicated that a significant improvement in plant height, number of leaves, roots, and root length of mycorrhizal sorghum plants grown in the roasted and sterilized substrate combinations, fiber, cow dung, and soil, and the potting mixture at 90 days after sowing. In the main substrate, so we can conclude that these treatments, roasted as well as sterilized substrate combinations, consist carpet compost, that is hill, and cow dung, and potting mixture are the best easily available and action of AMF at farm level. Because we calculated the cost also, because based on that, these two are the potting mixture, but the other one is also somewhat 100 rupees difference. So this is my presentation. Thank you. Hello, ma'am.
So thank you. Now it's open for discussion.
Hello.
Hello.
Sir, am I?
Yes, ma'am.
You're audible, sir.
Yeah.
Yes,
Purnima,
ma'am.
now it's over, isn't it?
Okay. Thank you.
Okay. Now it's open for discussion.
Oh, over, sir. Over.
Linu.
Thank you, sir. Purnima. I have
Yes, ma'am.
very few questions. I have only a few questions to ask. Why
Yeah, ma'am.
use foliar traces when you are testing the efficacy of worm?
Ma'am, can you repeat?
Why are you using foliar fertilizers?
Because the voice
when
is
you test
breaking,
the
ma'am. Can you
efficacy?
repeat?
Why are you using? Am I audible?
We are not, actually, ma'am. Yeah. We were not interested in giving any fertilizers or any maneuvers through this soil because it may sometimes contaminate it. Because of that fear, we are giving all the fertilizers through foliar.
What is the reason? What is the reason? What is the reason for using foliar fertilizers?
Actually, we are not going to we are not going to disturb the soil. Okay, ma'am.
That is the reason.
Hello.
That is the reason. That
Hello,
is the
ma'am?
reason you are using
Yeah,
foliar
because of that,
fertilizers not to disturb the soil.
Yeah, not to disturb because mycorrhizal we are trying the mycorrhiza because it is a soil medium we are going to take for the final one. So giving anything through the soil.
You have used a completely randomized
We covered
design
the
for
soil at the time of application.
Okay, you have used
Completely because we conducted this in the pot culture. Because
okay, what was the statistical
because
we
it is we use
no,
ANOVA,
I
simple
am
ANOVA.
confused about the design. Simple ANOVA. Okay, thank you.
Design completely right. CRD. Because this is a pot culture experiment that is in the controlled conditions. We are doing this. We selected this CRD. Yeah.
Okay,
Yeah,
I was curious about the statistical test you used. Over, sir.
Yeah, it is ANOVA. CRD because pot culture experiment. So that is in the controlled condition. That is we conducted this in the first in the our rain shelter. So it is in a controlled condition. So because of that, we selected this as a pot culture experiment. That is CRD. Okay, ma'am.
Okay. Okay. Thank you, Purnima.
Hello.
Purnima. Now it's over.
Sir, I am not.
Yeah,
It is breaking, sir. Is it over, sir?
Yeah. If it is no other question, so we can finish it now. We can start the next one.
Thank you.
Next one, Meenuthan. Next one, algal diversity.
Sir, our next presentation is OPRC 5, algal diversity in the midstream of the Achingovil River.
Okay.
Meera Krishnan, are you there? Hello, Meera Krishnan. Meera, can you hear me?
Meera, please unmute yourself and present, please, Meera. Yeah, it's right. It's fine now.
Okay.
Please
Good
go on to
afternoon,
the
all.
full screen presentation. Full screen mode, ma'am, please.
Okay.
So start full screen. Oh, okay. Now it's okay.
Meera, you can start. Please start.
Good afternoon, all. My topic is algal diversity in the midstream region of the Achingovil River. Algae are the autotrophic and a key part of the ocean, sea, and freshwater ecosystem. They are the primary producers and forming the first trophic level in the food chain. Algae play important and beneficial role in freshwater environment. They produce oxygen and consume carbon dioxide and act as base for the aquatic food chain.
They remove nutrients and pollutants from the water. Algal flora varies from season to season, and an important feature of freshwater algal flora is its cosmopolitan. Algal diversity study is a very useful tool for water quality assessment and also helps in understanding the aquatic ecosystem. The algal diversity is determined by the level of richness of species and their functional importance of ecosystem. Depending upon the seasons, the algae appeared and disappeared. Present results endorse the biodiversity of micro and macro algae of the midstream region of Achangovil River with relation to seasons such as monsoon and post-monsoon. Methodology. The study area selected for the present investigation was midstream region of the Achangovil River. The midstream of Achangovil River includes Tumbaman, Pandalam and Vettiya. Pandalam is a main base camp of Shabarimala, an important pilgrimage site in India. The samples are collected during the monsoon and post-monsoon seasons. The floating microalgal forms are well collected by plankton net by net number 20 silk floating cloth. Algae leaf attached to other types of substrates such as dead leaf strings and any underwater plants which growing in the water are collected manually. Microalgae and the attached microalgae collected by hand and with a knife, including part or all of the substrate. Collected samples were kept in sample bottles and it was allowed to settle by adding leukocidin for good result. 4% formaldehyde solution was also used for the preservation of algal samples. Microscopic observations were carried out with a Magnus light compound microscope and algae were identified with the help of freshwater algal identification key. The observations of the study revealed a total of 41 different algal forms belonging to the classes such as Chlorophyceae, Basidiophyceae, Cyanophyceae and Euglenoophyceae. 27 algae were recorded in monsoon season and 37 during the post-monsoon season. The member of the Chlorophyceae class dominated with 16 genera, followed by Basidiophyceae with 15 genera. The genera belong to the Cyanophyceae were represented by 7 and Euglenoophyceae by 3 items. These are the results. These algae are from Chlorophyceae class. In monsoon season, there are 9 members are identified and post-monsoon season, 13 members are identified. These are the Basidiophyceae members. In monsoon season, 12 members are identified. In post-monsoon season, 14 members are identified and total 15 species are identified. In Cyanophyceae, a total 17 species are identified. In monsoon, 4 species are get and post-monsoon 70 species are get. In Euglenoophyceae, total 3 species are identified. In monsoon, 2 species and post-monsoon season, 3 species are identified. The algal species showed diversity according to the seasonal variations. In the present report, the algae attained its maximum population during the post-monsoon and in minimum monsoon seasons. The presence of pollution indicating species increased in post-monsoon. During the post-monsoon season, Shabarimala pilgrimage season was started. Pilgrims used the water resources of Achangovil River for various hygienic purposes. The presence of euglena indicated the abundance of decaying organic materials for algal growth. The presence of pollution-tolerant algae like Melocera, Oscillatoria, Pediastrum, and Scindesmus is considered as an indicative of enriched water, thus providing evidences of water pollution. Inularia, Schimbilla, Nischia, Navicula are indicators which act as the source of organic as well as anthropogenic pollution. Conclusion. Investigation on algal diversity of the midstream area of the Achangovil River revealed the existence of 41 algae belong to 4 major algal classes. Among these 4 groups, Chlorophyceae were dominant over all other groups. Euglenoophyceae were the least present. Present study conclude that water of the Achangovil River is polluted due to the contamination, especially during the post-monsoon season. Thank you.
So thank you, Meera Krishnan. Now it's open for discussion. Please ask questions.
Have you done any molecular identification of the microalgae?
Never, ma'am.
Have you done any molecular identification of the microalgae?
Never, ma'am.
Oh.
You haven't done?
No, ma'am.
Okay. And another question is, have you encountered any pathogenic algae?
No, ma'am.
No pathogens also. Okay. No more questions.
Okay. Meera, I want to ask one question. Why there is a difference in the post-monsoon and pre-monsoon sessions in the river with regard to the algal content?
In monsoon season, algal diversity is low. In post-monsoon season, algal diversity is high.
No, is there any similarity? Means the same algae can be seen after the monsoon? So that's what I want to know. So whatever algae that is seen before monsoon is visible after the monsoon also.
Some algae are also present in different seasons, and others
Now,
are not present.
If there is a difference, can you suggest any reason for that?
In monsoon seasons, water turbidity is high, but in post-monsoon seasons, turbidity is low. And post-monsoon season is in midstream area. Shabrimala pilgrim season is started. Pilgrims use Ashangol water for hygienic purposes. Pollution indicator algae are high in post-monsoon seasons.
No, algae, they are not depending on organic content of water, isn't it? They are autotrophic. They can depend on light for their nutrition. So why this difference? That's what I want to difference means in the flowing water as well as stagnant water.
Sir, in monsoon season, water current is high. Light penetration, turbidity is increased, so light penetration is low in monsoon season.
Okay, okay. So that may be. Any other questions? So there is no questions from other audience. If there is no questions from the audience, we can move on to the last one, the sixth one. Thank you, Meera, for your nice presentation.
Okay, so we are finally moving on to the last presentation of this session 2A, that is OPRC6, leaf epidermal studies in selected species of Ipomyelin. OPRC6, are you there? Okay. Linu ma'am and sir, please note OPRC6 is sharing the same screen as with the previous candidate. Her name is Rajalakshmi, OPRC6. Rajalakshmi, are you there? Please unmute your mic. Rajlakshmi, are you there? Can you hear us? Rajlakshmi, please respond. Are you there?
Sharin, I think the problem is she has not downloaded the PPT from mail. That's the problem. If she is not ready, I think we can skip this and allocate her to the next session 2B. If Dr. Smita Vijayan is ready, OPRC 7. If she ready, Smita ma'am, are you ready?
Yes, I am ready.
Okay, ma'am. So, Miss Meera, please stop screen sharing now and get prepared with the PPT, and we will allocate you into the next session. So, I think we can move on with Smita Vijayan. Dr. Smita Vijayan, OPRC 7, I think. Please be ready.
Yes, I am ready.
Ma'am, Shabana ma'am, are you there?
Yes, dear. Yes. Yes. Tell me. Because
Ma'am, we have a change in the chairpersons too for session 2B. I think we need a 5 minutes break. They haven't joined the session yet.
So my
but I
role is now.
think we can include Miss Rajalakshmi to the next session, or otherwise we can wind up now. Hello.
Okay, ma'am. So we can wind up with the session 2A.
Okay, Dr. Smita, please wait for the next section. Sorry for the inconvenience.
Yeah, sure. Thank you.
So this session is over, isn't it? Hello?
Yes, sir. Yes, sir. The discussion
Yes,
is
sir.
over.
Yes, sir. It's finished. Thank you so much, sir.
Okay. Now we have to send the result by mail.
Result to the mail. We will provide you the mail, sir. Bio-Inventiyon 20. I will message you, sir, the mail ID.
Yeah. Okay. Thank you. Thank you for
Thank you so much, sir.
Okay.
Thank you so much, Dr. Jayakumaran, sir, and Dr. Linu Matthew, for your keen attention, graceful
Thank you.
responses.
Thank you, Lidu.
Thank you so much.
Thank you for your nice questions and interactions. Okay.
Thank you, sir. Thank you, ma'am.
Thank you, Shabana. Thank you, Shabana. Thank you, Jisha teacher, for inviting us for this particular session. And it was very enlightening. And thank you, sir, for giving me so much of opportunities for asking questions. Thank you, sir.
Thank you, ma'am.
Miss Rajalakshmi and Miss Meera, if you both of you are here, please contact us in person to discuss the problem. Okay. And I think our next judges are joining. Sharon, please take care. Sharon.
Yes, ma'am. I can see Dr. Annie has already joined the session. Annie Y.
Yes.
Dr. Annie Y.
Quote.
Yes, Dr. Annie is here with us. Let's wait for.
Ma'am, are you there? Annie, ma'am, are you there?
Annie, ma'am, please unmute yourself and respond, please.
Hello.
Hello, ma'am. Are you there?
Thank you,
I didn't hear anything. Can you repeat it?
Okay, ma'am. Did you receive the slot time?
Yeah, it is 3:45. I think I was trying to check whether this works or not.
Okay, ma'am. Ma'am, did you
Your time
receive
is actually
the?
3:45.
Yeah, that's fine, ma'am. So, ma'am, did you receive the list of the participants where you'll be evaluating them?
No, I didn't get that.
Ma'am, please check your WhatsApp, please. I have sent it.
Okay. Can you send it to my mail? Hello, is it difficult for you to send to the mail?
No, ma'am, but please provide your mail ID in the chat box so that I can send.
Chat box.
Ma'am, can you please share your email ID in the chat box?
Yeah, I'm just, yes, I'm typing. Did you get that now?
Okay.
Yes.
Just a minute,
Yes,
I made a
ma'am.
mistake. Just a minute, I made a mistake. The second mail ID is the correct one, that is mgu.ac.in.
Okay, ma'am, we'll send it to you shortly.
Okay, thank you.
Shabana, ma'am, are you there?
Yeah, ma'am. Yeah, Sharon, I'm sending the mail to Annie, ma'am. Annie, ma'am, the PPT, I mean, the slot chart is starting with OPRC 8. There is a slight change in that. Annie, ma'am, are you? Can you hear me?
I'm hearing. I'm hearing. Proceed.
Okay, ma'am. So the chart is for contestants of research category oral presentation. It starts with OPRC 8. But there is a slight change that we are starting with OPRC 7.
Okay. So I should start.
Start with OPRC 7.
7. So should I leave now? So what is
ma'am,
the?
you please stay. We are starting. Please stay.
Okay. Then you start with the 7th number.
Yeah, OPRC 7. Sharon, could you please announce the title and invite the participant to present, please? If the
Okay, ma'am. Ma'am, so the OPRC 7 has a title as bioactive potential of taxol extracted from the endophytic fungi Colletotrichum gliosporites isolated from Vidania somnifera. OPRC 7?
Yes.
Smita ma'am, are you there?
Yes. Hope I am audible.
Yes, ma'am, you are audible.
Can I start sharing my screen?
Yes, ma'am. Yes, ma'am. It's visible now. Can you please change it to the slideshow?
Yes, I'm trying. Visible?
Yes, ma'am, it's visible. Just change it to the slideshow mode.
I think it's in slideshow mode.
Yeah, ma'am.
Is
Now
it
it's
visible?
visible in slideshow, ma'am.
Okay. Good afternoon to the experts in the chair and participants and organizers. I am Dr. Smita Vijayan. I am going to present a preliminary work which is based on bioactive potential of taxol extracted from the endophytic fungi Colletotrichum gliosporides isolated from Vidania somnifera. And taxol
Excuse me, ഒരു മിനിറ്റ് സ്മിത. ആനി സെന്റ് സേവിയേഴ്സ് കോളേജിൽ ആനി എത്തിയോ? Shabana, please.
Ma'am, I think she's not there. I was searching. Just a minute. I will call her personally. Please wait a while. Smita ma'am, please wait a while. Please.
Wait, Smita, please.
Okay.
Enter ചെയ്തോ ആനി? Okay, share.
Ma'am, she's joining now.
Okay, Smita, you can start.
Okay, ma'am. So Taxol, which is a naturally derived anti-cancer drug, and it is derived from the yew tree, which is in the Pacific region, Taxus brevifolia. And the main drawback in this is that we can avail this drug only in nanograms, and the plant is a very slow growing one. The removal of bark and stem for extraction of this taxol may lead the plant to death, and that may lead to extinction. But this drug Taxol, which is very useful in breast, ovarian, lung, and many other types of solid tumor cancers. And it is very expensive because the compound is harvested from a scarce resource. So the present study, which focus on endophytic community of Withania somnifera as an alternative source for Taxol production. And there are certain previous reports available on endophytic Colletotrichum species on Taxol production. Something happened, I can't skip to the next slide. Hello. Hello.
Yes, ma'am, you can continue.
But something happened. I cannot share my screen.
Oh, it was visible up till now.
It's not visible now. It's gone. Sharon, please check.
Shabana ma'am, are you there?
Is it visible?
Yes. What about Smita ma'am? Can you share now? Are you able to share
Yes.
now?
Yes, I hope I can share. Is it visible?
No,
No,
not yet. Not yet.
No. It's not sharing. It's not sharing.
Heavy rain here, that's the problem. Maybe connection problem.
Maybe.
I'm trying it again. Hope it will.
No problem, ma'am. You just try. It's fine now. Please move on to the full screen mode. And
Smita ma'am, you can press it now. It's visible.
Okay. So moving to
Yeah.
the next slide. This is the plant selected here is Withania somnifera, that is ashwagandha. It is a regularly used traditional Ayurvedic medicine with many properties like anti-inflammatory, anti-tumor, anti-stress, antioxidant, immunomodulator, hematopoietic, and also have rejuvenating characteristics. It can also influence endocrine, cardiovascular, pulmonary systems, and it is already known that the plant is nurtured with large population of endophytes. Numerous medicament compounds present in this plant are bring out by the endophytes present in the plant. And here the objectives of the study include isolation, screening, and identification of potent taxol producing endophytic fungi from Withania somnifera, extraction of taxol and studying its bioactive potentials, then moving to cytotoxicity studies. And the methods which we have followed is first is collection and identification of the plant, and the Withania somnifera plant were collected and identified at the regional herbarium at SB College. And isolation of endophytes from the leaf, stem, and roots of Withania somnifera. Then screening for the potential microendophyte using UV spectroscopy and HPLC method. Then identification of the selected isolate by morphological, cultural, and molecular characteristics. Then evaluation of antibacterial, antifungal, antibiofilm, antioxidant, and anti-cancer activities. Then studying the cytotoxicity of the extract. Then this is the plant which we have collected and grown in pots. And from these plants, we have isolated about 52 morphologically different fungal colonies. And these fungal colonies, the photo shows that these are the colonies which is arising from the plant parts such as leaf, stem, and roots. And among from these fungal isolates, we have grown these fungal isolates in 500 ml flask in a potato dextrose broth, and then this is homogenized at 3000 rpm for homogenization, and this is again mixed with equal volume of ethyl acetate and incubated overnight with gentle shaking. This mixture was then separated by centrifugation at 3000 rpm. Organic phase was collected, and then it is subjected to rotary vacuum evaporator, and the dry solid residue was collected, which is dissolved in HPLC grade methanol for the identification of taxol. And next is morphological identification of the potential endophyte which produce taxol. This is the microscopic examination of the fungus showing hyaline hyphae with highly branched septate hyphae, and it is surrounded by micro and macrospores. Then this is the cultural characteristic both in potato dextrose agar and potato dextrose broth. And this is molecular characterization shows about 99% similarity with Coryptotrichum gliosporoides, and we have submitted it to the Gen Bank and we got their session number. And this is the overall process. The isolated fungus is from the stem of the plant, and the dry residue is collected in HPLC methanol and subjected to UV visible spectroscopy, where we got a peak between 235 to 273 nanometer, which is the Taxol specific peaks. And then moving to the HPLC analysis, we got a peak which is exactly at the retention time 4.7 minutes, which is in comparison with standard Taxol, it is showing similarity with the pattern. Then the extract is again carried out with antibacterial assay by well diffusion method, and we have tried it against Klebsiella pneumoniae, Escherichia coli, Staphylococcus aureus, Pseudomonas species, and Salmonella typhi. All these are pathogenic MGCC strains, and it showed characteristic well diffusion pattern, that is zone of inhibition, which is increasing according to the increase in concentration of the Taxol extract. And when it is compared with ciprofloxacin, it shows characteristic increase in the zone of inhibition. And this is anti-biofilm assay, where we can show a characteristic black crystallized colonies which shows biofilm formation, and this biofilm formation is very important in pathogenicity of a microorganism. Here, when the Congo red agar is mixed with Taxol extract, there is no black color colonies, which shows inhibition of biofilm formation by the Taxol extract. This is again to the antifungal assay, where we have used different species of Candida, which is isolated from human. These are human opportunistic pathogens, which we have collected from Lakeshore Hospital, Cochin. And here also it showed dose sensitive increase in antifungal activity against Candida albicans, tropicalis, Candida dupliensis, Candida glabrata, and Candida QC. And this is antioxidant assay of the extract, where we have tried DPPH assay and phosphomolybdenum assay. In both of the assays, it showed characteristic enhanced antioxidant activity against the standard gallic acid. And this is cytotoxicity assay of the extract. We have tried RBC lysis assay and MTT assay on normal fibroblast cell lines, L929 fibroblast cell lines, and where in all these cases, the normal cell lines showed high percentage of viability on treatment with the sample. It shows the safety of the Taxol extract against normal cell lines. This is the anti-cancer study against MDA, MB, or breast cancer cell lines. Here also we can observe the first one, which is controlled. There we can observe a continuous layer of cell culture, but at the first concentration itself, there is distortion of the cell continuous layer, and the cell started dying, and it increases according to increase in concentration. And we have calculated the IC50 value for this extract, which is 0.9851 microgram per ml. And according to the world standard, this shows a high anti-cancer activity against MDA, MB cell lines. And this is the cytotoxicity pattern of Taxol extract on MDA, MB cells. And as a summary, we can say that the endophytic fungi Coryptotrichum gliosporoides shows production of Taxol, but we need further studies to prove the exact production and the purification of the particular compound. And even though it brought the possibility for a cheaper and more generally available product, which can eventually be an alternative for this Taxol, and we can produce it by industrial fermentation. And the amount of production may be very low, but the generation time of fungus and high growth rate of fungus will be worthwhile to continue our investigation in this species. And very few reports are available on screening of Taxol by this Withania somnifera. It is a tropical therapeutic plant, and I think it is pioneer in the field. And the extract showed good antibacterial, antifungal, antioxidant, and anti-cancer activity with safer aspect in the cytotoxicity to normal cell lines. So this we can study it in the future and extend our studies to another cancer cell lines and to in vivo experiments. And thank you. And I like to acknowledge BIC Mahatma Gandhi University for the seed funding.
Okay, thank you, ma'am. The dais is open for discussion. Annie, ma'am, are you there?
Annie?
Annie Mole, ma'am, are you there? Can you please
Yes, she is
unmute
here.
your mic?
Annie ma'am says that she cannot unmute her mic. Ma'am, please unmute. Just try now, please. Ma'am, can you unmute now? Annie ma'am. Hello.
Annie from Bioscience. Annie, you can
Annie
ask.
ma'am. Okay, Annie ma'am is saying that she cannot unmute. I'm just giving the options for.
There is one question in the chat box.
Yeah, I think on behalf of Annie ma'am, Sharon, please ask the question for on behalf of Mrs.
Yes, ma'am. Yes, ma'am. So,
Please ask the
Smita ma'am,
question
we
to
have a question from one of our participants. Are you
Yes.
planning to produce any drug formulation for this since it has lot of anti-properties?
Yeah, sure. We can plan for a production of drug. We can formulate any drugs, but it needs a lot of studies, such as in vivo studies and purification of the product. Everything is needed. Toxicity studies in vivo, that means in rats. Everything is needed for formulating it in an industrial basis or a drug formulation, and we are proceeding with it.
Okay, ma'am.
Okay. Okay. Dr. Smita, you have a question from our judge, Dr. Animal. How long
Yes,
did
ma'am.
Taxol side effect last?
Taxol side effect, that depends on the particular individual. That means how healthy he is. Even though Taxol is very good anti-cancer agent, definitely it has some side effects. In case of certain patients, it shows destruction of normal cell lines. But when we are deriving it from a natural resource, we can diminish that particular type of toxicity to normal cell lines. That's what we are trying here.
Reconnected.
Okay, then, and the question, next question is, how do you optimize it?
Actually, we are trying to optimize it. That needs further studies. The fungal culture should be optimized according to the temperature, pH, and all. Now we are in a preliminary stage, so that we are just trying for optimization of this production. Then only we can move to an industrial production or pilot scale cultivation.
Okay, thank you, Smita ma'am. No more question from the judges side. I think you can stop presentation. Thank you.
Okay, thank you so much.
Okay, thank you, ma'am. We are moving on to the next presentation. OPRC 8, characterization of gametophyte specific expression pattern in MSP1 gene promoter. OPRC 8, are you there?
Yes, yes.
Participants, please mute your mics.
Hello.
Yes, OPRC 8.
Yes. Hello.
Okay, so can you please share your screen? OPRC
I can't
8,
share
are you
my
there?
screen. I'm there, but I can't share my screen now. I don't know what's the problem.
Mr. Ajit, I think, Mr. Ajit, actually
Yes,
the
yes,
screen sharing option is open for everyone.
but where it is showing my
Sir, are you able
browser
to click on the
cannot.
share screen?
Yeah,
Sir,
I
are
have
you
to.
able to?
It's showing that cannot allow permanent access to your screen, like that.
Would you
Send the
like to?
PPT then.
Would you
Okay,
like to present by
Bala ma'am, we can
yourself?
do
We
one
will
thing.
share. Yeah, move on to the next.
Yeah, we can move
Ajit
on to
sir,
the
you
next
do one thing.
presentation.
Okay.
Ajit sir, you may please mail your presentation to Bio-Inventiyon 20, our official mail ID. We will present on behalf of you, and you can present. And meanwhile, we can utilize that time by calling the next candidate, I think. Is it okay, sir?
Sure, no problem. Okay, no problem.
Thank you, sir. Please be ready. Please mail. Sharon, please move on to the next candidate.
Okay, ma'am. Okay, ma'am. So we are calling for the next presentation, OPRC 9, Analysis of Diversity and Metal Tolerance in Riparian Flora from the industrial area of Kanjangod, Palakkad. OPRC 9, are you there?
Hello?
Yeah,
Ma'am?
she's there.
Can you hear me?
Yes, you're audible. Can you please share your screen?
Ma'am, is it visible?
Yes, it's visible. You can present. Put it in presenting mode.
Okay.
Full screen. Okay.
Okay, so I'm here to present the effects of metal contamination on the herbaceous community composition and species diversity on the industrial areas of Kanjikode, Palakkad. Recent years have evinced an unparalleled growth in population growth, which has accelerated the industrialization, and pollution was a result of this accelerated industrialization. There are many types of pollutants which falls under the category organic and inorganic, and heavy metals are those hazardous inorganic pollutants, which on long-term persistence in environment pose serious threat to plant and animal life. These industrial effluents are the major source of heavy metals, and there are certain plant species which are considered suitable for growing in industrially polluted regions. These pollutants or the heavy metals cause generally alters the community structure, and phytosociological studies on the existing vegetation structure and the diversity indices are applied to assess the changes in plant diversity. So this study aims to evaluate the adverse effects of contaminants on natural vegetation by studying the phytosociological aspects and also determine the diversity of riparian plant species at the study site. So the study area is Kanchikode, which is the second largest industrial area in Kerala, comprising of various large and small scale industries. And for the study, the riparian vegetation along Valaya River was considered. This Valaya River flows through Kanchikode and receives high load of pollutants. And these are the sample sites, plot 1 to 10, and the location is given. For the floristic analysis, plants were collected and identified with the aid of taxonomists and using regional floras. The primary data was collected by using quadrant method, and 10 quadrats of 1 meter into 1 meter size, 1 square meter size, which were laid randomly in 10 plots. Frequency, density, abundance, basal area was calculated, and the relative values were also calculated to give the important value index of each species.
Various diversity indices were calculated using PAS software, which includes Shannon-Weiner Diversity Index and Simpson's Dominance Index. And from the plants which we got after the phytosociological studies, Alternanthera tenella was the plant which showed the maximum IVI, and it was used for the further studies. For the analysis of heavy metal accumulation in plant and soil samples, samples were collected from the field and they were pre-treated. After digestion, they were analyzed for the content of heavy metals such as chromium, zinc, lead, manganese, etc. by using inductively coupled plasma atomic emission spectroscopy using the instrument Perkin-Elmer AVO 200. And various factors, biological accumulation factor, biological transfer coefficient, and bioconcentration factor was also analyzed or calculated for the plant. The results show that there are 99 species belonging to 86 genera and 33 families in the plot, which comprises of 76 herbs, and among the herb families, Asteraceae and Poaceae dominated. The most specious family with 11 species under 9 genera was the Poaceae, and which was followed by 3 species belonging to the genus Sida and Alternanthera. Diversity indices showed that the plot reflected higher diversity, that is the Shannon-Weiner diversity index ranged from 2.42 to 3.2, and Simpson's index of dominance from 0.04 to 0.13. The heavy metal content analysis was also done and it's showed in table. This is a complete list of plants which are which was observed in the field in all the 10 plots, and the Alternanthera tenella was present in 9 plots except for the plot 1, and also showed high IVI or important value index, which was followed by dactylon and Eleusin indica in the family Poaceae. This was the graph showing different diversity indices, and we have quantified heavy metals, but the most concentration was seen for the 3 metals: chromium, manganese, and zinc. And these values showed the concentration of these metals in soil, root, and shoot, which comprises the stem and leaves of the plant sample. And here is the transfer coefficients, and we have calculated 3 transfer coefficients, and only the zinc has shown to be accumulated in the shoots, which were promising result. So coming to the conclusions, the industrial effluents have negative repercussions on plant diversity, which with effects observed on both the species richness and diversity indices. The plants that showed higher important value index and Shannon's diversity index can be exploited for the removal of heavy metals from contaminated soils. Further investigation on the accumulation of heavy metals in the different plant organs is recommended to assess the phytoremediation potential of the different species. Furthermore, the plant diversity found in the industrial riparian zone can be considered for selecting plant species for the revegetation of riverbanks, thereby restoring polluted rivers. It's major differences. Thank you.
Thank you. The dais is open for discussion.
Annie, ma'am. We got the question from Dr. Annie.
Okay, ma'am.
Can you hear me?
Yes, ma'am.
So the question from our judge to you is, what is the anatomical changes of heavy metal on plants?
Anatomical
changes of heavy metal in plants.
Ma'am, we have studied the anatomical changes, and it was shown that various heavy metals accumulate inside the cells. So they cause different transformations. Sometimes the accumulation of graphites, crystal oxalates, etc. Then much more studies haven't gone for the anatomical.
Annie ma'am, okay. Hello, Firdaus, can you hear me?
Yes. Yes.
One more question is from the judge side. How plants cope with the heavy metals? How plants cope up with the heavy metals?
Okay. The different plants show different mechanisms to cope up with the heavy metals. Some accumulate them in their roots, that is, they show rhizofiltration. Some accumulate them in the other. Some shows vacuolar compartmentalization or chelation. Some detoxify the metals, or even they show volatilization, phytovolatilization. There are different mechanisms of coping with the heavy metals.
Okay, thank you. I think no more questions from the audience. So we
Okay,
can go on
ma'am,
to the
we'll
next.
move on to the next presentation.
Thank you.
Okay, so I call upon the next
Sherin.
presenter. Yes, ma'am.
കേൾക്കുന്നില്ലേ?
കേൾക്കുന്നുണ്ട് മാം.
അയ്യോ, എനിക്ക് കുറച്ച് no problem. I want to ask, no problem. Next presentation.
Okay.
Okay, fine, ma'am. So our next presenter is OPRC 10, bioactive potential and metabolite richness of a new record, Pseudocercospora haiki on Circuligo orchioides. Ma'am, are you there? OPRC 10?
Yes.
Ma'am, can you please share your screen?
Okay.
Okay. I hope Dr. Shaini and Dr. Annie, our two judges, are ready now.
Can you see?
Ma'am, your screen has not been shared yet.
Share.
The screen is not visible, ma'am.
I'm sharing, but it.
Shabana ma'am, are you there?
Sherin, ask Ajit to present.
I am here.
Let Anila send her PPT to the Bio-Inventiyon.
What happened, Anila ma'am? Could you cannot share?
That I can share.
Now it's
Anila,
shared.
don't worry. Anla, don't worry, you send the PPT to us. We'll do it.
Okay, ma'am.
Okay.
Okay,
Karen,
so
let
yes, ma'am.
Ajit present. Ajit is ready now.
Okay, so is Ajit ready with the presentation? Are you there?
Yes, 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 yes.
Okay, so please share your screen.
Okay, Dr. Shaini NK, Dr. Annie ma'am, both please note the next candidate is OPRC 8.
Is it visible now?
Hello.
OPRC 8.
No, not yet.
Parani. ഞാൻ അല്ലേ
Hello.
Yes. Yes,
Yeah.
sir.
Now that option is not there. I mean, I'm sharing, but it's not visible. I don't know.
Okay, then Ajit sir, can I share for you on behalf of you?
So what about changing the slides and
You may
how
tell
I
the next.
change the
I will
slides?
when you
Okay, okay, fine.
telling change or next, I will do.
Okay,
Okay.
okay, okay, okay. Okay. Hello.
One minute.
Yes,
One minute.
sir.
Hello. Is it okay?
Yes, ma'am. Let me start.
Okay. Okay, sir. You don't worry. We are with you. The
Okay.
session you can start. Don't worry about presentation and all those things. You are there with us.
Okay, okay. Thank you very much. So it's my pleasure to be here. A warm welcome to all. Thank you for giving me
Excuse
the
me,
opportunity to present.
1 minute, sir. Excuse me.
Okay, okay.
Shabana ma'am, can you repeat the code for the contestant?
Yes, ma'am. It is
OPRC 8.
OPRC 8.
Okay. Okay, thank you, thank you, thank you.
Okay. Can I start?
Yes,
Please
sir. Yes,
continue,
sir. You
please
can
continue.
start.
Okay. Warm welcome to all. Thanks for giving me the opportunity to present my work. So in this study, I'm characterizing a gene promoter known as MSP1 from rice. So please think. So before entering the core of my research, let me introduce you the base of my work and rationale and hypothesis of my studies. So what is a promoter? As we know all, the promoters are the structure of DNA itself, okay? Mostly commonly seen in the upstream of a gene, and which have a crucial role in determining the expression pattern of a gene. So apart from some common sequences or motifs you can see in the promoters, there are some elements, regulatory elements, commonly known as cis-regulatory elements, which determine the function or the fate of a particular gene. So, uh, it will help to migrate some transcription factors and thereby initiate the transcription of the gene. So next. So there are different types of promoters you can see in the plants. One among the type are known as is tissue-specific promoters. So this tissue-specific promoters these specific promoters is that it will determine the position of the gene expression. So actually, if you want to determine a position of a gene, the tissue-specific promoters we can use. In a gene's position is determined by the tissue-specific. So usually the activity of those promoters is characterized by the experiments like gas histochemical assays or GFP related assays, that is green fluorescent protein assays. This more. This more. So now I am focusing on gametophyte specific promoters in plants. So as you can see in this table, there are 6 types of gametophyte specific promoters. These gametophyte specific promoters are coming under this tissue specific promoter. Forgot to mention first. Okay. So there are 6 types of gametophyte specific promoters, which is expressing only in pollen, and sporophyte tissues, embryo sac and sporophyte tissues, pollen, embryo sac, sporophyte tissues, and 6th one is a pollen and embryo sac. Even though there are 6, you can see here only 5 of them are reported in angiosperm up to now. So the 6th one, which is exclusively expressing in the pollen grain and embryo sac, is not yet reported in the angiosperm. So I am focusing on the 6th type of promoters. Move on, please. So the importance of this type of promoters, that is, which is exclusively expressing in the pollen grain and embryo sac, is that its expression is in the initial stage of the plant life cycle. That is in its haploid stage. So if we have this type of promoters, we can apply our gene of interest in the initial stage of plant life cycle. So that's the benefit of this type of promoters. So during my review of literature, I didn't find any gene promoters which is expressing exclusively in the gametophyte tissues, that is in pollen grain and embryo sac. But interestingly, I found out that there is one transposon known as tag1 in Arabidopsis, and its transposon, its promoter tag1, is expressing exclusively in pollen grain and embryo sac. Since it's transposon, we can't take that promoter for our analysis or for our future work. So what I did is that I just compared the upstream sequences. I did the gametophyte specific gene promoters of other related species. So interestingly, I found out that there's a gene known as MSP1, which is expressed in megaspore mother cell, pollen mother cells, and that's in rice, which is sharing some common motifs that you can see in the tag one. Hello, is it audible? Hello.
Yes, sir, you are audible. You can continue.
Okay. Okay. So this MSP1 promoter sequences and tag1 promoter sequences sharing some common motifs. Move on, please. Next. Please move on. Next slide. So these all are the motifs that present commonly in the MSP1 gene promoter and tag1 transposon. So next, move on. So briefly, the objectives of my study is to predict this gametophyte specific promoter and to construct a novel plant expression vector and to find the expression patterns of this promoter in the plants like tobacco and rice. Next, move on. So this all of the for the first objective of my study was to construct a new novel plant expression vector. For this, I use these 3 vectors in plasmids. So next. So this is the design of the vector that I used to construct. So this is very important because you can see the test promoter, that is our promoter of interest is distantly separated from the promoter of the hygromycin, that is represented as HYG, that is the selection marker. So the other plant expression vector that is commercially available have this promoter sequences near to this gas is near to the other promoter of this selected marker. So one of my objectives was to construct a vector whose promoters is distantly placed so that the expression of my promoter won't be affected by the other promoter, because there is already a reported that CAM35S-like promoters shows a bidirectional activity and can affect the expression of other promoters near to the promoter of those CAM35-like promoters. Move on, please. So this is the first step included in the construction of vector. For this, I used two vectors known as PIG221 and PCAMBA0390, and I cloned this gus and the polyadenic tail to PCAMBA0390, and then I got a plasmid known as PIG4 named as PIG4. Move on, please. Please. Hello. So I confirmed that clone by restriction digestion, and move on, please. So this is the vector map of the first step that I did in vector construction, that's PH. So in this, you can see the gus gene along with the polyadeniation tail that I constructed. Move on to next. So the next step of construction, so I used a vector known as PRAM1. From that, I took the P-ubiquitin promoter along with hygromycin resistant genes, and I cloned it to PH4 in a such a way that the promoter or the gus of the PH4 is distantly placed to the ubiquitin promoter of hygromycin. That's because I got two options. There's two options. The resultant of this cloning is the two. And I need to properly check the orientation. So as you can see, the orientation which is in the upside that I selected for my construction name for my vector. Next. So that also I confirmed using restriction digestion. Next. Next, please. So this is the novel plant expression vector that I constructed. And the peculiarity of this vector is that it is the promoter of the promoter we can clone into the upstream of this gene is distant from the promoter of the hygromycin plant selection, so that the promoters will not co-act together. This move. This move. So I predicted some promoter regions in MSP1 gene, and for that I used plant promoter databases, and I cloned, I designed primers. You can see in the down the figures of the primers that I designed. MSP1 gene and also region of promoter also visible in this. So different length of promoters I predicted and designed primers. Next move. So these are all the details of the primers that I used for the experiment. Next move. So among that I got the four different lengths of promoters: 982 base pair, 865 base pair, 1008.
So you have 1 more minute left.
1320 base pair. Okay, move on. Please move.
You have one.
Yes, please move on. So after that, I cloned the promoter with 1320 base pair into the P-agit5 vector, and the promoter with the 865 base pair to the P-agit5 vector. Okay, next go on. And I mobilized with two agrobacterium and before using the right carbon technique. Next move on. Please move on. And I transformed it into rice and tobacco. Next move on. And transformed it into tobacco. As you can see the figure of tobacco transformed plant. And left side you can see the rooted plant, and the right side I hardened the plant and now it's in the soil. Okay, next move on. Please. And this is the wild plants. Next move on. Next move on. So on the conclusion, I constructed a new novel vector, and I designed the amplified specific promoters and cloned into the vector and successfully transformed it to the vector. Next move on. Actually, the promoter which is the relevance of the studies I want to tell is that the promoters which is specifically expressing in this gametophytes can be used in CRISPR-Cas9 mediated gene editing. It would avoid the hectic task of screening this homozygous mutant line in model plants like tobacco or rice and all, because it helps in generating the homozygous mutant in its first generation itself. Thank you. Actually, some technical error is there, so I'm sorry for the panel, but I am able to present it properly. That's all in my
Okay, thank you, sir. The dais is open for the discussion. Annie ma'am, are you there?
Annie ma'am, if you cannot unmute, please type. Okay, Shiny ma'am, if you have any question, you can.
Hello.
Shiny ma'am, are you there? Yes, Mr. Ajit, you have a question from Dr. Shiny. If the GUS gene is expressed in the rice plant, is there any side effects?
Side effects to rice plant? No, there is no side effects. In fact, we are not hardening it into, or we are not opening it to the garden. It's just an experimental procedure where to express, to where to understand where the gene is expressing. Just a technique, and we are not fielding it out. It's an expression skills. We have a biosafety methods to following the national. In fact, this gene is not a toxic gene. There is no harmful effect also for rice plant. Not only the rice plant, any other plant. You can see the green spots or blue spots, like the result is a blue spots, which indicates the location of the gene where it is expressing. It's a common technique.
Thank you, Ajit.
Thank
you.
Okay. Thank you, sir.
Annie ma'am?
Ma'am, can we call
Do you
the
have
next
any other
participant?
questions? 1 minute, please. Annie ma'am, do you have any questions? I think she's having some connection problem, that's why.
Oh, okay.
Okay.
Shiny ma'am has no questions.
Yeah, she finished her questions. Okay, then we can move on to the next session. Thank you, Ajit.
you.
Okay, ma'am. So we are moving on to our next presentation. Judges, please note the code number is OPRC6. This participant couldn't share her screen previously, so we are calling her now again. OPRC6, leaf epidermal studies in selected species of Ipomia. Rajlakshmi. Rajlakshmi, are you there?
Yes, ma'am.
Okay, so please share your screen.
Can you see my slides, ma'am?
Not yet.
Slides are visible?
No, ma'am, not yet.
Hello, Ms. Rajalakshmi, we got your presentation. Would you like to present by us?
Excuse me, ma'am.
I think that particular echo and all those things is coming from your side. I think so. Please take care of the audio also.
Rajlakshmi.
Okay, it's visible now. Thank you. Yes, it's visible. You can go on to the full screen mode.
Sorry, ma'am, I can't connect this PPT with the Zoom meet.
The PPT is visible to us. Hello. What happened? Rajalakshmi, can you move the slides? If it's still in the minimized mode, can you move the slides? Hello. Hello. Hello, can you hear me? Rajalakshmi, are you
I
there?
think she's connection.
Hello.
Ma'am, they are meeting.
Could you move the PPT? Can you
Shall
are
I
you able to
send my
move the
PPT to
PPT?
your mail or tablet?
Move the slides.
Shabana, if you have her PPT with you, then it's better you share the screen.
Yeah, I have. Is it okay, Rajalakshmi? I am sharing.
Rajalakshmi, you please stop sharing. Shabana
Okay,
will
ma'am.
share the screen for you. You just have to
Okay, ma'am.
present it. Sharon, please not. Linu Matthias joined as a judge. Okay, please not.
Okay, ma'am. Okay, ma'am. Okay, ma'am. Rajlakshmi, the screen is ready for you. Is it visible to you?
Yes, ma'am. Yes, ma'am.
So please start presenting it. We're running out of time.
Yes, ma'am. Good evening to all. My topic is leaf epidermal studies in selected species of Ipomia. Ma'am, introduction. Okay.
Rajlakshmi, just tell move. Okay, just tell move. No need
Okay,
to.
okay. Ipomia is the largest genus of family Convolvulaceae, which consists of nearly 1,650 species, among which 150 species are reported from India. The genus Ipomia shows considerable variations. Ma'am? Considerable variations in habits such as herbs, shrubs, or twining. Epidermal characters are of great value in identification and classification of disputed taxa. This present study intended to analyze the micromorphological features of 5 selected species of Ipomia, species which shows ecological variations. Micromorphological characters like a qualitative and a quantitative nature of stomata and trichomes were studied. The methods I used for this study for micromorphological analysis, plant accession were collected from different ecological habitats. The collected plant accessions was conserved in the greenhouse of college garden. Plant specimens were identified by using different identification flora and digital keys. For trichomes and stomata analysis, leaf epidermal peels were used. The peels were first washed in distilled water and stained with 1% aqueous safranin. The stained plant specimens were washed in distilled water and mounted in 50% glycerin and sealed with the paraffin wax and observed with the Magnus light compound microscope. Here is the observations. Here I selected 5 species among genus Ipomia: Ipomia carnea, Ipomia quamoclite, Ipomia purpurea, Ipomia aquatica, and Ipomia hedraceae. Among these 5 species, the stomatal type is parasitic, and the stomatal size varies from species to species. Here we can notice that Ipomia aquatica have the smallest stomatal size. Here also I study stomatal index and stomatal frequency of both the abaxial and the adaxial surface.
എഡ്ന ചേച്ചി ജോയിൻ ചെയ്തിട്ടുണ്ട്, ഒന്ന് പറഞ്ഞേക്കാവോ? ഇനിയിപ്പോൾ ഇത് കഴിഞ്ഞിട്ട് അനിലയുടെ ഇത് തൊട്ട് തുടങ്ങും. എഡ്ന ചേച്ചിയോട് പറഞ്ഞോ? ഇല്ലില്ല, മീനു പറ, ഞാനിത് പാക്ക് ചെയ്യാം.
Ma'am, move to the next slide.
Shabana, next slide. Shabana, next slide.
Okay, thank you, ma'am. Here the trichome studies are noted. Also the 5 selected species. The trichomes is eglandular and unbranched for all species. And the trichome length and the trichome breadth are vary from species to species. Next slide. Here I concluded that the stomatal and the trichome characters are varies among the 5 selected species. Stomatal frequency and stomatal index between abaxial and adaxial surface of the leaf shows remarkable variation. Stomatal index and stomatal frequency rate is high at abaxial surface in all the 5 selected species. Size of stomata varies from species to species. Size of trichomes varies from species to species. Thank you.
Thank you, Rajlakshmi. The dice is open for discussion. Linu ma'am, are you there?
Yes, I am here. May I ask questions?
Yes, ma'am. Yes, ma'am.
Okay. Rajalakshmi, what are ecotypes? What are ecotypes?
Ecotypes mean I can now getting ma'am.
Huh?
I can't now. I know ma'am, but I don't know its actual definition. Sorry.
Okay, what is a type species? Type species.
Sorry ma'am.
Okay, another question is there any taxonomical confusion about these species, these selected species? Or are they all well defined morphologically and taxonomically?
No ma'am, there is some issues in, not issues, there is some problem in Hypomia quamoclete. There is some genus problem. Now it is separated from this Hypomia genus. So there is some problem, taxonomical problem in that.
The new genus name?
Quamoclete.
Okay. Have you thought of doing any molecular identification?
Yes, ma'am. This is only my part of my project work. I need to
The PhD work?
Yes, ma'am. PhD work.
What are the other features other than this trichome and trichome number and stomatal morphology? What are the other features that distinguish these species? There may be so many other species.
Yes, ma'am. In my
So many other morphological characters. What are they?
Morphological characters are selected. Then also palynological characters. Then cytological characters also included in my work.
Is there any difference in pollen morphology?
Yes, ma'am. Pollen morphology. These are it's
So
ongoing.
all these species are well distinguished. Okay, well distinguished. There is no taxonomic confusion. Then what is the relevance of the work? Can you substantiate the relevance of the work?
There is a taxonomical difference in. I point out that, ma'am, Hypomia quamoclete has some differences in. It shows in our taxonomic keys. So I need to observe and more works about these differences. More studies are needed.
Okay, thank you, Rajalakshmi.
Thank you, ma'am.
Shiny ma'am, you have any questions?
Are you asking me?
No, ma'am. I'm asking to Dr. Shiny. Okay, ma'am. Annie, ma'am, are you there now? Okay. No question from Annie, ma'am. So I think we can move on to the next candidate.
Shabana, one minute, Shabana. I was
Yes,
late to
ma'am.
join. And what is the number of this candidate? Is it OPRC 6?
Yes, it is OPRC 6.
Thank you.
Okay, thank you, ma'am. Thank you, Rajalakshmi. Now, I think the next candidate is OPRC 11, Reshma TS.
No, Shabana ma'am, it's
No, ma'am.
OPRC 10.
Yeah, but I have to download her PPT and I need some time. So in between, I think we can call Miss Reshma TS, OPRC 11. Please move on, Sharon, please.
Okay, this is Edna, Shabana ma'am. Okay, the next participant is OPRC 11, Reshma TS. Reshma, are you there?
Yes, ma'am, I'm here.
Okay, you are audible. Yes, we can see your screen now.
Okay. Shall I start?
You can put in the slideshow mode. Okay.
Is it okay now?
Okay. Okay, now you can start presenting.
Okay, thank you. Good evening, one and all. I am Reshma T.S., a research scholar, ST College, Alapura, under the guidance of Dr. Sri Dilli. Title of my work is Characterization of a Potential Antagonistic Strain, Pseudomonas aeruginosa, KFC1, against brown spot disease in paddy from post-flooded soil deposits of Kutanad, Kerala. As you all know, the plant growth promoting rhizobacteria is a very well known for the biocontrol agent. Kutanad is a unique agro-ecosystem known as the rice bowl of Kerala. This is the only land in India where the rice cultivation is carried out under below sea level. Here you can see some images. The soil texture of the Kutanad is typically waterlogged, and they made bunds using these soils and wood. Actually, the mode of farming is extremely adventurous. In the from these pictures we can see. Then you can see some images of the Kutanad flood pictures during the Great Flood of during the 2018. These images show the severity of the flood, and it affects the Kutanad region very much, and also the agricultural ecosystem also. So the objective of my present study is the isolation and population rate estimation of rhizobacteria from post-flooded soils of Kutanad region, to screen the rhizobacteria through Lopat test, to analyze the plant growth promoting traits of selected bacteria, to find the molecular identity of the selected organism, and the estimation of the in vitro antibiosis of the selected strain against bipolaris oryzae causing brown spot disease in paddy. This is the map showing the study area. The study area is broadly divided into Upper Kutanad and Lower Kutanad, and total 5 sample locations were selected, and the isolation of the bacteria is done. And here we move on to the methodology. First one is the isolation. Soil samples collection was done after 2 weeks of flood. Isolation of bacteria was done according to the Bergey's Manual of Systemic Bacteriology. The rhizobacterial populations were kept in an incubation period of 24 and 48 hours respectively for the morphological character analysis. The population of rhizobacteria calculated after the serodilution by pour plate method. The fluorescent pseudomonads were selected primarily by the fluorescence production under UV light. The selected colonies are streaked on a KB plates for further purification. Here you can see the difference in the fluorescence under normal light and UV light of the selected bacteria. Then we go for the plant growth promoting traits of the selected organisms: phosphate solubilization test, HCN production, ammonia production, and indole acetic acid production of the organism was done. Then we move on to the siderophore production of the organism. As you all know, siderophores are low molecular weight compound which are ion chelators, which are produced by both plants and microorganisms. And when they produce these siderophores in excess amount when there is limitation in the habitat. The siderophore production is estimated through growing the bacterium in an iron deficient synthetic media. They incubated for 48 hours for 32 degrees centigrade. Then the culture filtrates are then centrifuged at 10,000 rpm for 20 minutes. The supernatants were filtered and measured the absorption at 300 to 600 nanometer in a UV visible spectrophotometer. Then we go for the molecular identification. Molecular identification is done through the 16S rRNA sequencing by using the primer. The sequencing process was done at RGCB Trivandrum. Then the isolation of the fungal pathogen Bipolaris oryzae. The infected leaves are collected from the field itself. Surface sterilized leaves with 2% sodium hypochlorite. Then the leaf cuttings were placed on the PDA plates, subcultured, and will get purified fungal mycelium. Then in vitro antibiosis study. To know the in vitro antibiosis capability of the organism, the dual culture method was adopted. An actively growing mycelial disc of Bipolaris oryzae was placed in the center of the Kingsby media petri plate. Then the Pseudomonas aeruginosa strain streaked 2 centimeters away from the periphery of the plate. It is incubated at 28 degrees centigrade for 72 hours and measured the area of inhibition. The petri plate with mycelial disc only is kept as a control. Then the results and discussion. The population of rhizobacteria is found to be much higher
in lower Kutanad when compared to the upper Kutanad region. These are the images showing the results of the LOPAT test. LOPAT test is a set of 5 biochemical tests which characterizes the Pseudomonas species. First one is the result of the leaf fan formation. Second one is the potato rot test, and third one is the arginine dehydrolyze activity. And the total 96 isolates were selected by LOPAT test. Then the plant growth promoting rice test of analysis shows that the KFS-C1 has a potential strain. Here you can see 4 images here. First one is the result of the HCN production. You can see the filter paper turned into the brown color. And second one is the phosphate solubilization. The KFS strain, which shows the clear halo zone around the spot inoculation. The third one is the ammonia production, and fourth one is the IAA production. This graph showing the siderophore production by the selected organism. Here you can see a clear peak is obtained at 404 nanometer, which presents or indicates it is a good source of siderophore. This is a result of the molecular identification. Molecular identification is done, and the resulting sequences were submitted in GenBank and attained the accession number as MT218376. This is the results of the isolation of the fungal pathogen. Here you can see 2 images. The infected leaf is the first image, and we purified the culture. That is the second image. We can show the purified pure culture of the Bipolaris oryzae here. And the identity of the organism is confirmed by the microscopic analysis of the conidia. Here you can see the microscopic images to the different maturation stage of the conidia of the fungal mycelium. This is the result showing the in vitro antagonism between the KFS-C1 and Bipolaris oryzae. First one is the control plate, and second one is the result of the antagonistic study. Here you can see the mycelial disc was the pathogen is completely suppressed by the actively growing Pseudomonas strain. With the efficient siderophore and the secondary metabolites produced by the Pseudomonas, it can suppress and actively suppress the pathogen. And we go for the summary and conclusion. The present study reveals the population rate of PGPRs, including the fluorescent Pseudomonas, is higher in lower Kutanad region when compared to the upper Kutanad. The acidic soils of Kutanad is favorable for the growth of PGPRs, which can be exploited for various agricultural purposes. The native strains of Pseudomonas attracts much more importance nowadays because it can be an effective alternative for chemical fertilizers. The brown spot in rice is one of the important diseases in rice throughout the world, and it is very common in Kutanad region. The indiscriminate use of chemical pesticide results in residual toxicity, development of chemical resistance, environmental pollution, and different health hazards, which often targets the non-targeted beneficial organisms too. So the present study suggests the Pseudomonas aeruginosa strain KFS-C1 as a strong siderophore producing organism and which can effectively suppress the pathogen. So it can be recommended as an effective biocontrol agent against brown spot in paddy, as well as a plant growth promoter for a sustainable cultural practice in Kutanad region. With this message, I would like to conclude my presentation. A sustainable agriculture is one which depletes neither people nor the land. Thank you.
Is the topic open for discussion now?
Yes, ma'am.
Reshma, thank you for the wonderful presentation. I have a few questions to ask.
Okay,
Why did you select brown spot as your targeted disease?
Yes, ma'am. Actually, we are continuously monitoring the Kutanad region, selected areas of Kutanad after flood. We can identify that after the crop season, Punja, we can a bumper harvest is obtained. The number of there is so many additional advantage because of the flood depositions. There is number of weeds are very less, effect of pathogens are very less, and we have a good healthy agricultural practice is going on for the Punja season. And after that, on last February, we noticed some areas are affected by this particular pathogen. So we isolate it from the different fields which are affected areas, and we try to what is the effectiveness of the selected fungi, selected pseudomonas from the flooded region with now. Actually, the in vitro studies are completed until now.
What was the
yes, we have to move on other studies. Yeah.
What was the earlier name of this one? Earlier name of this fungus? Is it Helminthosporium oryzae? Huh? What was the
Helminthosporium oryzae?
Helminthosporium oryzae. Okay, Helminthosporium oryzae. Why did you selected your sampling after the flood, post-flood situation? Actually, post-flood situation was actually advantage for paddy crop cultivation. I am a paddy cultivator. That's why I'm
yes,
asking.
ma'am. Yes, we have to know the post-flooded population dynamics of the Rhizobacterium in that Kutanad region. So we carried out a study, a comparative study between the two areas with the post-flood deposits. And how can we, if suppose we get a beneficial organism, our plan is to recommend that particular species to the agricultural system, directly to the farmers.
Another question: what are siderophores?
Siderophores are iron chelators. Actually, they are low molecular weight compounds, approximately 500 Dalton weight. They are produced by both microorganisms and plants.
How do they help the plant and microorganisms?
Yes, siderophores help to chelate the iron. First one, if an organism possessing a beneficial organism possessing good siderophore, it can beneficial to the plant in two ways. First one, it can compete or it can attract more or chelate more iron and can be absorbed by the plant. Second one, if a beneficial organism with strong siderophore is there, the deleterious bacteria will deprived of iron and they will depleted off, and the plant become healthy.
Okay, my question is, what is the nature of Kutanad soil? Is it acidic or alkaline?
Yes, acidic, highly acidic soil is there. And what is
What
the
about-
advantage is that after post-flood, yes.
What about iron toxicity?
Post-flood, the acidity, iron
What about
toxicity is high because yes,
So there
iron
is
is
no-
much higher. Normally,
iron is much higher.
yes, ma'am,
Then
iron is much higher. Yeah.
so there is no
Iron is much higher, but we are
okay.
no.
Continue.
Actually, we have been moving. Yes. That's we are work is moving on back. That's the way. How can these organisms can be effectively used in that particular species, particular soil type? We are analyzing the soil different textures and the activity of the organism and the siderophore production. The studies are going on actually.
What is meant by 16S rRNA typing?
16S rRNA typing, we are sequencing based on the 16S rRNA type, different 16S rRNA primers are using for the identification of the organism.
Okay, when you are doing the 16S rRNA typing, are you sequencing rRNA or some other regions of the genome? Are you isolating RNA and you are isolating RNA and using it for
16S rRNA is using for the sequencing process.
You are isolating rRNA and this rRNA is being used for sequencing. Am I correct?
I think so, ma'am. Actually, the sequencing process was done at RTCB Trivandrum.
Okay, how many samples were taken from Upper Kutanad and Lower Kutanad?
Total 96 samples were soil samples much more, 15 sample locations, 3 samples from each location are taken, and 96 isolates was taken for the preliminary, after preliminary selection, 96 isolates were taken. Then the plant growth promoting traits.
How did you demarcate the Kutanad region as Upper Kutanad and Lower Kutanad?
Based on the geographical distribution, we can the upper Kutanad region is comparatively geographically somewhat higher, like Chenganur, that area coming under the upper Haripad, Mavelikara Taluk. Different taluks are there. We collected the map from the in a particular panchayat, and in collaboration with the agriculture officers, and we can select the study area in different locations. That's Upper Kutnad and Lower Kutnad regions.
Okay, thank you, Reshma. Best of luck.
Thank you,
Reshma,
ma'am.
I
Thank you
have
very
some
much.
questions. Reshma,
Yes.
I have
Yes.
some questions. One
Yes,
thing,
ma'am.
have you checked the soil characteristics of the soil?
Yes, ma'am. Yes, yes. I have checked the soil different soil pH, soil NPK. Different parameters are checked but not included in the slide.
Have you checked the metal toxicity?
No, metal toxicity we haven't targeted. Actually, there are some metal toxicity present in the Kutanad. There are some literatures are here. Now, after flood, the content of silicon is high, and the sulfur content also increased after flood.
So, what is the main difference you noticed between Upper Kutanad and Lower Kutanad in the case of soil characteristics?
First one is the pH is the typical difference. In the Lower Kutanad region, much acidic pH is obtained for the soil, and Upper Kutanad region nearly 5 to 6 is the pH level. And the Lower Kutanad region is highly acidic because of the excess. As I mentioned earlier, the sulfur content of the soil is very high, and later after years, in recent times, it may be a leading cause to increase the soil acidity.
You identified the organism as Pseudomonas aeruginosa, isn't it?
Yeah. Yes. Yes.
So, what is the main drawback of this isolate?
Actually, Pseudomonas aeruginosa is sometimes called as a opportunistic pathogen. But we checked the pathogenicity of the organism. But in our lab, we are continuously monitoring or dealing with the Pseudomonas aeruginosa. The rhizobacterium Pseudomonas aeruginosa is most of harmless, and it has too many plant beneficial properties also. So we can.
So, how did you check the pathogenicity? How did you check the pathogenicity?
Pathogenicity, we first potato root test itself represents the it is pathogenic or lethal to the plant. If there is no rot obtained, we can conclude. Then another one more for biochemical tests are there. But we are dealing with Pseudomonas aeruginosa for 2 more years, and our seniors also dealing having the plant growth promoters as Pseudomonas species. But there is no problem with this organism as yet.
Have you checked the human toxicity?
No.
Hemolysis,
No. We don't check that. But I think there is even some people or some allergic problems of skin or respiratory some infections, something like mild symptoms are there. But we don't check the toxicity in humans now.
And you told one statement that acidic soils are better for cultivation. It's better to exploit in sustainable agriculture.
No. Actually,
justify.
No, ma'am. Actually, what I mean that the acidic soils of Kutanad is a rich source of a rich collection. There is a rich collection of pseudomonads are there. Even though the soil is acidic, so many organisms are existing, and so many good beneficial plant growth promoters are there, and the native strains are not exploited as much. When we talk with the agriculture officers and farmers, very feeble farmers are utilizing the pseudomonas for cultivations, or even they are using the samples or the imported samples from Tamil Nadu state and all. But they are not much suitable for the or they can't make any effective result in that particular zone. So if we focus on our native species, and we can commercially multiply the organism, and if we provide a native strain to the farmers, it will be more eco-friendly and will be beneficial for us.
Okay, Reshma, there are questions from the other chairperson also.
Okay.
Okay, she has asked, have you done a study on soil deposit? Because these organisms shows antagonistic activity otherwise too.
Just we analyze the soil characters. As I mentioned earlier, just we analyze the soil characters only.
Okay, the other question is it a new strain which you isolated, KFSC1?
No, actually, this belongs to when we deposited in Gen Bank, it is actually belongs to the Pseudomonas aeruginosa. It is not a new strain. We named it as Kerala flood strain C1. That is KFSC1.
Okay.
Reshma, an advice, you have to check the pathogenicity. Okay.
Okay, will do, ma'am. Definitely. Thank you.
Okay, Reshma, thank you very much. The
Thank
next
you, ma'am.
participant.
Thank you all.
Okay, we'll move on to the next participant. OPRC 10, Anila PS. Anila? Anila, are you there?
Yes, ma'am.
Okay, you're audible.
Yes.
Okay, I think your PPT is shared by Shabana, ma'am.
Okay.
Slideshow, please. Okay, you can start, Anila.
Okay, ma'am. Good afternoon, all. I am Anila PS, research scholar, Department of Botany, Sri Nilakantha Government Science College, Pattambi. I am here to present the work Bioactive Potential and Metabolite Richness of a New Record, Pseudocercospora hackeae on Curculigo orchioides. Endophytes are the symbiotic association of microbes within plants that support the plants to accumulate secondary metabolites. Most of the secondary metabolites are important drugs in the field of medicine as they possess anti-cancer, antiviral, anti-diabetic, and immunosuppressant properties. Though endophytic fungi are rich sources for potent natural antioxidants, these are relatively unexplored area among microbe isolation. Curculigo orchioides garten is a threatened species under the family Hypoxidaceae. It is an effective immunomodulator and aphrodisiac in the Ayurvedic medicinal system. Curculigo orchioides is rich in secondary metabolites like alkaloids, flavonoids, phenols, saponins, and glycosides. Most of the therapeutic effects of the plant are provided by the characteristic phenolic glycoside, curculigoside. Moreover, it is a host for endophytic fungi like Penicillium aspergillus, mucor, etc. Objectives of the study are to isolate endophytic fungi from Curculigo orchioides, to evaluate antibacterial activity of isolated fungi, to identify the most vigorous endophytic fungus among the isolates, to quantify secondary metabolites in the identified endophyte, and to estimate the antioxidant ability of the identified endophyte. Materials and methods. Leaf and tuber of Curculigo orchioides was used as the plant material. PD and PDB were used as media for culture, and chloramphenicol is used as antibiotic. Then different strains of bacteria such as Klebsiella pneumoniae, Staphylococcus aureus, and Pseudomonas aeruginosa. The three reference human pathogens were used for screening of antibacterial activities. Then glasswares, instruments, and chemicals for quantitative estimation were also used for this study. Methods. Isolation of endophytic fungi was done based on the protocol reported by Suryanarayan et al. 2005. Plant materials collected from home sheets of Pattambi and surface sterilized plant part placed on PDA that containing chloramphenicol and incubated at 27 degrees Celsius until growth appeared. Actively growing mycelia. Agar blocks of actively growing mycelia were placed on PDB at room temperature with periodical shaking at 150 RPM for 3 weeks for mass cultivation of the endophyte. The secondary metabolites produced by the endophytes were extracted with ethyl acetate according to the protocol provided by Raviraj et al. 2006. The crude extract was dissolved in 0.1% DMSO for the antibacterial screening. Antibacterial screening was done by disk diffusion assay. 35 microliter of crude extract dissolved DMSO was pipetted into the circular disc and placed on the MH agar plates that contain different bacterial inoculum. 0.1% DMSO was used as the negative control. After 24 hours, zone of inhibition measured and selected one fungus that responded to all the 3 pathogens. Selected endophyte was identified at genomic level. For that, DNA of the endophytic fungus was isolated and amplified its ITS region with a universal ITS primers, ITS1 and ITS4, and sequenced the amplicon and subjected to blast analysis for homology search. Then sequences of the putative fungus and related fungi were multiple aligned using Bio-Edit 7261. The sequence obtained by after sequencing was submitted to the GenBank. Secondary metabolites were quantified by the protocol of Damodar et al. 2011. Total phenol content was quantified by following calcium method, and total phenol was estimated by ALCL3 method. Antioxidant properties were also measured by 3 different methods: total antioxidant activity, reducing power assay, and DPPS radical scavenging assay. These are the reagents and equations used for measuring antioxidant potential of the endophytic fungus. Statistical analysis: all experiments were carried out in triplicates, means plus or minus standard deviation calculated, and ANOVA was done using SPSS program considering P less than 0.05 as significant. Graphs were plotted with Excel 2013. Results: we got a total of 7 endophytic fungi from both leaves and tuber, of which 5 from leaves, CLF1, CLF2, CLF3, CLF4, and CLF5 were subjected to antimicrobial antibacterial screening. Among the 5 endophytes, COLF1, CLF2, and CLF5 inhibited the growth of Klebsiella and Staphylococcus, but COLF4 created zone of inhibition against only Klebsiella, while COLF3 responded to all the 3 pathogens, and hence it was selected for molecular identification. Amplicon of ITS region of CLF3 revealed a nucleotide sequence of GenBank accession number MW024066. This sequence shows 97.58% similarity with the sequence of previously identified fungus Pseudocercospora hackeae, and hence CLF3 was identified as Pseudocercospora hackeae. These are the images of different morphology of different endophytic strains obtained from leaves and tuber. So figure 3 shows the antibacterial activity of COLF3, and then by quantifying secondary metabolites, we got 44.5 plus or minus 0.0085 microgram gallic acid equivalent of total phenol content, and 24.09 plus or minus 0.081 microgram quercetin equivalent of total flavonoid content from the crude extract of Pseudocercospora. These are sufficient to induce biological activities and are relatable to other literature data. From antioxidant assays, we estimated a total antioxidant activity of 0.247 plus or minus 0.113 microgram of ascorbic acid equivalent. There is a report, Sadanta et al. 2014 reported a maximum TAC of 348.54 microgram per mg ascorbic acid equivalent by Fusarium oxysporum from Viscum album. By reducing assay, we estimated ascorbic acid equivalent at 500 microgram per ml sample concentration of 71.05 plus or minus 1.255 microgram per gram dry weight of extract. There is a report of Sharma and Kumar. Aspergillus terras exhibited maximum reducing potential of absorbance 0.34 plus or minus 0.04 among 4 endophytes from Osimum sanctum. By DPPH assay, we calculated IC50 value as 103.632 microgram per ml, and it showed higher scavenging activity of 85.72% at 250 microgram per ml concentration. Our results with various antioxidant assays clearly indicate that the endophytic fungal isolate obtained from the traditionally used ethnomedicinal plant Curculigo orchioides possess different groups of antioxidants that are comparable or even better in terms of their potency with the standard antioxidant, and may be useful as microbial cell factories for the production of antioxidants in the near future. The same strain showed maximum antibacterial activity against Klebsiella pneumoniae, Staphylococcus aureus, and Pseudomonas aeruginosa. conclusion. Here I am conclude that this study gives an insight to the in vitro growth and antibacterial ability of fungal isolates, as well as secondary metabolite production and the antioxidant capacity of most dominant isolates, Pseudocercospora hackeae, from the medicinally important plant Curculigo orchioides. It is a slow-growing fungus. Significant production of phenols and flavonoids were noticed. It exhibited remarkable antibacterial activity, and it is a potent antioxidant producer with broad-spectrum activity against various free radicals. Relevance of the study. As Pseudocercospora hackeae in Curculigo orchioides is a new record, this study will provide an introduction to more comprehensive work on bioactive compounds produced by this endophyte. Pseudocercospora hackeae is a potent treasure trove of novel antimicrobial molecules and antioxidants. These are the references, some of the references. Thank you.
Okay, the session is open for discussion.
Yeah.
Yes, ma'am.
Am I audible?
Yes.
Okay,
Yes,
I have observed that you have used 3 human pathogens in your study. Okay.
Yes. Yes.
Did you get IBSC clearance, Institutional Biosafety Committee clearance, before using human pathogens?
Yes.
Huh?
Yes.
If you have IBSC clearance, you have to give the IBSC number. Okay. Clearance number.
Okay, ma'am.
Is there an IBSC in your center?
Excuse me, ma'am?
Is there IBSC in your center?
No.
How did you get IBSC clearance then?
Our guide has.
Okay, Anila, how did you make sure these isolates are truly endophytes? They may be present epiphytically also. How did you make sure they are truly endophytes?
Plant parts are used after cutting. Then it will come from inside.
Then also there may be epiphytic organism present on the plant surface which may grow.
We sterilize. We placed it on medium after surface sterilization. It will surface sterilization will remove the epiphytes.
Is surface sterilization 10% effective?
Yes, ma'am.
Okay, what is meant by ITS regions?
Internal transcribed tracer region.
Where is it present?
rRNA. Itness rRNA.
Okay, what is the significance of this ITS region?
It is a universal region for fungus.
Only fungus.
Yes.
Used for any other organisms other than
Anila,
fungus.
Anila,
Yes, ma'am.
what's the role of phenols and flavonoids? Hello.
Hello, ma'am.
What's the role of phenols and flavonoids?
Phenols and flavonoids help the plants to resist biotic and abiotic stresses.
How it helps? Whether it interferes with any metabolic pathways?
Ma'am, I can't get.
Hello, whether it interferes with any metabolic pathway or which type of stress it's effective. Hello.
Biotic stress.
Biotics, it's broad range. You specify it. Hello.
Ma'am, എനിക്ക് ക്ലിയർ ആവണില്ല മാം പറയുന്നു മനസ്സിലാവണില്ല.
ബയോട്ടിക് സ്ട്രെസ് ഏത് സ്പെസിഫിക് ചെയ്യാവോ? ഏത് സ്പെസിഫിക് ആയിട്ട് പറയാവോ? ഏത് ടൈപ്പ് ഓഫ് സ്ട്രെസ് ആണ്?
Biotic stress.
Not hearing? It's not audible. I'm not audible?
Yes, ma'am, audible, but I can't get.
Okay, you can leave it. Leave it. Okay, no problem.
Okay, thank you, ma'am.
Lino ma'am, you have any other questions?
Okay, what is the significance of this organism, Pseudocercospora hacke? Is it a plant pathogen?
No, ma'am, it is an endophyte.
It's not a plant pathogen.
No.
No, okay.
Okay, we have questions from the other chairperson.
Okay, ma'am.
One second. Okay. Is Curculigo orchoides available in market as any product?
Yes, it's an aphrodisiac, Kali Musli. Commonly known as Kali Musli.
Okay. Okay, the second question, what is the significance of ITS?
ITS is the universal internal transcribed spacer region of 18S rRNA. It is used for the identification of fungus. Variations in this region, sequence variations, we can identify fungi. Fungal identification and using.
Okay, the third question is, can you name any other region which can be sequenced other than ITS?
No, I don't know.
Okay, any other questions? If not, we'll move on to the next participant. Okay, there are no questions, I suppose. So thank you, Anila. So I call upon the next presenter, OPRC 12, Farah Barami. Farha, are you there?
Yes, I'm here, ma'am.
Okay, you're audible. Can you please share your screen?
Okay, ma'am. Just a second. Is it okay?
It's loading. Wait. Okay, it's visible now. Can you please put it in the slideshow mode? Okay, done. Now you can start.
Okay, thank you, ma'am. A very good evening to one and all. I'm Farah Barami, PhD scholar from Department of Microbiology, Marudu Pandiyar College. Today I'm going to present my topic on endophytic bacteria of Allium sativum as biocontrol agents of black mold disease. So the widespread use of toxic agricultural chemicals has been a huge concern in this modern age, and hence an increased recognition of the biological agents against these pests should be developed and implemented in the fields. So the potential use of microbe-based agents in the control of pests has gained more importance and also has been addressed in many reports. And in our study, we have used the endophytes, which are microorganisms which live in the intracellular spaces of healthy plant tissues, which generally does not cause any ill effects or a disease. Instead, they are responsible for symbiotic, mutualistic, and tropobiotic activities, or give a synergistic relationship with the plants. Now, Allium sativum, which is generally known as the garlic, is a species coming under the Liliaceae family. So it is basically known to have immense amount of active compounds showing antimicrobial, anti-cancer, antioxidant properties. But there are seldom reports on the endophytic microbes of garlic, which is targeting at the plant growth, biocontrol property, and the synthesis of allelochemicals. So the origin of my research problem was the black mold disease, which is caused by Aspergillus niger, which is one of the serious threats to garlic production worldwide. And we are in the quest of finding or developing an environmental friendly or an eco-friendly a biocontrol agent against the black mold disease causing Aspergillus niger. The demand for the alternative control strategies And the use of biopesticides has been increasing steadily due to environmental awareness and potential health hazards from the chemical pesticides. So to investigate the possibilities of using native endophytic microbes of garlic as a biopotential agent of the disease is what we are studying here. So the objectives of the study is, first, we are isolating the endophytic microbes from Allium sativum. We are finding the antagonistic activity of the endophytes against the Aspergillus niger, which is a causative agent of the black mold disease. Partial identification of the endophytic strains by morphological, cultural, and biochemical tests, and the detection of biocontrol mechanisms. Now, coming on to the methodology of study, first we have done the sample collection, the isolation, screening for in vitro antagonism, and selection of the specific or the potent strains of endophytic microbes, which shows biochemical antagonism, and we have also done the biocontrol mechanisms of the selected strains. Firstly, we have done the sample collection. Sample collection was done from Calicut. Initially, the pre-treatment was done. Pre-treatment was done by washing firstly to remove the dust particles and all with clean distilled water, sterile distilled water, and tween 20. After that, we've done the surface sterilization of the garlic cloves. For that, we have used sodium hypochlorite and then 70% of ethanol. It was washed properly again with distilled water, and as the question raised before, to kill the epiphytic organisms, we took 0.1 ml sample, and we've put it into the tryptic soy agar to test whether our garlic is properly sterilized, surface sterilized. And then after washing, it was blot dried and then excised, and then put it into the tryptic soy agar, and it was incubated at room temperature. Then next we have, after isolation, we have done the, we've also isolated the fungal pathogen, which is the Aspergillus niger, from the infected garlic cloves, and we have microscopically also identified. And then we have done the in vitro antagonism test. Here we are using the dual culture test. So we've isolated 14 endophytic bacterial cultures, and then we have checked for the antagonistic activity against the pathogenic fungus Aspergillus niger. So the dual culture test was done by taking the NAPDA media, which is nutrient agar plus the potato dextrose agar to complement the growth of the endophytic bacteria as well as the Aspergillus niger. So for that, we streaked our endophytic bacteria on one side, and on the other side we've put a plug of the Aspergillus niger. And then we checked for the antagonistic activity. For that we have to calculate the percentage of reduction in the growth of Aspergillus niger. So growth in control minus growth in antagonist divided by the growth in control into 100 was done to find the percentage of reduction. And for that, out of the 14 isolates, we found out that 6 isolates of endophytic bacteria showed maximum activity. So among them, ASEB, which is Allium sativum endophytic bacteria 1, 2, 3, 4, 5, and 9 has showed the antagonism. Next, we are moving on to the biochemical mechanisms. For that, siderophore production was done. Siderophore production. For siderophore production, we took 1 ml of the culture medium, the culture broth, and we've inoculated into King's Bee broth, and we've incubated for 3 days at 28 to 30 degrees Celsius. And after 3 days, we've taken the samples and then centrifuged at 10,000 RPM for 10 minutes. And then the supernatant was collected. 2 ml of the supernatant was then added to 2% of ferric chloride, and the appearance of orange to reddish brown coloration had indicated the presence of siderophores. The next we have done is to check the production of volatile compounds. To check the volatile compound production, we've taken 2 sterile Petri plates. To one, we've added nutrient agar, and to the other, we've added potato dextrose agar. So to the nutrient agar plate, we've swabbed the selected selected endophytic bacterial culture, and to the other we have put a plug of a disc of the Aspergillus niger strain. And taking out both of the lid, we just took both the bottom part where we have inoculated the organisms, and then the lids were removed and the bottom plates were then sealed with the parafilm and then incubated for 7 days at room temperature. So the production of volatile compounds was monitored by the absence of sclerotium germination and mycelium expansion. Next, we had done for these are the results which we have got from the volatile compound production, in which ASEB 1, 5 and 9 showed that there was absence of sclerotium germination and the mycelial expansion. And next we had done the volatile compound production. So the non-volatile compound production, we used 1 ml of the standard inoculum, which was added to 5 ml of tryptic soy broth, and it was kept for 7 days, centrifuged at 10,000 RPM for 10 minutes. And the supernatant was collected, and 100 ml of the culture filtrate was added to the wells of nutrient agar and PDA mixture. And in the center, we placed the Aspergillus niger strain. And observations were made for sclerotium germination and mycelium expansion of Aspergillus niger. Next, we've done the HCN production. So for the HCN production, we've basically analyzed the production of hydrocyanic acid. So the plates were inoculated with the bacterial isolate, the endophytic bacteria, and then a sterile filter paper strip was dipped in picric acid and was then placed over the lid, and it was closed, and then it was incubated for 48 hours. The coloration of the filter paper strip, if it had turned from yellow to brown, it states that the hydrocyanic acid production was done. So these are the results of the biocontrol mechanisms as stated before: the siderophore production, volatile organic compounds, non-volatile organic compounds, and HCN, in which we can see that all the organisms.
Excuse me, Farah. You have
Yes.
only 1 more minute left.
Okay, okay. Showed the product. Showed siderophore activity, and basically ASEB 1, 5 and 9 showed maximum biocontrol mechanism. And we've also analyzed the lytic enzyme production. The microorganisms use the ability of hydrolytic enzymes for the production of enzymes, such as primarily as part of the biocontrol mechanism by degrading fungal cell wall and preventing plant pathogen to cause infection. So we've done lytic enzyme production of amylase, protease, chitinase, lipase, and lipase. So in that we found out that amylase, cellulase, protease were produced by most of the strains, but chitinase and lipase were produced by none. And concluding and everything into a nutshell, the present study put forwards the biocontrol ability of the native endophytic antagonistic bacteria, in which ASEB 1, 5, and 9 showed maximum activity, but comparing to the biochemical mechanisms, ASEB 1 and 5 showed maximum activity, and they were identified by the basic biochemical tests as Bacillus species and Pseudomonas, which showed excellent in vitro antifungal mechanism. And as the outcome or the future prospects, this can be used as a biocontrol agent to explore the new vistas of biocontrol agents. And further studies can also be done, checking out the molecular analysis, or also going for the endophytic production, silver nanoparticle production of the endophytic bacteria. These are the references. Thank you.
Okay, the session is open for discussion.
May I ask questions?
Yes, ma'am, you can ask.
Parha, you have mentioned about allelochemicals. What are allelochemicals?
Sorry, ma'am. Can you repeat the question once again? I just missed it.
Okay, you have mentioned about allelochemicals. What are allelochemicals?
Allelochemicals are basically for example, the siderophores, the hydrocyanic acid production. Then these are basically the allelochemicals which which is produced by the endophytic bacteria, which will cause a negative effect for the pathogen.
These allelochemicals, are they produced by plants?
No, I have mentioned about the production of the endophytic bacteria, ma'am.
They are not produced by plants. Allelochemicals.
Allelochemicals can be produced by plants as well, but here I've just mentioned about the endophytic microbe.
You're looking for allelochemicals produced by the endophytes.
Endophytic microbes, yes, ma'am.
What are the health concerns of Aspergillus contamination?
The health concerns in the sense of humans or the plants? Humans, okay. As we know that there are mycotoxins which are produced by the Aspergillus species. There are ochratoxins. There is there are aspergillosis, which causes serious respiratory failures in humans. Then, so basically, respiratory illnesses are more into the
Aspergillus niger, a human pathogen.
Aspergillus niger is actually Aspergillus niger can be considered when ochratoxins come. Usually we learn more about the flavors, but here in Aspergillus niger, I have read in a paper that they have the capability of producing ochratoxins as well.
What's the relevance of silver nanoparticle production by this endophyte?
Okay, as we know that silver, ma'am, did you say siderophores?
Silver nanoparticles. You have mentioned that the future prospects involve this silver nanoparticle production by endophytes.
Okay. There are a lot of reports that silver nanoparticles, gold nanoparticles, they are all which are produced from the bacteria itself have the capability of inhibiting the growth of the pathogens. There are reports, basically they infect the cell wall synthesis and that is how they prevent them from expansion and germination.
Okay, thank you for that.
Hello.
Okay, ma'am, are you done?
Yes, Edna, yes.
Okay, there are a few more questions from the other chairperson. The first one is, how did you do the chitinase test? What is the significance of chitinase?
Actually, as we are mentioning about the fungal pathogen, that is the reason why we've done the chitinase test. So chitin agar was generally used so that it might to check whether it might inhibit the chitin. But here, the current study has showed that these bacteria, endophytic bacteria, have no capability of chitinase, so chitinase production. But there are other factors like siderophore. So there are siderophore, the production of volatile and non-volatile compounds, which can be a reason for the inhibition of the respective pathogen.
Okay, the other question is, did you say Aspergillus niger produce aflatoxin?
Aflatoxin is produced by Aspergillus flavus. I've mentioned that there are certain ochratoxins which can be produced by certain species of Aspergillus niger.
Okay, that's it for the questions. Thank you, Farah.
Thank you, ma'am.
Okay, now we move on to the last participant. OPRC 13, Girish Kumar KK.
Yes, ma'am. Can I audible?
Yeah, you are audible.
Am I
Can you
audible?
please share your screen? So you cannot share, I think. Is Shabana ma'am sharing?
I can't.
Okay. Okay, please share your screen.
Yes. I think. Can you see me?
No, it's
Hello?
not visible now.
Okay, ma'am. Sort of problem.
Oh no, it's still not visible.
Uh,
Shabana ma'am?
Yes, Edna.
Ma'am, I think he's having problem with screen sharing.
Is it Girish sir? What? How can I help you?
Yeah.
I think he has sent the slides to our mail.
Yeah, I have his slide. Sir, is it sure that you cannot present?
Let's see. Let's see.
Can you see my, ma'am? Can you see?
No, still it's not visible.
Okay, ma'am.
When you are
Ma'am, please
clicking
put
in the
the.
share screen option, what are you seeing?
When I'm clicking the share screen, stop sharing, and
Stop sharing.
yeah.
Okay. Now, yeah, it started, sir.
Yeah, now it's
Yeah,
coming.
ma'am. It's in Google Drive, photo, documents. Then
Is it in your
screen.
downloads or?
Yeah, ma'am. In my.
Do you have any difficulty
Google
to
Drive.
identify whether the file is?
No, ma'am.
Where the file
Actually,
is?
no, ma'am. I can.
Okay. Don't worry. Meanwhile, I have a general announcement to poster presenting people, the contest category.
Yeah.
See, I'm sorry that we are lagging by 15 minutes.
Okay,
Still, by 6 o'clock,
ma'am,
I
I
think we can
can't.
start the poster presentation. You cannot? Okay. Okay then. Sir,
I
shall
can't
I
share
present
my.
for you?
Yeah. Of course. Ma'am, ma'am.
You try in between, no problem. I will also. Okay,
Yeah.
I would like to make an announcement for the poster presenting people, and we have got 4 papers. I mean posters to show from the Bio-Inventiyon side. We are presenting. We are just explaining what is happening. That is PPS1, PPS5, PPS6, and PPS7. Please make sure that please inform in the chat box whether you are presenting or shall we present. Okay.
Can you show me? Hello, ma'am.
Yeah, I think your screen is now getting visible. Yes.
Yeah.
It's there.
Good evening all. Myself, Girish Kumar Kepe, SIAS SNTP College under guidelines of Dr. Priyasan.
Yes, sir, but okay. It's okay, sir. Ready. Fine, sir.
Yeah, my production of bioethanol from local fruit waste and its optimization. Yeah, moving to the introduction. Bioethanol is an alcohol made by fermentation, mostly from carbohydrate, produced sugar, starch-rich crops, and its waste. Cell-like biomass derived from non-food sources such as trees, grasses, is also being developed as feedstock of fuel production. Fruit paste such as pineapple peel and cashew peel and plantain peel can be used as a materials. The significance of biofuels. They are easy to use. Biofuel provides energy securities and build economic development and greenhouse emission reduction and energy balancing and recyclable and biodegradable. There are 3 mainly ways to be produce bioethanol. Is first sugar converted to ethanol, and then starch converted to sugar and ethanol, and the third one is celluloid and hemicellulosic and converted into ethanol. The bioethanol production is mainly up on three ways. The main method is sugar fermentation, and the other type of method is hydrolysis. There are three kind of hydrolysis: concentrated acid hydrolysis, and the second one is enzymatic hydrolysis, and the third one is dilute acid hydrolysis. Then aim up the objectives of my project. Find out the potential of the food waste to producing reducing sugar, and check whether the fruit waste peels such as pineapple peels, cashew fruit, and plantain peel are able to produce bioethanol or biofuel. Check the potential of the above fruit material to produce ethanol, and optimization of the ethanol production from the fruit waste. The materials of biofuel production. The first one is pineapple peel. These are collected from the local food waste out in the Koni. Hello? Hello, ma'am, am I audible?
Yes, you are audible.
Yeah,
You can continue. No problem.
Okay, ma'am. The second one is cashew fruit. These are collected from nearby household of my. The third thing is banana peels. These also are a biodegradable waste, and which are found in our environmental. But we are moving to the methodology. First one is estimation of reducing sugar. When we are looking in this project, the substance have the capacity to reduce sugar. So first of all, we are estimating the reducing sugar content in all the subject. Food waste containing a wide variety of reducing sugar, notably glucose and fructose. The amount of reducing sugar is a vital parameter of the production of ethanol. The glucose serves as the substrate of enzyme SIAS for the conversion into ethanol. The amount of reducing sugar was estimated calorimetrically at 540 nanometer doing DNS method, and 2 mg per ml of maltose is taken as a standard. The second is immobilization of fermentation is a very big part of the project. So I used the immobilized yeast for the fermentation, and this is a material to make yeast. The third one is estimation of specific gravity. The specific gravity is a main content in my project. Specific gravity is the ratio of density of a substance, the density of a. Here I am taken as the reference substance is water, and the pure water density is 1 gram centimeter cube. The value of specific gravity is used before and the fermentation after reflect the amount of ethanol is formed. In the studies, specific gravity was used to measure of specific gravity's ambient temperature. The bottle is vein empty, filled with the liquid whose has specific gravity to be found at the end. Pre-veined. The difference in the weight is divided in the weight of an equal volume of water to give the specific gravity of the liquid. And then major step of the product is fermentation. 1 ml of extract of each pineapple, plantain, or cashew were transferred into 500 ml of sterile conical flask. The flask were secured tightly in cotton plugs, and the juice then sterilized pressure. Upon the cooling, the juice respectively flask into 50 immobilized beads of kept and closed with the cotton plug and kept in undisturbed. After 7 to 10 days, 10 ml of sample is withdrawn in a sterile pipette and measured the specific gravity, the percentage of alcohol. The percentage of alcohol is determined in following the formula. The percentage of alcohol is specific gravity before minus specific gravity after divided by 0.0074. Here SG1 and SG2 are the gravity respectively. And we are moving to the result and discussions. Apathetic specific gravity or concentration of ethanol in plantain we can clear that the maximum yield of ethanol was in the pineapple at the specific gravity of 0.865 and the minimum at the 0.859. Approximately close to the constant value of ethanol. The effect of specific gravity concentration ethanol cashew fruit is the maximum yield at the specific gravity 785 and the minimum at the 986. Those are also close to the constant value of ethanol. And when we are moving to the last, the plantain peels, we are calculating the specific gravity. The maximum yield of ethanol is formed in at the specific gravity of 0.972 and the minimum at the 1.310. When what next things, the effect of temperature of the concentration of ethanol in the pineapple peel and how the temperature is affecting the yield of the ethanol. In the table we can clear that the maximum yield of ethanol is obtained at the room temperature or 36 degree, 32 degrees Celsius, and the minimum at 36 degree Celsius. In the case of pineapple peels, when we are looking the cashew, the maximum yield is at 32 degree and the minimum at 36 degree. And the effect of temperature of concentration ripe plantain peel, the maximum at 34 degree and the minimum at 36 degree. When we are looking the pH of the ethanol, production of ethanol, and how the pH will affect the ethanol production of pineapple peel, the result resembles that the maximum yield of ethanol was obtained the pH of 6 and the minimum at the pH of 8. And the effect of pH on concentration of the cashew fruit, the maximum yield was obtained in the pH of 6 and the minimum at the 8. And the maximum effect of ethanol was produced in the plantain peel at the pH of 6 and the minimum at the 8. When we are comparing to the three substances which are used to be the pineapple and the cashew, where maximum yield of ethanol is obtained at the 32 degrees Celsius and the plantain peels is at the 34. The effect of pH is the pineapple peels and cashew, they both among the three are the maximum yield of ethanol was at the pH of 6, which are approximately close to the constant value of ethanol. When we are compared to the specific gravity of the maximum yield of ethanol is obtained at the, in the case of pineapple, is at 0.865 and the specific gravity of cashew is 0.785 and the plantain which is 0.972. Those three are which approximately close to the constant value of ethanols. These are the graph showing together. Studies that it is clear that the minimum yield of the ethanol was obtained at the temperature of 32 and pH of 6, specific gravity of 0.865. In the case of pineapple, the temperature is 32 and pH is 6 and specific gravity is 0.785. In the case of cashew fruit, the temperature is at the 34 and the pH is 6 and specific gravity is 0.972. Those are the approximately close to the constant value of ethanol. Let me conclude my From the studies, the cashew fruit, pineapple peel, rye plantain peel have the ability to produce the
Girish, your
reduced
time
sugar.
is over.
So, yeah. Samaya, so can you conclude the bioethanol? It is a
Yes, you can
cheaper method
conclude.
for. Yeah, ma'am. When we are moving to the conclude, from the studies, the cashew and pineapple peels and the rye plantain peels are capable of producing reducing sugar. And with the conclude, the production of bioethanol food waste is a cheaper method. It is also a good method for the waste management. When we are moving to the large scale of production, will help you. The future perspectives of the bioethanol become the more sustainable and replaceable for in the pace of petrol production, and processes have been more efficient. Microbes are used to convert. The social impact of this project is to be it's good for waste management. Okay, thank you all.
Okay, the session is open for discussion.
May I ask questions?
Yeah, ma'am.
Okay, what is
Yes,
the
ma'am.
difficulty? What are the difficulties you will be facing when you try to produce ethanol from cellulose?
Now, actually, hello, ma'am. Actually, my intention is to be, ma'am, how to produce in a cheaper method. So I used to do this kind of material.
Cellulose containing materials are the cheapest. But we are generally not using the cellulose for production of ethanol. Why?
Actually, I think it's ma'am, I know or no. So,
Okay, one more question. You have used specific gravity for estimating the ethanol. Change in specific gravity for estimating ethanol. Am I correct?
yes,
Okay, what are the common procedure for estimating ethanol?
actually done by the fermentation. It's very costly method. So it's not
Is there
affordable
a
for me.
method
So I
using
use
potassium
the
dichromate?
specific.
Is there a method using potassium dichromate?
Actually, much no never about putting method, ma'am.
So, what is the optimum temperature for the growth of yeast cells? Hello, Girish, are you there?
Hello, Girish. Hello,
ma'am.
Girish.
Hello.
Okay. Okay, answer the question.
Okay, what is the optimum growth for yeast? Optimum temperature for growth, maximum growth of yeast.
I think, ma'am, it's 32 degrees Celsius.
Sure, it is 32 degrees Celsius.
Yeah.
Have you checked?
I seen
What is meant by
normal room
constant
temperature.
value of ethanol? What is meant by constant value of ethanol?
Constant value of ethanol is Ma'am,
If you are not comfortable in English, please answer in Malayalam. Okay.
okay, ma'am.
Okay. What is constant value of ethanol?
Ma'am, constant value of ethanol എന്ന് പറഞ്ഞാൽ ethanol production വേണ്ടി ഒരു ലിമിറ്റഡ് വാല്യൂ ഉണ്ടാവും മാം. അത് ഇനിയാണ് യൂസ് ചെയ്യുന്നത്. ഇപ്പൊ എത്തനോൾ ഇത്ര ഇതിൽ വാല്യൂല ഉണ്ടാവുന്നതിന് നമുക്കൊരു ഇതുണ്ടല്ലോ.
കുറച്ച് കഴിയുമ്പോൾ ഇൻഹിബിഷൻ വരുന്നതിന് മുമ്പേ ഉള്ള എത്തനോൾ പ്രൊഡക്ഷന്റെ ആണ് ഈ പറയുന്നത്.
ഇൻഹിബിഷന് മുമ്പും ഉണ്ട്, ഇൻഹിബിഷന് ശേഷം ഉള്ളതുണ്ട് മാം. അങ്ങനെയാണ് എടുത്തേക്കുന്നത്.
Thank you, Girish.
Thank you.
Okay, Girish, you have another question from the other chairperson.
Yeah.
What is the impact of bioethanol on water resource? Will this mitigate climate change? Girish, are you there?
Yeah, ma'am. It may be. Yeah, ma'am. കേൾക്കാം മാം. ഹലോ.
പറഞ്ഞോളൂ.
ഹലോ മാം. It may be.
You can speak in Malayalam.
But hello ma'am, കേൾക്കാം.
Your connection is not stable, Girish.
Yeah.
Hello. Hello. Okay, പറഞ്ഞോളൂ. ഒന്നുകൂടെ ഒന്ന് പറഞ്ഞോളൂ.
Etna, Linu ma'am, and
yes.
Annie ma'am, I think he's having some network problem. Can we move on to the next session?
Okay, Shabana. Thank you for inviting me. Thank you
so much, ma'am. It was a really wonderful participation and a dedicated participation too, I must say. Thank you so much from the organizing team of Bio-Inventiyon 2020. I think we can move on to the next session, our poster presentation category. We have two judges to chair the session. One is Dr. Jasim Bashir. He is from Olomouc University, Czech Republic. And the next is Dr. Mansoor Ali PP, Head, Department of Biosciences, Marambali College, MES College, Marambali, Ernakulam. So I invite both of you for chairing the session. Sir, please.
Thank you, Jishanis. Am I audible to you?
Thank you.
Yes, Mansoor. Yes, Mansoor.
Okay, thank you. So,
hello, Jasim.
Hi.
Good afternoon, Jasim. Yes.
You two are audible, sir.
Okay.
Good afternoon, Jasim and Mansoor.
Okay. Good afternoon.
Okay, Etna, please carry on.
Okay, Shabana ma'am. So we'll start with the poster sessions. The general instructions is that the total time allotted to the participants is 10 minutes, 5 minutes for presentation and 5 minutes for discussion. So the participants can either present their poster or you can send your posters to the conference mail, so that if you have any problems with the screen sharing, we can solve it by sharing our screen. Okay, shall we start? Jasim sir and Mansoor sir?
Yeah, sure, you can start.
Okay. The first participant is PPS1.
PPS 1, Naima Abdul Samit. Naima Abdul Samit. Naima, are you there?
Yes, ma'am. Ma'am, I'm audible.
Wait,
Yes,
I am
you are
sharing for
audible.
her on I am sharing on behalf of Naima. Please wait a wait.
Okay. Okay.
Is it visible?
It's not visible.
Wait a wait, sir, please.
Yes. Yes, Shabana, I see visible.
Okay, Naima, your time
Okay,
starts now.
Naima.
Okay, ma'am. Good evening. Myself, I'm Naima Abdul Samad from MEM College, Marambali. So, the research paper I am presenting here is on the topic development of potential biocontrol agents and its exploitation in agriculture. So, as we all know, biocontrol is a self-sustaining and long-term treatment method for managing the invasive plants. The disease bacterial leaf blight, that is BLB, it is caused by Xanthomonas oryzae, which causes wilting of seedlings and yellowing and drying of the leaves. So, to obtain an effective disease management strategy for the control of BLB in rice, endophytic bacteria isolated from 3 medicinal plants, namely Ocimum tenuiflorum, Biophytum sensitivum, and Erva lanata, were tested for their efficacy against the BLB pathogen. Thus, after proper further research, environmentally safe, long-lasting, and effective biocontrol methods for the management of plant diseases can be developed. So, the aim of the study was to obtain an effective disease management strategy for the control of BLB in rice. So, the methodology first was the isolation and characterization of the BLB pathogen. Then next was isolation of endophytic bacteria from medicinal plants. The third one was antibacterial activity of endophytic bacteria isolated from the plant extracts, biochemical identification of potent bacteria, and finally the evaluation of in vivo action of bacterial biocontrol agent against the BLB pathogen. So from the result, the first result shows yellow circular smooth convex and mucoid colonies on the East dextrose agar, which show after biochemical characterization, it was found that it was Xanthomonas oryzae. Then endophytic bacteria were isolated from the 3 medicinal plants. The extracts were plated on agar plates, and we got 8 isolates. After certain tests like biochemical characterization, among the 8 isolates, the 2 showed most potent activity against the BLB pathogen. We can see that the antibacterial activity of endophytic bacteria, the isolate 2 showed maximum zone of inhibition, and it was taken for further tests. From Rasazurin plate assay, that is MIC assay, it was confirmed that isolate 2 was the most potent one. Then when we did the in vivo action of bacterial biocontrol agents in the BLB disease prevention, it was found that the plant that were both treated with Xanthomonas oryzae as well as Klebsiella species. Klebsiella species was the endophytic bacteria that we took for got from the endophytic bacteria. Then the plant that was treated with both of them showed less lesion compared to that alone infected by the Xanthomonas. So from this present study, it was concluded that the Xanthomonas oryzae was a causative agent of the BLB disease in rice plants, and also from the three medicinal plants, the endophytic bacteria, the most potent was selected from the MIC studies, and it was identified biochemically as Klebsiella species. So this result of our studies revealed that the individual endophytic bacteria reduce the BLB infection under in vivo conditions. So thank you.
Good name. It is an interesting piece of work, and I have a question. Why you choose three? I don't know which medicinal plant you choose, and which are those medicinal plants?
Sir, scientifically it is named as Ocimum tenuiflorum, Biophytum sensitivum, and Airwell annata, that is Tulasi, Mukuti, and Cherula.
Why you were very specifically with those three plants? There are so many medicinal plants.
We selected 6 of the plants, but the most effective ones were these 3. So we took it for further studies. And these 3 were available in the area where we were doing our studies also.
Okay, thank you.
Okay, thank you, sir.
Did you? How did you did the MIC? Did you did from the supernatant or from the extracted fraction?
Sir, I didn't get you.
You said you did the MIC analysis, right?
Yes, sir.
How did you do that? From the supernatant of the bacteria or?
Yeah, we took the supernatant extra. Both were used along with the imipenem antibiotic.
Extract in which solvent?
So, sir, I didn't understand.
You did the extraction, right? In which
Yes,
solvent you did the extraction?
Rasazurin.
Yeah, that is the method used. The chemical used for MIC. Okay. You didn't did any compound analysis on the chemicals, right?
No, sir.
Okay. I think that's all from me.
Thank you, sir.
I have a question.
Yes, ma'am.
You did the in vivo test of the biocontrol agents, right?
Yes.
Okay. Did you re-isolate the endophytic bacteria from the plant? I think you did it in the rice plant. So, did you re-isolate the bacteria?
Ma'am, actually, yes. The one kept was controlled was not at all infected. The one we took was 2 plants we took. One was treated both with Xanthomonas and the endophytic by that is Klebsiella, and the one was alone treated with Xanthomonas only. So it is clear that we have already infected that plant with Klebsiella.
Okay, but then re-isolation only proves that it is because of the endophytic bacteria.
Yes, the lesion is low compared to the other plant.
Okay, any other questions?
Did you establish the endophytic nature?
Sorry, ma'am?
How did you establish the endophytic nature of the biocontrol agent?
Yes, ma'am. We did the antibacterial test. Sorry, antibiotic test. So from that, it was evident that the endophytic bacteria showed more potency against the Xanthomonas because the zone of inhibition was high.
Why did you go for endophyte?
Because endophytes are, we know endophytes are mostly found in plants. Already, almost every plants have endophytes in them.
Oh, okay. Okay, Etna.
Okay, thank you,
Thank you,
Naima.
thank you. Okay, ma'am, thank you.
Okay, we'll move on to the next presentation. The next one is a PPS2 Anas AP. Anas, are you there?
I think he's absent.
Okay.
Edna, I think.
Okay, I think he's absent. We'll call him again later. Okay, the next one. We'll move on to the next one. PPS3 Priyadarshini J. Is Priyadarshini here?
Yes, ma'am, I'm here, ma'am.
Okay, you are audible. Now you can present your poster.
Judges, please note. Second one is absent.
Ma'am, I had mailed my poster to the mail ID, ma'am, provided by
Yes,
the
yes.
conference.
Yes, I'm sharing for you.
Okay, ma'am.
The code number is PP3.
Mami, എന്തെന്റെ വീഡിയോ എടുക്കാഞ്ഞേ?
Naima, please mute your mic.
Okay, Priyadarshini, your time starts now.
Okay, ma'am. So, good evening to all the chairpersons and my co-contestants. I am Priyadarshini from Wales Institute of Science, Technology and Advanced Studies, Chennai. I am going to explain about my review paper titled Pharmacogenetics of Cannabinoids to Cure Cancer and Gastrointestinal Disorders. So, pharmacogenetics is a study of role of genome in drug response. It analyzes how the genetic makeup of an individual affects response to drugs. So the term pharmacogenetics first began around 1990s and the term was coined by Vogel. So now we can see the involvement of cannabinoids in pharmacogenetics. Cannabinoids are a form of chemical substances that are found in cannabis, and it is a compound that make up the plants hemp and marijuana. Cannabinoids that are derived from marijuana plant, which contains tetrahydrocannabinol, that is THC, that provides high depending on the strand from which the drug can come to exist. Currently, the two main cannabinoids that are derived from the marijuana plant are THC and cannabidiol. Both are involved in controlling diseases like cancer and gastrointestinal disorders. CBD has 99% protein binding capability, which leaves 1% accessible across blood-brain barrier for pharmacological actions. Cancer and its therapy. Cancer is the uncontrollable growth of abnormal cells in the body. Cancer cells develop when the body's normal control mechanism stops working. There are more than 100 types of cancers and include various treatments. One among the treatment is pharmacology, including cannabinoids. Cannabinoids produce analgesic effect by activating CB1 receptors, which is present in the central nervous system. So here I have mentioned in figure 1, I had mentioned metabolism of CBD. that is pharmacokinetic that is the pharmacokinetic interaction in the enzyme inhibition involving CYP to 2C9, 2C19 and 2D6-3A4 receptors. In figure 2, I had described the dose dependency of CBD, which involves the clinical experience that CBD is effective in controlled randomized trials. CBD at the doses of 10 and 20 mg per kg in comparison to placebo based on meta-analysis from the available control studies. So now we can see the involvement of cannabinoid in gastrointestinal tract. CB1 receptors are located in gastrointestinal tract, mainly in mesenteric and submucosal neurons, but also expressed by epithelial cells. Endogenous ligands which activate CB1 receptor, that is the endocannabinoids that have identified in mammalian tissue. The best example for the gastrointestinal tract is dronabinol, and it is a non-selective cannabinoid receptor agonist to inhibit gastric emptying and motility. Pharmacogenetic knowledge base has been providing a solution with the research area of genomics, and it has a significant implications.
Hello, Priyadarshini. Sorry to interrupt you. Have you done any experimental work on this particular topic, or this is a kind of reviewing an article?
This is a review, sir. Review article, sir.
Okay. Jisha, miss, this is allowed, right? Reviewing is allowed, right?
No problem, sir.
No problem. Okay, okay. Thank you. You can continue.
Okay, sir. So coming to the clinical trial applications, the goal for the clinical trial application is to have an interface which is simple and easy to use. The web application can reassign a patient to another doctor if the attending doctor is on vacation. And this system was elaborated to support the following core types of users, that is the academic researchers, treatment for clinical trial coordinators, and system administrators. So coming to the applications of cannabinoids includes that the cannabinoids are able to inhibit the growth of in vitro as well as in breast tumor cells. The possible therapeutic applications of cannabinoids are strongly supported, and some novel tricyclic cannabinoid will exhibit fluorescent property. Uses of cannabinoids in medicine includes in controlling blood pressure, helps with painful scoliosis, and preserve healthy eyes, treats with nausea, anxiety, and so on. So future perspective are, though there are potential benefits of this type of medicine, the use of pharmacogenetics in functional gastrointestinal disease and treatment of cancer is still challenging. A complete and holistic strategic treatment that consists of pharmacogenetic-based precision therapy would be at most beneficial. Further pharmacogenetic research examining the appropriate directions for precision therapy of cannabinoids for pain management. So I am concluding my presentation by saying that the cannabinoid system is an almost a ubiquitous signaling system which has been involving in control of cell fate. Recent studies have investigated in the possibility that the drugs targeting the endocannabinoid system might be used to retard or block cancer growth. The endocannabinoids have been shown to inhibit the growth of tumor cells in culture and animal models by modulating key cell signaling pathways. Thank you.
Okay, I have a question, Priyadarshini. So you are talking about cannabinoids that is isolated from cannabis, right?
Yes, sir.
Okay. So what is your suggestion to use, usage of cannabinoids for the treatment? Because already the cannabis is, popular as a drug abuse, right?
Sir, can't hear properly, sir. Can you explain me again?
No, it's like, cannabis, what is cannabis? It is very popular, it will intoxicate and, that's a substance used for abuse, right? Substance abuse.
Yes, sir.
Yeah. So what's your personal suggestion using, cannabinoids for? Or do you have any specific compound for the treatment of cancer and breast failure? Or in other way, I have a question, those who are using cannabis, they are resistant to cancer and gastrointestinal kind of diseases.
Sir, cannabis have good qualities like it can easily fight cancer and it reduces nausea and it helps to protect the brain and reduce the pains. So my suggestion is by using cannabinoids, it is
It protects brain. Already those who are using will be hallucinated, right? Anyway, there is a propagator, there are many propagators, especially, in the film world, and the literary world, people suggest to use this cannabis. I don't know, scientifically, I cannot support this. So that's up to me. We have.
I think there are few countries they are starting to legalize it for medicine, but still it is a question of misuse and misuse.
Yeah.
And I think there are like very few researchers going on in this area who are concentrated on isolating compounds or on do quality research on this area because of the regulation. Very few countries can do that. A few institutes can do that. The main question is, when you bring this to market, what is your suggestion? Will it be used as a drug or it will be like used for misuse? That's the main question.
Sir,
Because the active compound and the active compound which is used in therapeutic is the same compound which is used as the drug, as a drug. That's the main problem in this area. That's a big question. A big.
Actually, morning we had a nice. Actually, morning we had a nice presentation on cannabis, plant gene functioning of cannabis, and its research is going on in USA. But as far as our state is concerned, its usage and cultivation is prohibited. So, though it has, we can't agree with the argument that it is, we can use it as anti-cancerous agent, and also its availability also is it's very difficult because from where we'll get this sample.
Yeah, previously, please, Bashir, please.
Those who are using it are easily getting it, but if you want to do it for research and you want to go through the proper channel, it will be very difficult to do that.
Yeah, in 18th century, there had a war for this cannabis between China and UK, and later only China realized this trade is dangerous for the youth, and they banned this trade of cannabis. So anyway, I don't know, Priyadarshini has any explanation with regard to this?
Etna, I think it's finished. We can move on to the next. Okay. Etna is here.
Okay, sure.
Yes. Okay. I think the next participant is facing some issues and net connection issues. So we'll keep her for the last. Okay. So the next participant will be Rutuja Badle. PPS 5. The code number is PPS 5. Rutuja, are you there? Rutuja? Is Rutuja there?
I think she's having some problem. She just called me and she told me that she will be online. She was there in the room. Yeah. So if she's
Many
not
of them
there, I
facing
think,
such issues.
yeah, if so that we can move on to the next one.
Okay. So the next one is code number PPS6, Minu Matthew. PPS6, Minu Matthew. Minu,
Yes, ma'am.
are you there?
Yes, ma'am. Yes, ma'am.
Yes, you are audible. Yes, you are audible. Can you please start your presentation?
Yes, ma'am. Myself, Meenu. The topic of my ma'am, can I begin?
Yeah,
Hello.
yeah. Yes, you can.
Myself, Meenu. Topic of my work is antibacterial activity of Markantia linearis. Coming to introduction, bryophytes are the amphibians of plant kingdom. They are the largest group of terrestrial plant. However, they are least explored due to small size and lack of experts in taxonomic identification and lack of availability of biomass. Bryophyte has several economic and ecological importance. The studies on bryophyte as a source of antimicrobial components are gaining momentum with the evolution of novel strains of antibiotic-resistant bacteria. It has several unique biochemical pathways that produce bioactive compounds. The present work has thus an attempt on evaluating the antibacterial activity of Mercantia linearis. Coming to objective, collection of Mercantia linearis thallus and sequential extraction of a phytochemical from the dried thallus using a sourceless extraction apparatus. Materials and method. Gametophytic plant was collected from Mutam. The thallus was identified.
Excuse me. It is not possible to view the whole poster, so I think you have to put it. You have to either bring down the size, or it's not visible. The whole.
Okay, sir.
Or you can scroll
Yeah.
down when you go.
Okay. Now, okay.
Okay, you continue. You scroll down when you present
Coming
it.
to okay, now it is okay, ma'am.
Yeah.
Coming to materials and method, gametophytic plant was collected from Mutam. The thallus was identified based on morphological and anatomical character. The thallus was washed well to remove the soil particle and then dried under shade. 30 grams of dried thallus was taken in the sourcelet apparatus and sequential extraction with the petroleum ether, ethyl acetate, methanol, and water has been carried out. The extract was filtered and concentrated using rotary evaporator. The 5 strains of bacteria, Escherichia coli, Salmonella typhi, Pseudomonas aeruginosa, Bacillus subtilis, Xanthomonas were used in the present study. Antibacterial activity of the extract was measured using this diffusion agar method, and the experiment was being replicated. Coming to result and discussion, the gametophytic plant collected was identified as Mercantia linearis. Thallus showed prominent midrib with barrel-shaped air pore. Numerous gemme cup with gemme were seen. Ethyl acetate and methanolic fraction showed antibacterial activity against all the tested bacteria. However, petroleum ether extract and the aqueous extract had no effect. This is a table showing the antibacterial activity. From this table, it is clear that ethyl acetate extract showed more antibacterial activity compared to methanol extract. Highest inhibition was noted to be Bacillus subtilis with a zone of inhibition of 18 mm. Both gram-positive and gram-negative bacteria showed inhibition by the extract, indicated the wide antibiotic effect of the bioactive principle. Several species of Mercantia produce Mercantia A, which is reported to have antibacterial and antifungal activity as per the report of Asakawa in 2007. Pesticidal property of Mercantia linearis has been reported by Krishna and Murugan in 2015.
antifungal activity has been also been reported. However, antibacterial activity of Mercantia linearis is not reported. This is the figure showing the antibacterial activity of Mercantia linearis against the bacteria Bacillus subtilis. Coming to conclusion, present work indicated the ability of bryophyte in having secondary metabolites of economic importance. Further work like axonic culture of Mercantia is going on. It can be used to produce the biomass, which in turn will reduce the impact of collecting the thallus in a large scale on ecosystem. The condition that is to be provided in vitro for the cultivation of the condition that has to be provided in vitro for the production of metabolites as well as the characterization of active principle has to be carried out. Coming to acknowledgement, I acknowledge CSIR for funding me and St. Peter's College for providing me the condition for working. Thank you, sir. Hello.
Thank you, Meenu. Meenu,
Hello.
can you hear
Yes,
me?
sir.
Yeah.
Yes, sir.
So I have a very basic question. So
yes, sir.
you have taken, extract of bryophyte, right?
Yes, sir.
And what is the present status of other bryophytes in terms of antibacterial activity? The one which you have chosen is superior or inferior or at par with the existing bryophyte.
Several antibacterial activity has been reported from biophytes, like bacterial activity has been reported from lunularic acid has been reported to have antibacterial activity. From plagiocalin has been reported to have antibacterial activity. From Mercantia, antibacterial activity is being reported is produced by Mercantin A.
Okay. So the antibacterial activity is superior to the existing bryophyte? That's my question. That was my question.
I didn't get you, sir.
No, it's like, you were, reporting a bryophyte which shows antibacterial activity, right? So my question is that the antibacterial activity showing Mercantia is superior to the existing other bryophytes that has antibacterial activity potential. That's my
question. Sure, sir. Not sure, sir.
Not sure. You didn't go through any literature.
I am not, sir. I didn't get you.
It's very simple. So there are so many bryophytes, right?
Yes, sir.
This is not the only bryophytes. So there might be, studies undergone with regard to the antibacterial activity with the other bryophytes, right?
Yes, sir.
So the one which you have reported is superior to that one or inferior to that one? That's my question.
It's
just selling a product, right? You are selling a product. You should say my product is good or bad or at par with the existing. That's what my question. If it is not sure, it's fine. If it is not sure, it's fine. It's fine. It's fine.
It's not sure, sir.
Okay. Good. Thank you.
Hi. I like to ask you what is the application? First question is, what is the application in which area you are planning to apply this compound? Second one is, what is the novelty? Because there are like a lot of compounds isolated from plants. If you take the list of studies, there are like a lot of compounds. What is the specialty of this isolation? What is the specialty of the compounds which you are expecting? These are the two questions.
Sir, Mercantia produce Mercantian A. This compound has been can be used for many treatment like for liver diseases, kidney diseases. The report shows that in ancient, traditional Adivasis has used the Mercantia for their liver disease and all. So we can use these products for for treatment of liver disease and all. And the second question.
What is the novelty? What is the?
It means further work has to be carried out regarding this. This compounds, the further work is being carried out. We can use this if it is my PhD topic. Means it can be used for preservation of food, and the means and all we can use. And
you are talking about a large area because you are planning to apply it in medical field, and then you are planning to put it to food industry.
Yeah, sir. And also the bryophyte has got a unique property of means the insects are reluctant to eat this bryophytes. We can if we test the anti-fident property of bryophytes, we can apply as a means to in the as an insecticide also.
Okay, thank you.
I have one question. What might be the compound responsible for biopesticidal activity of this plant? Is it the same compound or different?
Not at worked out. The work has to be means is being carried out, ma'am.
So you have to check the secondary metabolite, isn't it?
I just means I wanted to check the secondary metabolites what are present, and I want to analyze its structure, and also whether it means whether this Mercantia linearis has got means many bryophyte means I has been working for many means not Mercantia. There are many bryophyte I am being working, so I want to test the anti-fident property for it and study which compound is the responsible for this anti-fident property also.
Have you done the preliminary studies?
Preliminary means means I am doing culture. After that, I am means in vitro preliminary work on this anti-fident property. Are you asking, ma'am?
No, on the biopesticide property of this plant.
Yeah, I am carrying out the work on biopesticide activity using silkworm. Now now I am doing it. Means I want to apply this on the silkworm, and whether it is reluctant to eat this means the leaf coated with this extract. I want to check it out, and the LCB value and all I want to check. Which compound, after that I wanted to analyze which compound is responsible for this anti-fident property. I want to carry it out.
So it's only a hypothesis, isn't it? You didn't do any work based on this. You're just assuming that it has got biopesticide property, isn't it?
No, ma'am. The insecticidal activity of the Mercantia linearis has been reported by Krishna and Murugan.
So it is already reported.
They reported that it has got means anti-fident property, but they didn't study the compound responsible for this anti-fident property, ma'am.
Okay. What's the Malayalam name of this plant?
Actually, Kallupayal means some Adivasi colony. It is called as Kallupayal, and the in recently I has heard a webinar on by Dr. Manjusi Nair. In that webinar, she has said it is Pasadam.
Pasadam.
Pasadam means
it is Malayalam.
Yeah, it is Malayalam, and actually there is no Malayalam for this bryophyte. And she means Dr. Manju Sinha told that we can call it that it as Pasadam or a Kallupayal. It is up to us. We wanted to name introduce a Malayalam name for this bryophyte.
Okay, okay. Thank you.
Thank you, ma'am.
Okay, thank you. Okay, thank you, Meenu Matthew.
Okay, thank
Okay,
you.
we'll move on to the next presentation. PPS 5. Next is PPS 5. Rutuja Badle.
Yes, ma'am, present.
Okay.
Am I
Rutuja,
audible?
yes, you are audible.
Okay. I'll share my screen.
Okay, please.
Is it now visible?
Yes.
Is my screen visible? Okay.
Yes, it is.
It's
It's not visible completely.
Yeah, it's just to be.
It will come like this only.
Yes. Okay. So can I start?
Okay. Okay, you can start.
Okay. Good evening, one and all. My name is Prituja Badli, and I am a student from D.Y. Patil University. And my topic for the presentation is green synthesis of silver nanoparticles and characterization. Now, green synthesis being a common procedure nowadays because of its low toxic procedures, we chose a plant material that is an agricultural waste to add up to the novelty of this research project. So the leaves were procured from a local market in Thane. They were washed with tap water and cut in medium-sized pieces. These pieces were dried using a tray dryer at 70 degrees Celsius and then grinded into a fine powder. Now, phytochemical extraction of this powder was carried out with distilled water and methanol as solvent using a oxalate extractor. For 16 cycles. So 10% of this extract is used for further synthesis of silver nanoparticles. Quantitative phytochemical analysis of this extract was also performed. Next up was the optimization of synthesis parameters. So first parameter that was chosen was the concentration of silver nitrate. So varying concentration of silver nitrate was used for synthesis of silver nanoparticles, and it was 2 millimolar, 4 millimolar, 6 millimolar, 8 millimolar, and 10 millimolar. Now for the second thing, the ratio of AgNO3 to extract volume was varied. For example, for 1 ml AgNO3, 9 ml extract was added, and for 2 ml AgNO3, 8 ml extract was added, and so on. But the total volume was kept constant at 10 ml. So the third thing that was optimized was the condition. That is, the AgNO3 and extract solution was exposed for 30 minutes to sunlight, dark, and 45 degrees of heat. So all the synthesis that was carried out, so like all the optimization was only by taking one parameter at a time, and rest all were kept constant. Now, by combining the individual results of all the three syntheses, an optimized method was obtained. So 180 ml of 10% extract was dropwise added to 20 ml 2 millimolar AgNO3 solution. Synthesis was confirmed usually when the color of the solution changed from faint brown to dark brown. Characterization of these silver nanoparticles was performed using UV visible spectrophotometer, particle size analyzer, and zeta potentiometer. Lambda max of the silver nanoparticles was obtained at 420 nanometer, which is characteristic to silver nanoparticles. The average particle size, that is Z average, was observed to be at 8.26 nanometer with a very narrow particle size ranging from 7.97 nanometer to 9.10 nanometer. the polydispersity index was 0.546 and zeta potential value was minus 24.63 plus minus 5 millivolts. So the SMP extract solution was then later subjected to repeated centrifugation at 10,000 RPM for about 10 minutes, and a fine shiny black powder of silver nanoparticles was obtained. There was also another study carried out, and that was a kinetic study, which can be seen over here in this graph. So a kinetic study of these silver nanoparticles synthesis shows that the silver nanoparticle synthesis is complete within the first 4 hours of the reaction, which is actually very fast. Like here we can see the values of time that was used and the graphs. And then these are the zeta potential and PSA values, and this is the fine black silver nanoparticle powder. These are the other graphs, changing concentration, changing conditions, and changing volume ratio. So the phytochemical analysis of that plant extract showed the presence of flavonoid, tannins, reducing sugars, alkaloids, cardiac glycosides, and saponins. So talking about the future prospects of this experiment, the active pharmacological ingredient present in the methanolic extract of this plant material has been found to have a potential inhibiting activity over the main protease MPRO of SARS-CoV-2 virus, and that was proved using molecular docking studies. So with further studies, the nanoparticles that have been synthesized here could be used by incorporation in filter layer of mask or the or other equipments of the PPE kit. Thank you.
Thank you, Rituja. So I have a
Thank you.
question. So how do you conclude that the silver nanoparticle which you have synthesized is at nano size?
Can you please repeat the question, sir? I didn't hear.
No, it's like you were claiming that it's nanoparticle, right? So
Yes.
how do you conclude that this is a nanoparticle?
First of all, the color of the solution, just as the reaction started, the color of the solution changed from faint brown to dark brown, and this reaction started within 10 to 15 seconds of the addition. And secondly, the absorbance spectrum of UV visible spectrophotometer showed the lambda max value at 420 nanometer, which is characteristic to silver nanoparticles. Actually, there were many further studies that were to be carried out, but due to the time constraint due to lockdown, we could not do further analysis.
So which analysis would you prefer to prove that this is a nanoparticle?
We were actually AAS can be used to see if it is silver nanoparticles or if silver is present. Elemental analysis.
Yeah, actually, you are checking, the presence of silver. So I am surprised to see that you are claiming that this is a nanoparticle. So there are some basic, experimental procedures to prove nanoparticle. Like TEM, you can go for TEM, right? Transmission electron microscopy. Then you should measure what is the size, and only we can claim that this is a nanoparticle. There can be aggregates of nanoparticles.
Yes, sir.
If it is aggregated, you can't use it for any application.
Yes, sir. The powder that was obtained was not at all aggregated. It was very fine and shiny black powder.
From the eyes, it is not possible to differentiate that.
That's true.
My main question is, you're claiming that it can be used for applying to mask, right?
Yes, sir.
What about the quantity? How can you make up with the quantity?
Yes, sir. The minimum inhibitory concentration is to be calculated.
I'm asking about the efficiency of the production. How much is the efficiency?
For 20 ml AgNO3, we got 20 milligram of the silver nanoparticle powder. So it is quite efficient.
Yeah, but all these for these applications, mainly chemical synthesis is used because you can't manage the high volume using green production. What is your strategy?
Okay.
What is your opinion in overcoming that?
Actually, I didn't know about this thing, but I guess that further studies to increase the efficiency could work, like by increasing the extract concentration. I guess this should work.
Is it possible? Because when you want
Yes.
to apply to basic mask like things, you probably need like kilograms.
Yes, that's actually true. But this is just a basic experiment, or just basic dissertation project that I conducted, and actually it was incomplete due to lockdown. It could not be completed.
Okay, so you were actually planned for XRD and TEM analysis, but you didn't able to complete.
Not XRD, but SEM, FTIR, and antibacterial and antiviral through molecular docking studies through for antiviral.
Okay. Okay.
I have one question. Your synthesis is biogenically. Is there any, in the case of characteristics or in the case of properties, is there any difference between the biogenic synthesis and chemical synthesis of nanoparticles?
Characteristics, I guess. In this experiment, there could not be seen any difference in the characteristics as such, because the lambda max values were at optimum range, that is 420, and there are not much differences between the two procedures that the procedure that I have used and a chemical synthesis procedure.
And also the silver nanoparticles, it's causing some environmental issues. So the disposal of this mask, it is also will be a problem now.
Yes, the disposal was a problem. We were planning to do ultrasonication technique to see that if it gets completely dissolved or no, but again, time constraint.
Actually, in human system means we have some mechanisms to digest it, but in environmental application means it is usually we have to prove some this toxical analysis, something else we have to prove. Then only we can use it.
Okay. Okay.
Okay, now. Okay, thank you.
Okay, thank you.
Thank you, ma'am. Thank you so much.
Shabana ma'am, is Chippy there in the participant list?
Yeah, she joined, but
He's having connection problems.
Yeah, we can move on to the next one, I think.
Okay,
PPS 7.
Okay, we'll move on to the next one. PPS 7. Ajay P. Dev.
Yes, ma'am. Yes, ma'am. I'm here.
Okay, you can share your screen.
Are you sharing or?
No, ma'am. I already mailed the posted to the common mail.
Okay, okay. Visible, right?
Yeah, yeah. Yeah, this is it.
Okay, your time starts now, please.
Okay, ma'am. Good evening. I am Ajay P. Dev from KBM College of Science and Technology. Today I am here to give a short presentation on contemplating the antifungal activity of different solvent extracts of anacardium occidental. And this is a small piece of work which is done as a part of my degree project work. Then, as an introduction, the Ayurvedic system of medicine has been established in India since a number of decades, and it still remains the foundation of the Indian traditional medicine. And with an increasing commercialization of herbal products, there is a need for the scientific community to introduce a quality control for the plant-based medicines. And the infectious disease caused by pathogens, the bacteria, fungi, viruses are always major challenges to the public health. And in that, the microbial activity promotes the morbidity and mortality of the human host. Then the aspergillosis is an infection caused by the type of mold, and the illness resulting from these usually affect the respiratory system. And then the Anacardium occidental is a multi-purpose tree which belongs to the family of Anacardiaceae and which grow on the tropics. And they're relatively grow on dry soil in nature, but in cultivation they also grow well in the tropical rainforest. The present study is designed to study the phytochemical constituents, antifungal properties, and the inhibitory activities of Anacardium occidental leaf extracts against Aspergillus species. Then the objectives, the main objectives is to study the phytochemical constituents of the leaf extracts of Anacardium occidental in different solvents, and also to study the antifungal properties of Anacardium occidental leaf extracts. Then I would now like move on to the methodology. That were the collection and preparation of the leaf samples. Then the preparation of the extracts by cold extraction technique. Then the phytochemical analysis was performed as per standard protocols. Then the antifungal activity by agar-well diffusion method, and the thin layer chromatography was also performed to separate the components from the mix. Then moving to the results, the phytochemical analysis was done, and they revealed the presence of phytochemicals such as the carbohydrates, alkaloids, flavonoids, cardiac glycoside, coumarin glycoside, saponins, phytosterols, fat, oils, phenols, tannins, and proteins in different extracts as secondary metabolites. And this is a graph showing the antifungal activity of different solvent extracts. As you can see, of the 5 different solvents that were the hexane, chloroform, acetone, ethanol, and aqueous, the ethanol extract showed the greatest zone of inhibition that recorded the zones of about 8.8 millimeter against the Aspergillus species. As a part of the initial step for the identification, we have done the thin layer chromatography, that is separation of components from the mix, and the RF values recorded were 0.34, 0.65, and 0.72. The spots were appeared as yellow and orange colors. Then moving to the conclusion, the phytochemical screening of the Anacardium occidental in hexane, chloroform, ethanol, acetone, and aqueous leaf extracts indicated the presence of alkaloids, flavonoids, and coumarin glycoside, phenols, metabolites, and then the acetone leaf extracts of Anacardium occidental against Aspergillus species that showed characteristic zones, and out of which the ethanol and the acetone leaf extract showed the maximum zone of inhibition. And in light of these results, this work justifies the therapeutic use of Anacardium occidental in different medicines to treat various diseases, and also the antifungal efficacy of the leaf extracts lends credence to the ethnomedical use of the plant to treat various diseases. And this is a small piece of graduate level project work, and further research is required to isolate the bioactive compound which is responsible for this antifungal activity, and also to study the active principle, chemical structure, and the mechanical action of the activated compounds. That brings me to the end of the presentation. Thank you for your attention. Thank you, sir. Thank you, ma'am.
Thank you, Ajay. So, which was your fungus for testing antifungal activity?
That were Aspergillus species, sir.
Aspergillus species.
Aspergillus.
Okay, so how you design the experiment for different extract preparation? Did you do duplicates or triplicates?
Sir, we did the triplicates. We did the triplicates, and that in that graph we shown the average of the triplicates.
Okay, you could have, instead of that, you could have shown the standard deviation. That is the usual protocol for
Sir, can't hear you, sir. Well, can you please say once more?
No, it's like we should show standard deviation while you are designing. That is the right way. Doing triplicate is the right way. So we must show the standard deviation in the graph instead of showing average.
Yeah, sir, but in the poster, I didn't got much space to show all of the triplicate data and this graph. That's why I selected the graph.
Standard deviation is different. You don't need much space for that.
So
Anyway,
sorry, sir.
you are a graduate student, so it's taking time, right? So it's a general
Sorry,
statement, right? It can be used for therapeutic as therapeutic agent, and all the compounds that is available in the plant is there in your extract house, glucocorticoids, carbohydrate, everything is there, right?
Yes, sir.
Flavonoids.
Yes, sir. Yes, sir.
So what is your thought, which will be which compound or which specific compound will be there in your, this is cashew, right? Anacardium is cashew, right?
Yes, cashew, sir.
Yeah, the cashew leaf. So do you have any suggestion? This will be the active principle, and that is a possible reason for antifungal activity.
Sir, I don't have a clear answer because I didn't do any further experiment or research to find which is that bioactive compound. But we can say that on the phytochemical screening, we have got almost all of the secondary metabolites. That is, I already mentioned about all of them. But so of these, on the light of that, the results of the phytochemical screening, we can say that any of them would be the cause for the or the chemical component for this antifungal activity. And as a initial step for finding which compound, we also just done the TLC, thin layer chromatography, and we compared the RF values and just identified that compounds were alkaloids, flavonoids, and terpenoids.
And which leaf was your choice? A matured leaf or a tender leaf? And
Sir, mature leaves, sir. Mature leaves. Mature leaves.
Thank you, Ajay. Wish you all the best.
Thank you, sir. Thank you.
Hello.
Yes, sir. Jasim, sir.
Why did you select this particular solvents?
Solvents,
usually.
Sir, I already told that this is just part of my degree project, and for my main project, I was also using this plant for the antimicrobial activity. So I choose these extract solvents on the base of their polarity, order of their polarity.
Order of their polarity. Okay. What about the cashew itself? The fruits
Cashew-
are having much more metabolic property than in the case of cashew, I think.
Yeah, sir, but well, I saying that in my house I have seen that when my in my house when on the preparation of pickles, I seen that my grandmom using the fresh cleaned anacardium leaves for covering it, and she said that it will avoid the contamination. So as a biotechnology student, I thought that it would have a compound which support that antimicrobial activity. That's why I selected this plant or this leaf, sir.
Okay.
Okay, thank you, sir.
Wonderful explanation.
So I told that we are biotechnology students, right? So we have to think about that, and it's a real life experience that and Anma and nothing. Okay.
Yeah, it's like we must appreciate him.
His presentation.
Yeah, he has used that traditional knowledge.
Yeah.
Traditional wisdom.
That is not me, sir. That appreciation will goes to directly to my grandmother. she is the one who used these freshly green leaves used to that cover that pickles, and when I asked that, she told that it will avoid the contamination, and so there is no then when she use the organic vinegars, right? Organic vinegar, and also use these leaves to cover it, and she said that it will reduce the rate of contamination, or it will prevent contamination. So I just thought there will be any component to support that activity or something.
Good.
Thank you, sir.
Okay, thank you, Ajay. Next
Thank you.
Next presentation is PPS4. PPS4, Chippy Suresh.
Thank you.
Thank you, Ajay. Chippy, are you there?
Yes, she is in the room.
Chippy, please respond. Are you there?
Chippy, please unmute and respond, please.
Chippy?
Hello?
Okay. Yeah,
Hello?
yes, you are audible. Okay, Shabana ma'am, can you please
Okay.
present her poster?
Actually, she didn't give
Yes,
the
she has
poster,
sent.
I think.
She has sent.
You sent?
I saw it now.
I had made
Wait.
you. Hello, ma'am?
Chippy, you have sent to me, right?
Yes, yes, she has sent
Yes.
To Bio-Inventiyon or
Yes.
it. Yes, yes.
okay. Let me check. Actually, I didn't note on it because other file I got.
Actually, ma'am, I
I'm
have
sorry.
some.
Please give me.
She's having network problem,
Okay.
Shabana ma'am. So okay, Chippy, can you please try sharing the screen?
Yeah, you got it. Recently only she shared. That's why. I'm sorry.
Okay,
I'm
okay.
sharing it.
Okay. Shabana ma'am, you share it. Okay, Chippy, is it visible? Hello, Chippy.
No, she's there only.
Chippy, is it visible to you?
Hello?
Yes, Chippy. Chippy? I think she has left the meeting.
Yeah. So
Etna, you can present.
Will it be okay, Shabana ma'am, if I present?
I think if the chairpersons are okay, I am also okay. Sir,
Actually, I'm
they are the
seeing this
co-authors.
even though I'm
Hello?
the second author, I'm seeing the poster for the first time now only.
It's fine. You can present it.
Hello, ma'am.
You can do that.
Chippy is back.
Hello,
I
Chippy,
think.
you are here.
Okay,
You are there.
back.
Okay, Chippy, you are there. Hello, Chippy. You are wasting our time.
No, she may be. No.
Let Etna present.
Hello?
Okay.
Okay.
Yes.
Hello?
Chippy,
Okay,
you
you
can
are
continue.
audible.
You are audible. We can get your response. Don't worry.
Hello, ma'am?
Continue. Yes.
I think she's not able to hear also.
Yeah.
Hello, Chippy, you are there? Etna, you can present, no problem.
Are the chairpersons okay with it?
Mansur sir said okay. Jasim sir? The registered
Jasim,
participant is facing some network issue, so she cannot present.
Yeah.
Is the second author? Will you allow the second author to present?
Yeah,
She is
it's
present
okay.
here.
It's okay. Okay. If you are low, yeah, it's fine.
Okay, now carry on.
Okay. Hi, good evening, everyone. The topic of the presentation is in situ biodegradation chlorpyrifos by an optimized bacillus consortium. This was my project done 3 years back. Then, as we all know, chlorpyrifos is an organophosphate pesticide, which is a xenobiotic and can persist in the soil for a longer time. The figure shows the harmful effects of the persistence of chlorpyrifos. So the only alternative, eco-friendly alternative to remove the chlorpyrifos residues from soil would be the use of bioremediating microorganisms. So the objectives of our work. Shabana ma'am, can you just move the slide a little down? The objectives of my work were to optimize the physiochemical parameters of chlorpyrifos biodegradation. And Shabana ma'am, can you just move the slide downwards? Okay. And to evaluate and statistically analyze the effect of bacterial isolates in consortium on the total microbial count and dehydrogenase activity, and also to understand the relationship between the total microbial count and dehydrogenase activity using statistical analysis. So basically what we done is we got a bacillus consortium which showed high degradation, chlorpyrifos degradation, and we optimized the physiochemical parameters like the inoculum density, chlorpyrifos concentration, temperature, pH, and incubation time. And then we used this bacillus consortium in situ in agricultural soil, and then we checked the soil degradation analysis, total microbial count, and soil dehydrogenase activity. There were treatments. The bacillus consortium had four isolates, all belonging to bacillus species, and we had treatments with single isolates and the last treatment was having all the 4 isolates. That's a consortium. And then the degradation analysis was done using HPLC analysis, and after optimization we could get the following values as as it is depicted in the table. The inoculum density. Shabana ma'am, can you just move the slide? Okay, the inoculum density was 1%, chlorpyrifos concentration was 100 ppm, temperature 30 degrees Celsius, pH 7, incubation time 6 days. And using this optimized parameters, we did the in situ degradation studies in agriculture soil. And then we could find higher degradation in consortium treatment rather than treatments having single isolates. Also, the total microbial count of each treatment was studied, and the total microbial count was higher in the treatment having a consortium rather than the control treatment. And the hydrogenase activity was also studied, and it also showed consortium also showed higher dehydrogenase activity. We also did a correlation analysis of both the total microbial count and the soil dehydrogenase activity, and it was positively correlated, which proves that total microbial count is a measure of dehydrogenase activity is a measure of the total microbial count in the particular soil. So finally, we can conclude that the particular consortium can be used in agriculture as as a fertilizer in order to reduce CP residues in agricultural soil. Thank you.
Thank you, Edna.
Thank
Sir,
you, Edna.
any questions?
Yeah, it's like, Edna was saying in introductory session. This is a 3 years back work while she was doing her project. So my question is, how relevant this particular work now? It's been 3 years now.
So now, as of now, the pesticide is banned over here, and 3 years back it was not banned. And 3 years back, people were using this, so there might be residues in soil still. So in order to wipe out all these residues, we can use my consortium.
It's like, what is happening? This is a kind of biotransformation, right? Using the bacillus, consortium, you are transforming the CP, right? The chemical into some other chemical. So, which is the transformed chemical? Do you have any idea of that?
No, we did not go for the compound analysis. So we don't know the intermediates. So we just did the work till now, till this.
So how it affects the microbial diversity? That is another concern, right?
So that we have done here, sir. We just checked the total microbial count also. And the total microbial count was unaffected. In fact, it was increased, I think. Yes, it was increased in the consortium
Okay,
treatment.
while you are treating the consortium, yeah.
Yes.
Okay. Thank you.
Thank you, sir.
How did you select this consortium? What was the source
Sir,
of this?
that was our previous work. It's actually like we isolated from the agricultural fields itself. Organisms, bacterial organisms, we isolated from the agricultural fields, and then we screened them for chlorpyrifos degradation, and then the candidates showing higher degradation were assembled into a consortium, and the consortium was also screened for chlorpyrifos degradation and showed, I think it is 94% of CP degradation. It showed in vitro.
Sorry.
So these all these microbes were compatible with each other. You checked that
Yes,
also.
we tested the compatibility also.
So they were not inhibiting others.
No. Actually, we have submitted that in a journal, and it is yet to be published.
Okay. It is already 3 years. So, what was your idea to like? Did you design a carrier or something? No.
Didn't get you, sir.
Did you design a carrier or something for application?
No, sir. We did not go to the formulation level. We just stopped our work till here.
Okay. Okay.
Actually, though the chlorpyrifos is banned, still farmers are using it. Because of the efficiency of the pesticide, farmers are unauthorizedly using this pesticide. And it has so many toxic effects, even it's in the aquatic fish. Because in Kutanad area, they are dumping this pesticide and it's affecting the fish, causing fish toxicity.
But the problem is, Mansoor sir said, like when you biotransform on compound, it will go to probably the transformed compound can be also more dangerous than the parent compound itself. That can be possible also. In some
that
cases,
is there, but they can use it as the carbon compound, the pesticide. And in another PhD thesis, we found that up to carbon dioxide level, we could find out the pathways. We could analyze the pathway, metabolic pathways.
Okay. Because that is more important when we do something on these kind of things, because it is going to the soil. That's the main thing.
Actually, the first compound is TPP. It is more toxic than it is chlorpyrifos.
Okay, okay.
It is more antimicrobial.
Okay, sir, I think it's finished. So we have concluded the post presentation contest category of students with the unexpected presentation of our program manager right now. So let us move on to the post presentation faculty or researcher category for contest. Let me invite the first participant with the code PPFR1. PPFR1. Dhanraj, are you there?
Yes. Am I audible?
Yeah, you are audible. Please share your screen. I hope Edna will manage from this. Okay, Edna.
Okay, okay, Shabana ma'am.
Thank you.
Okay, Dhanraj, you can share your screen now.
Is it visible?
Yes, it is visible.
Okay.
But it is not visible.
It's not clear.
The full poster is not visible. So you have to scroll down, I think.
Yes, I have to scroll down it while taking.
Okay. Okay, okay, Dhanraj, I think you can start.
Good evening, respected teachers and colleagues. I'm going to present a small topic, like a small diversity study of particle associated bacteria in mud banks of Alabora, which is a dissertation work which I have done 4 years back. It is carried out in National Institute of Oceanography under the guidance of Dr. Anas Abdul Aziz. So, mudbag, it's actually is a unique phenomena which is found in southwest coast of India, especially in Kerala coast. You might have heard about Chagara and all those things. When it comes, there will be profound fisheries over the coast, and there will be more availability of fishes in the coast. It is due to a large amount of suspended particulate matter, and this suspended particulate matter found in mud bank is mostly composed of particulate organic matter, POM, and this POM, which plays a major role in the in that area where mud bank is there, and this POM may have a major role in the vertical transport of nutrients, like upwelling of the nutrients from the benthic zone of the seed to the upper surface portion, so that many surface pelagic fishes will have it. And this inside the POM, also there is more number of microorganisms are also residing by attaching by associating with the POM, that is particle associated bacteria. So my study was mainly concentrated to the particle associated bacteria which is associated with that POM. Here you can see a microbial loop. Here the POC, that particulate organic carbon, which is found in the particular organic matter, and inside that the enzymes. It is many previous studies shows that enzyme in the particular organic matter will convert the they have many hydrolytic enzymes they have studied, and those hydrolyzes more particular organic carbon or matter than required, more carbon which is required uh, um, by the microbes associated with that particular organic matter, and ultimately it will not only benefit those bacteria which is residing inside the particular organic matter, which is also that will also benefit for other microorganisms and phytoplankton, zooplanktons, and higher trophic level or organisms and all, and also to the benthic organisms and all. So that is why the POM is very important. Also the particle associated bacteria seeks its important in that case. And here the materials and methods, here the already available, like collected samples or purified samples. I have done studies on that. They are again purified and extra, and they were subjected to the 16S PCR for 16S PCR. And by using this 16S PCR products, I have performed ARDRA. For grouping the isolates, because I have got more than 100 isolates, and also for sequencing and all those things, I have to cut short it. So for selecting a representative group, selecting this identifying the similar groups, I have to perform ARDRA. Based on the bands obtained from the ARDRA, I have selected the representative members from each group, then subjected for, and thus representative members have been subjected for sequencing. And next is a box PCR was performed. Actually it will gives more clarity within the strain, and based on it clustering was obtained. Clustering was done based on that, and results showing I've captured the result into the most dominant group was found to be Pseudomonas. And here I have found the most Pseudomonas species like Pseudomonas mendocina, Pseudomonas otidis, Pseudomonas geniculata, Pseudomonas angulicipeta, and Pseudomonas aerogenata, etc. And next comes the Acinetobacter species. The Acinetobacter species found that Acidobacter oleovirens, Acidobacter piti, and Acinetobacter baumannii, Acidobacter venetianus, etc. And third one was third more diverse species was the Bacillus species. And this was one like Bacillus infantis, Bacillus thuringiensis, etc. And other species includes like Halomona species, Stenotropomona species, Alcaniora species, Enterobacter species, Psychropacter.
Dhanraj, your time is up.
So by this study, we can conclude that a good spectrum of diversity was found, and the future study should have to be focused on the ability of those particle associated bacteria and its wider applications, like in bioremediation or any other as a biocontrol agents or anything. So future studies have to be focused in that area. By this I am concluding. Thank you.
Thank you, then, Raj. So I would like to know that what is this marine snow?
Marine snow, actually it is actually it is an aggregate of particulate organic matter only, sir. Inside that marine snow there are many microorganisms are associated with it, and the enzymatic activity releases more amount of decomposes the that organic matter as a carbon source and releases nutrients, which will ultimately helps all the food chain on web, indirectly and directly.
Okay, so you have identified a lot of microorganisms that is associated with the particle,
Yes.
right? Organic
Yes,
particle, right?
yes.
So in this study, do you have any control? So as to show with this control, we have found this much microorganisms which are beneficial, and they are highly producing hydrolytic enzymes. And do you have any control?
No, sir. No, sir. Actually, here the enzymatic studies have not been done. I have the enzymatic hydrolytic enzyme activities. Actually, I have told based on the reviews and all.
Literature. That's fine. That's fine. No, still, you are reporting there are so many beneficial microflora that exist with the mud bank. And I just wanted to know whether you have any control with some other, organic matter.
No, not some other.
So as to compare,
no, sir.
This mud bank is having this much microflora, and the other organic matter is having only this much microflora. That might be one of the reason for this diagram. Do you have those kind of conclusion? I am asking just out of curiosity.
Yes, sir.
I think that question is more relevant because the Pseudomonas acinetobacter and Bacillus are the common species. You can find it in anywhere.
Yeah, elsewhere. Yeah.
There is something interesting. You have to have a better control.
Yes, sir.
To see the difference. To say that in this case it is different from normal soil.
Yes, sir.
So do you have anything to say then, Raj, regarding that?
No, no, sir. Controlled, we didn't compare it with any controlled experiment.
Yeah.
Like in experiment ways, we didn't done that thing. Actually, it is a dissertation, so time span is very
Yeah,
less.
it's like otherwise we cannot say,
Mm.
the superior in the, in having, so many microorganisms that is associated with the mud bank. That is very, noticeable phenomena in our place. So normally it will be more, scientific.
Yes, sir.
And there
Yes,
is
sir.
another spell check. Please check your spelling of bacteria in the lower part. Good for
Staphylococcus.
the study agility, those particle associated. Yeah.
Okay, okay.
Yeah. That's it. Yeah.
Any more questions?
I have a question. Why Adra?
Actually, this was like just a preliminary grouping for doing a preliminary grouping, because I have like purified and gram stained almost 214 isolates, something like that. So that due to like similar members or same species can be obtained for the repeated times. For reducing that chance, like my guide suggested to first go for Adra, then group the similar members, because the bands will be similar for the same species. So by that, how many number of similar species are there, we can sort out, and from that any one of the species can be taken as one of the member can be taken for and considered for the sequencing. For that easiness, we have done the Adra.
Okay. Please correct your spelling for Staphylococcus also. In the other species section. Okay. That's all.
Okay. Thank you, Dhanraj.
Okay.
Now we'll move on to the next presentation. Code number PPFR2.
Yeah,
Dr.
ma'am.
Dr. Sheikh Adil Hamid.
Yeah, ma'am. I'm here.
Yes, sir, you are audible.
Okay, ma'am.
Dhanraj,
Thank you.
meanwhile, please stop sharing.
Okay, miss. 1 minute. Oh my god.
Yeah, it's fine.
I stop sharing. Okay, miss. Okay. Thank you.
Thank you.
Is it visible, ma'am?
Yes, it is visible.
Okay, thank you, ma'am.
Can you make it a little more larger?
Yeah, I'll do it section by section so that it will be viewed very easily.
Yeah.
Okay, sir. So you can start.
Okay, ma'am. Thank you. A very good evening to the organizing committee and jury. I'm Dr. Sheikh Adil Hamid, assistant professor from SCOS, Kashmir. I'm presenting my research entitled Effect of Water Acidification and Sanitation on Gut Morphometry and Intestinal Microflora of Broiler Chicken. So let me start with the introduction. I'll zoom in section by section. Let me start with the introduction. Providing good quality water is important both from health and welfare perspective, and same goes for the animals and poultry birds as well. The quality of drinking water for poultry and its effect on performance, it has been a concern for poultry producers from many years. Good quality water, it's very important for good digestion and to create a healthy gut flora, which helps the bird to absorb better and grow better. There are a number of ways. The water mostly in poultry farms it's mostly contaminated. So there are a number of ways by which we can improve the quality. In our study, what we did two things. We used acidifier and sanitizer. Uh, the sand acidifiers they are beneficial in a way that they decrease gut pH. What happens by that? Most of the harmful bacteria they grow at relatively higher pH. So this way it decreases the number of harmful bacteria. And acidifiers also they have direct antimicrobial antibacterial activity, and additionally they have benefit of gut. They have quality of gut cell proliferation. Next was the sanitizer. Uh, the um, the sanitizer we used that had active ingredient didecyl dimethyl ammonium chloride. These are quaternary ammonium compounds, and what they do, they also have antibacterial activity. The oxidability of these compounds it results in antibacterial activity. So the study was set with two objectives. We wanted to see the effect of water acidification and sanitation on gut morphometry and also on intestinal microflora of broiler chicken. We divided the 144 broiler birds into four treatment groups. Treatment groups. Each treatment had 3 replicates of 12 chicks each, 12 birds. T1 was kept as control, wherein routine water was used. In T2, acidifier was used at 1 ml per 20 liters of water. In T3, sanitizer was used at the rate of 5 ml per 20 liter water. In T4, combination of acidifier and sanitizer was used at same doses. Then afterwards, what we did at the end of trial, that is 42 days. In broilers, we researches the trial period is 6 weeks. After 6 weeks, 6 birds per treatment they were selected at random and sacrificed for the estimation of various parameters. Length and weight of small intestines were measured. Afterwards, small intestines, while contents from small intestines were collected aseptically and used for the estimation of cecal coliform count. The results suggested that there was a significant improvement uh, in the length and weight of small intestine in the groups wherein acidifier was used. That is T2 and T4. Sanitizer did not have that effect. As I already mentioned, that acidifiers they have got they have quality of direct stimulatory effect on gastrointestinal cell proliferation. That's why we found that the length and weight of small intestines was increased in acidifier groups, wherein acidifier was used alone and in combination with sanitizer. But sanitizer alone could not could not do this thing. Also, we checked the crop and duodenal pH. Crop and duodenal pH was again reduced in the groups wherein acidifier was used. And the most importantly, cecal coliform count. Cecal coliform count was there was significant reduction in cecal coliform count in all the treatment groups, wherein acidifier and sanitizer treated water was used. Highest this reduction was found in the group wherein combination of acidifier and sanitizers were used. This is a pictorial representation of the slides. Brain heart infusion agar was used. In conclusion, we can conclude that water acidification helps in improving intestinal length and weight. Both water acidifier and sanitizer are beneficial in reducing the number of harmful cecal bacteria in broiler chicken. Thus, they may be recommended for improving the water quality and gut health of broiler chicken. Thank you. Thanks a lot.
Thank you, Dr. Sheikh.
Thank you, sir.
Yeah, it's like, what was the pH of the water you have chosen?
Sorry, sir.
No, You have used water, right? And you
Yes, sir.
acidified that water, right? So what
Yes,
was
sir.
the pH of the water you have used? pH.
pH of, we didn't calculate the water pH, sir. We calculate the pH of gut contents.
Okay.
Gut content of the bird, crop and duodenum.
Okay. So what was the change in that?
Significant reduction, sir, in the groups wherein acidifier was used.
Okay. So what do you use for acidification purpose?
We used this Acivet is the one brand. So that is a cocktail of various these acids, short chain fatty acids like fumatic lactic acid. It was a cocktail.
Okay.
Yes, sir.
So this is usually used for poultry farms.
These are not usually used. That's why the people are complaining that those early mortality in early days. E. coli, this colibacillus, this is a very big problem, sir. People, they don't use it. These are, despite being cheap, these acidifiers, they should be used. They are very beneficial in improving, in controlling the early mortality of these forms, and thereby they can be economically very important.
Okay. So the weight of the broiler has been increased after using the acidifier. That is your finding?
Yes, sir. Economically, it was very viable.
Okay. Thank you.
Thank you, sir. Thanks a lot.
Is these acidic compounds are harmful to the poultry, or this?
Yes, sir. Yes, sir. These can be used either in powder form in the feed, or in the water in liquid form.
I'm asking whether it is it's harmful to the bird or it's not harmful.
No, sir. They can prevent the mortality, sir.
Because you are saying for sanitizer you use quaternary ammonium compounds, right?
Yes, sir.
But it can be harmful, right?
So I didn't hear you, sir.
When you use quaternary ammonium compound,
yes, sir,
it can be harmful.
But the dose designated by the company wouldn't find anything of that sort.
Okay.
Yes, sir.
Okay. But you didn't check it, right?
No, there would have been a mortality if it had been harmful. Mortality of chicks had been happened. That thing didn't happen, so
but you
we
are
can
using a very low concentration.
Yes, sir.
It will not lead to mortality, but it can be,
yes,
can lead to some other, like when you have contaminated meat, you'll have other problems.
Apparently the birds were healthy, but I don't think at that dose there may be that residual problem or something else that.
Okay.
Yes, sir.
Any more questions? Okay.
Thank you, sir.
Thank you, Dr. Sheikh.
Thanks a lot, ma'am.
Okay, we'll move on to the next poster. Next code number PPFR3. PPFR3, Vidhi Patel.
Yes, ma'am.
Yes, you are audible, Vidhi.
Yes, ma'am.
Okay. Can you just share your screen?
Yes, ma'am.
Vidhi, your voice is echoing.
Wait, ma'am. Hello.
Still it is echoing.
इश्यू आ रहा है पता नहीं.
Edna ma'am, I think she is joined the Zoom with 2 devices. If one of the device is muted, this echoing can be avoided.
Vidhi? Ms. Vidhi Patel?
Yes, ma'am. Okay, ma'am. अभी एक में हो गया मेरा.
Yeah, now it's fine.
Now it's fine. Now it's fine, Vidhi. You can just
Okay, ma'am.
share your screen.
Okay, ma'am. वेट मैम प्रॉब्लम आ रहा है मेरे फोन में.
Okay, okay.
Can I start in laptop?
Will you take time for that?
No, I in 5 minutes. 2 minutes. 2 minutes only.
Or shall I call the next one and then you can present after that?
Okay, ma'am.
Vidhi, if you're ready, I will share for you. Your PPT
Okay,
your is ready here.
Okay, ma'am.
Can I share for you?
Hi, yeah, ma'am.
Yeah, then please stay.
Okay.
Please stop sharing, Vidhi.
Okay, ma'am.
Please stop sharing.
Okay, ma'am. Done.
Okay.
Okay, Vidhi, is it visible to you?
Yes, ma'am.
Okay, Vidhi, you can start now.
Okay. Good evening, sir. Myself, Vidhi Patel. Today, I present over here bioremediations of paper and pulp influent using actinomycetes. This project is given by the GSBTM grant, government through. Wait. Okay. The overview of this presentation. Paper and pulp is a major problem. Paper and pulp influence is a major problem in India. Is the fifth largest contributor in the industrial water pollutions. The paper and pulp industry is a highly water intensive, consuming 100 to 250 freshwater ton paper. Effluent generated at the pulping stage is a dark brown color in the dissolved lignin and its degradation products, hemicellulose resins, acid, phenols. Biological treatment methods involves to use fungi, bacteria, algae, actinomycetes, enzymes, as well as single-step treatment or in combinations with other physical and chemical methods. Bioremediations of the paper and pulp influent which have the capacity of efficient bioremediations of organic and inorganic pollutants. In this research, we focused on the actinomycetes because of lots of work on degradations of paper and pulp waste, degradations in bacteria, fungi, protozoa. All works are finished. Then we focused on actinomycetes to degrade the paper and pulp waste present in pollutants like decrease the COD, BOD, carbon, nitrogen, etc. Then material and method, the collections of the waste into the paper and pulp industry near Kadi area. Study of the physical parameters of the waste, pH, conductivity, temperature, color, alkalinity, acidity, and the chemical parameter of the waste, phosphate, sulfate, COD, BOD, carbon, nitrogen, TS, CDS, all parameters as study of the waste. Then isolations of actinomycetes from the waste. by serial dilution method. Actinomycetes can grow on the different media, but hello, ma'am.
Yes, you are audible.
Wait.
Okay, स्क्रीन नहीं आ रही है मैम मेरी.
अभी आ जाएगा विधि.
Okay. We focus means actinomycetes can grow. We are grow actinomycetes grow on a SIAS medium, casein starch medium. Then identifications of actinomycetes by gram staining observed under microscope. Screening of the actinomycetes for a cellulose enzyme by extracellular method and estimation of cellulase by DENSA method. Enzyme assay by DENSA method. Screening of actinomycetes for xylanase enzyme by extracellular method and estimation of xylanase enzyme by DENSA method. Then study plate assay. After screening of the actinomycetes, we are focused on enzyme activity. Enzyme activity is done by DENSA method. Then enrichment of cellulase, pectin, xylan degrade the microorganism in liquid medium, followed by primary screening and isolation on solid medium. Cellulase activity is measured. Wait, ma'am. Hello. Okay.
Yes, yes.
Okay, ma'am. Then 15 actinomycetes are isolated from the paper and pulp waste, and then activation culture broth of the cellulase, xylanase, and pectinase degrade the degrading microorganisms. Primary screening of the cellulase, pectinase, and xylan. Optimization of the then activation of culture broth, and then enrichment of the culture. Then they perform the cellulase activity, xylanase activity, as well as pectinase activity. Then activity is measured by the taking OD at 540 nanometer, and standard graph was made by using glucose as a reference to measure the unknown cellulase activity. The OD is directly proportional to increase in enzyme activity. Then enzyme activity, day 1 to the till the day 10, the increasing order in the enzyme activity of the actinomycetes. Then proximate analysis of waste sample, pH is
Vidhi,
7.5.
your time is up. You can wind up.
Okay, ma'am. Then means we are just focusing on actinomycetes that degrade the pollutant present into the waste, and black color is removed, and we try to keep black color is removed from the paper and pulp waste. Okay, sir, thank you.
Thank you, Vidhi. What's your conclusion?
Primary screening of the enzyme xylanase cellulase gives a large zone of inhibition better than pectinase enzyme. Means selected isolates, they are degrading the they are base activity for the degrading the pollutants present into the waste.
Okay, what is the purpose of checking inhibition? I couldn't get that. So you are looking for enzyme activity, right?
No, sir. We are focus on the paper and pulp waste. Means paper and pulp waste, toxic pollutant present into the paper and pulp waste.
Okay.
So BOD, COD, or decrease the pollution degradation process.
Okay, so you have used actinomycetes for
Yes, sir.
degrading the pollutants, right?
Yes, sir.
Okay, so you have found that the BOD and COD level is found to be increasing while.
Yes, sir. First day we are, before treatment of actinomycetes, BOD is a high. After treatment, enrichment of culture, and after treatment, BOD is a decreased.
Okay, so from where you isolated these actinomycetes?
Because of no study on actinomycetes.
I'm asking, from which place or from which area you isolated these actinomycetes. That's my question.
Sir, I can't understand.
No, you might have isolated actinomycetes, right? So,
Yes, sir.
so from where? What was the source from where you isolated these actinomycetes?
Paper and pulp waste.
From paper and pulp waste only you isolated and you used enriched
Yes.
actinomycetes and again you used in the same. Okay.
Yes, sir. Yes, sir.
Thank you.
My question is,
Okay.
how you are planning to apply this thing? Because it's very hard to apply actinomycetes in the natural system, because you want to apply it in like a large volume probably. What is your strategy for that?
Sir, I can't understand. Can't hear.
Because basically your aim is to apply this to the waste, the area which is polluted, right?
Yes, sir.
So how you are planning to do that? You have like a source of culture. How are you planning to do that?
First, we are collecting the sample from the paper and pulp waste, waste industries, industrial waste collected, and then in the lab we are processing to the lab.
I was keen about the application. You have the bacteria, sorry, actinomycetes now. So
yes,
you want to apply it to the waste, right? For the degradation.
Yes, sir. Yes, sir.
How you are planning to do that? Like how you are planning to apply the actinomycetes to the natural source?
We are screening of the actinomycetes on the plate assay method. Who give a large zone of the inhibition, cellulase, xylanase, pectinase, then who give a large zone of the actinomyces, then we apply selected strain and we apply to the base, and then enrichment of the culture, and then enrichment process is started.
Okay. That's all.
So I have another question with you. So you are talking about the enzyme, right? Cellulase and cyanase. They are responsible for for reducing the pollutants. That's what your conclusion. Cellulase and cyanase.
Cellulase and xylanase.
No, actually, cellulase and xylanase, they are acting on polysaccharides, right? They are not acting on pollutants, right? Pollutant must be some other, right?
Sir, nahi samajh aa raha.
I think they are planning to use this to degrade the cellulose fibers which are washing out to the soil, washing out the soil. But
that's what you call as pollutants, right?
I think they are did it only very preliminary. I have asked this question because they have funding from government for this project. So that's why I asked how are they planning to apply this to the soil, because they have to have a plan for applying. Otherwise, you will not be able to apply. It's not like bacteria. It's very hard to do.
It's just an academic word. Thank you, Vidhi. Thank you.
Thank you.
Thank you.
Okay, any more questions? Okay, that's it. So thank you, Vithi. So we'll move on to the next poster. Code number PPFR4.
And I think she is absent.
Padmavati. She's absent?
Yeah, she sent a message to one of our conveners. She is not able. She's having some inconvenience. She sent the poster too, but she's not able to present.
Oh, I saw her in the participant list also
Yeah.
today.
Yeah. She was
Okay.
there. Now she got some problem.
So we'll mark it as absent. Okay. So PPFR4 is absent. And next is PPFR5. Dr. Sharad Biza.
He is also absent. Both are absent, 4 and 5.
Okay, so Shabana ma'am, the contest posters are over, I think.
Yeah, yes. Only
So,
two more is there.
Only two more, that is only presentation.
Yes.
Do we include that in the contest list?
No, they have asked for presentation only.
So, okay. So they are not part of the contest.
Yes.
Okay, chairs, please note the next two presenters are not part of the contest.
Yeah.
Their code numbers, the first presenter, PP1.
Yeah, okay, Edna. Okay, Edna, you can proceed. We have noticed.
Okay. PP1, Shruti Bajpai. Is she there?
Yes, ma'am. Am I audible?
Okay. Yes, you are audible. Can you please share your screen?
Shruti, you want to share by ourselves or you are going to share? Shruti? You have sent the mail to share. That's why I'm
Hello.
asking for. Are you sharing or shall we start sharing for you?
Ma'am, please use share because net is not coming properly.
Okay, okay. I am going to share it.
Okay, Shruti, is it visible to you? Shruti? Shruti? Hello?
She's there only, but I think network problem is there.
Shruti, we can't hear you. Okay, Shabana ma'am, shall we move on to the next?
Yeah, yes.
Okay, next is also not part of the contest. Code number PP2. Dr. Shiny KJ. Is she here?
She is also struggling with the network problem. Anyway, I am going to share her poster here.
Okay.
Edna, please find is she there or not, then we can conclude the session.
Okay.
Yeah, she's there. She's in the list.
Okay.
Dr. Shiny, are you ready?
Dr. Shiny, can you hear us?
She told she will be presenting.
She is there.
She is there.
Yeah. But she has gone now, I think.
She was struggling there for. She's getting in and out due to the
Okay,
network
is
problem.
Shruti here? Shruti? Shruti is there.
Etna, can you hear me?
Yes, ma'am.
Okay.
Hello, ma'am.
I just checked my audio.
Okay. Is this
Hello, ma'am.
is
Shruti here.
who is it? Okay, I
Shruti.
am.
Yes, ma'am.
Okay,
Have you shared?
Shabana ma'am, please put Shruti's poster.
Yeah, sure.
Shruti, is the poster visible? Shruti?
Hello.
Okay, is the poster visible to you, Shruti?
Ma'am, it's okay with me.
It's okay.
Yeah.
Okay. Okay.
Should I start, ma'am?
Okay, start fast.
I am Shruti Bajpayee. I am pursuing PhD in Bioprocess Technology under the guidance of Dr. Parag Nemade from ICT Mumbai Jalna campus. So my topic is cellulose nano. Hello.
Yes, Shruti, we can hear
Hello,
you.
ma'am. So, ma'am,
Yes, yes, we can hear.
all of us know, as all of us aware that dyes has become a necessary requirement in almost all the sectors of consumer-based products, ranging from textile, leather, paper, etc. But many of these dyes are synthetic and hazardous in nature, which cause serious impacts not only on the environment but also on the health and well-being of the individuals. This makes them a harmful pollutant. So there are many methods to remove the dye from the contaminated water, like adsorption, precipitation, photocatalysis, etc. But amongst the adsorption process is most common one due to its ease and efficiency. But the common adsorbent which is used for dye removal is activated carbon, which is difficult to generate in process. So the of my project is to generate low cost atomic efficient. So here
I think the connection is lost. We had only feeble signal.
Yeah, she's having network problems.
Yeah. Anyhow, I have gone through the poster.
Okay,
If
sir.
Shruti is available, we can ask 2 or 3 questions. That's it.
Yes,
Otherwise,
yes, sir.
otherwise we can just conclude. Is that fine? How about
Yes, sir.
Bashir?
Yeah.
Jasil, sir?
Yeah.
Yes, Mansoor.
Yeah.
I agree with him.
Yeah.
Okay.
Okay.
Let us wait for Shruti.
Shruti, shall we ask
Shruti?
questions?
Shruti? Shruti, can you hear us?
I think she is not available.
No, she's not available.
She's still there in the list.
But
her
she has got
connection.
some signal issues.
Yes.
So we will go to Dr. Shaini. Dr. Shaini is there. We will listen to her.
Okay. Dr. Shaini, are you ready?
hello.
Who is Shruti?
Yes, ma'am.
Shruti, the chairs have asked you to stop the presentation and they'll be asking you questions.
Shruti, are you there?
Yes, sir.
Okay. I just want to ask you a couple of questions. Like you are mentioning about carbon cellulosic nanofibers, right? Do you synthesize cellulosic nanofibers?
I don't think the connection is stable.
Yeah, it's not stable. It's fine, Shruti. It's fine. It's a nice presentation. Wishes you all the best. Thank you from my part.
Now call the next.
Yeah, that would be better.
Yeah,
We
Dr.
will go for
Shiny.
next.
Dr. Shiny. Is she here? Dr. Shiny? She's not here.
She's not there in the participant list.
Hello.
Hello.
Yeah, Shruti has.
Can I talk?
Yes, ma'am.
So, what was the question? So please repeat.
Mansur,
Sir, you
your
are
mic.
muted.
It's fine, Shruti. We have gone through your poster. It's very good. So you can do your further experiment. It's fine. Wishes you the best. Thank you, Shruti.
And I want to inform something that Dr. Shiny, she is not able to enter the Zoom room. So she informed me through WhatsApp that she is not presenting now. So that we are concluding this session, I think. Edna, please.
Okay, ma'am. I think all the presentations are over.
Yeah.
So, ma'am, you can conclude.
Okay. Thank you, Shruti. So this journey was started from I am sitting in front of this computer from 8 a.m. here till now. And I'm happy that we got about 75 responses, in which 14 were posters and the rest of their oral presentations. Contestant category, faculty, researchers were finished. Now posters are also finished.
Dr. Shabana is presently working at Ramanatagara Safi Institute, right?
Yes, sir. I am working there as a faculty in the Department of Microbiology.
Okay, my best regards to your principal, Dr. Inditi Goya.
Yeah, sure. I will convey, sir. Surely I will.
Okay, and it's nice to meet Bashir also.
Yes, Jasim.
We had a nice time.
Yeah,
Where are you, Bashir? Where are you now?
I'm in Czech Republic now.
Czech. Okay, very good. Nice to meet you, gentlemen.
Yeah. So we had a nice time with you judges also. I feel very proud and much, what to say, like refreshed. And it was very nice to sit with you also. You were very much kind towards all the participants. You were very patient. Thank you so much, both of you. Thank you so much. And I would like to welcome Dr. Jisha ma'am for concluding this session, convener of the program.
Thank you, Shabana. Thank you, Mansoor, and thank you, Jasim. You accepted our invitation in the last moment. But
it's our pleasure.
I know I have the freedom to ask you. That's why I approached you and immediately accepted the invitation. Thank you very much. And Mansoor, a special congrats. I heard that you became the vice principal.
I just came from the college after the administrative duties, and I didn't
I know. I know that you are very busy.
It's fine.
And congratulations for your A+ NAC accreditation also.
Thank you.
I know you are the person behind that also.
Thank you.
And so you two, both of you are very good in your respective fields. Congratulations for that, and thank you. Expecting
Thank you so
your
much.
kind cooperation in the futures also.
Oh, sure.
Okay, thank you.
Thank you, participants, also for your patience. Thank you so much. Good night. Have a nice sleep.
Okay, thank you. Okay,
You have to send the list. You have to send the
Tomorrow's
result evaluation report. Mansoor.
Yeah, what we have to send the first one only, right? The best one only, right? You don't have.
Contestant care.
Okay. So I will do that.
You can send it to the personal mail ID, mail or
Mail
WhatsApp.
ID or WhatsApp, it's enough.
WhatsApp it.
Okay. I will do it WhatsApp. That would be more convenient.
If you want to consult each other, you can do that also.
Yeah, sir, please include all the results. We can.
Oh, we have to do that. Okay.
Yeah, we have to keep it now for further reference and all those things.
So I have written it and I will just take a photo of it and I will send it. That's fine, right?
Okay, sir. It's okay. We need for documentation also. That's fine.
Okay.
Thank you so much once again.
Thank you. Good night.
Thank you.
Good night.
Night. So I
See
am
you all
stopping
tomorrow.
the. I am stopping this. I'm leaving the. I'm ending the session. The Zoom room I am ending. It was started from 8.
Okay.
We are closing this Zoom room.
Yeah.
Okay.
Thank you. Bye.
Thank you.
Okay.
See you all tomorrow morning 9:30.
Yeah, sure, ma'am. Inshallah. Thank you. Good night.
Good night, ma'am.
Shabana. Good night, Etna.
Good night, ma'am.